Dear colleagues, it's my great pleasure to welcome you to the Russian-Indian scientific webinar on marine biodiversity and blue economy. On behalf of the Russian Academy of Science, I would like to greet our Indian colleagues, participants of this webinar. Russian Academy of Sciences greatly appreciates your valuable participation in this meeting. I should say that our Department for International Cooperation and the Embassy of India in Moscow pay much attention to our cooperation in this particular area of scientific researchers. The main purpose of this webinar is to share current information on the problem of ocean exploration, inventory of marine biodiversity, and rational use of marine biological resources. The webinar provides an effective platform for Russian and Indian researchers to share experience and new ideas how to support productive marine ecosystems and save them for future generations. The program of this webinar covers most important topics in this area, including marine ecology, ecosystem functioning, coastal and deep sea biodiversity, marine biochemistry, marine drugs and pharmacology, biotechnology. I wish to all of us productive and interesting talks and discussions. Thank you very much for your participation in this meeting.
No sound.
Konstantin, could you please invite next speaker? Thank you.
Good morning, ladies and gentlemen in Moscow. Good afternoon and good evening. I'm sorry, but they didn't have sound for two welcome presentations. I'm sorry for these technical problems. Now they start our conference, and I would like to welcome all the participants of this India-Russia Scientific Conference, Marine Biology, Biodiversity and Blue Economy. This conference is scheduled for 2 days, and today they have presentations for 4 hours, in fact, and that in total for 2 days they will have 4 sessions. And today they have first session called Marine Biodiversity and Ecosystem Functioning. And I'm very pleased to see that they have now 40 participants from both India and Russia. I'm very happy to see Russian participants from various cities in the eastern part of country, Vladivostok, Magadan, from Crimea, Sevastopol, and from Moscow, and maybe from other cities. I'm not aware about this. I just would like to say a few welcome remarks. And first of all, I'm very, as one of the organizers, very grateful to the Embassy of India for joint effort in organizing of this conference, and to Ministry of Earth Sciences, Government of India, Council, National Scientific Council of India, and Russian Academy of Sciences, who jointly organized this India-Russia Scientific Conference. I'm very happy to see that very happy to see that in this difficult time, COVID-19 times, for last 2 years, then they didn't have opportunity to travel and to visit each other. They started very interesting program of joint webinars. As I'm acting as a representative of the forest branch of the Russian Academy of Sciences and the National Scientific Center of Marine Biology in Vladivostok, I just would like to mention that they already conducted a series of workshops and webinars in Vladivostok devoted to marine biology and other aspects. So several institutes of the forestry branch were involved in this activity. So that today conference is the continuation of this activity, and I hope that one day this collaboration would result in some, joint papers, expeditions, joint experiments, and I mean in more close collaboration. So I would like to thank all the participants who joined us today. Now I see 42 participants, and don't forget that tomorrow this conference will be continued. And again, thank you to all agencies involved in this organization. And I would like to invite the first speaker. So please, organizers, show us profile of the first speaker, Dr. Dmitry Petruk.
Konstantin, before we start, we have a welcome address. I will just introduce our Secretary of Sciences, and we will have his welcome address.
Okay, I'm sorry. So please, welcome address from India. I'm sorry. You have a floor.
Ladies and gentlemen, I have the honor to introduce Dr. M. Ravichandran, an illustrious scientist who has assumed the charge of Secretary, Ministry of Earth Science from 11th October this year. He has worked as a scientist in Indian Institute of Tropical Meteorology and also at National Institute of Ocean Technology. And he was the director of National Center for Ocean Information Services in Coex, Hyderabad, some time back. He is also the Vice President of Scientific Committee on Antarctic Research of International Science Council and a council member of various international bodies like the International Arctic Science Committee and Council of Managers of National Antarctic Programs, etc. Dr. Ravi sir has numerous publications, projects to his credit in the field of ocean development and ocean observation. He currently also heads and steers India's prestigious Deep Ocean Mission as Secretary Earth Science, Government of India. May I request you, sir, to give your welcome address, sir.
Thank you, Dr. Sisi. Dear Professor Andrei Adrino, Vice President of Russian Academy of Sciences, all participants of the India-Russia Scientific Conference on Marine Biology, Biotechnology and Blue Economy. A very good morning to those who are attending from Russia and good afternoon to those who are attending from India. First of all, I thank and congratulate Dr. Sisi, Science Counselor, Indian Embassy, and other colleagues from Russian Academy of Sciences for organizing this important virtual conference. As you all know, blue economy as a macroeconomic concept involves every aspect of national and international governance, economic development, environmental protection, and sustainability and international communication. Blue economy is an integration of sustainable development and green growth, and it needs compliance with the SDG 14, with the attribute focused on conserve and sustainable utilization of marine resources. As blue research expands with the world increasingly understanding its importance, policymakers and researchers worldwide concerned the ocean and coastal regions are demanding further and improved analysis of the blue economy. Towards this effort, this conference is very important. Marine biotechnology and its applications spread into many fields of social and economic life. Of course, the marine biotechnology is a knowledge-intensive sector. Public support for marine science, education and research may be the necessary in order to inspire greater application of innovations in marine biotechnology. There may be numerous other applications of marine-derived technologies and biological processes. I could see the conference covering wide spectrum of topics related to ocean biology, biodiversity, and its influence on blue economy. We need to understand the present diversity of the entire water column. As we are facing the warming world, the thermal stratification inhibit vertical mixing and the diversity pattern changes, and we need to understand and predict them well. Similarly, marine pollution, microplastics, acidification, and other external forces are contributing to degrade the health of the oceans, which we need to understand and protect them. Understanding and conserving marine genetic diversity is also essential, not just for the resilience of the species, but also for the economy. On behalf of Ministry of Earth Sciences, Government of India, I thank you all and send best wishes for the very successful conference, and we'll have more bilateral and multilateral collaboration for betterment of the healthy oceans. Thank you all.
Thank you, Dr. Ravichandran, for your welcome remarks. And I do hope that this conference, today conference, will be successful, and that is the next step of collaboration between Russian and Indian scientists. So before moving, I'm sorry again for technical problems and other problems in the beginning of the conference. And before moving to first speaker of the session one, I would like to mention that to all the participants that the duration of your presentation is 20 minutes, and please try to be not behind the schedule. Otherwise, they're already a little bit behind the schedule. So try to keep in the schedule, in line of the schedule of the conference. And if you would like to have some questions, please stop several minutes before the end of your presentations. Otherwise, we just move to the next speaker. So please keep in mind that you have 20 minutes for your presentation. Now, so please don't speak in the beginning, because first there would be small video profile of showing your presentation and your name, and please start your presentation after that. Okay, so we move to the first speaker of the session one, marine biodiversity and ecosystem functioning. May we have profile for Dr. Petru? Okay, Dr. Petruk, the floor is yours. Dr. Petruk, can you hear us? Can you start your presentation now? Dr. Petruk, Dmitry Petruk, can you start your presentation now? Can you hear us? I'm sorry, there seems again some technical problems. I don't see any response from Dr. Petruk. No, I can't see any response from Dr. Petruk. So in this case, they have to move to next speaker. Maybe after that they will contact Dr. Petruk, and he will be back with his presentation. I don't see him right now. So can we have profile for the next speaker, Dr. Jyoti Babu? Dr. Jyoti Babu, the floor is yours.
Hello. Am I audible, sir?
Yes, Dr. Jyoti Babu.
Yes,
Okay. Just a moment, sir. Just a moment.
Okay.
Yes.
Just a moment, sir. Some technical issue I to face. Just a moment, I'm correcting it.
Yes, of course, take your time. I'm sorry that I forgot to invite you for collective photo. So ladies and gentlemen, after this presentation, be ready for the collective photo. After this presentation.
Dr. Konstantin, we can have the collective photo now.
Right,
okay.
but
Okay, he started.
The doctor already started presentation. So after that, okay.
Sir, is it visible now?
Yes, it is visible. They can see and they can hear you. So please start.
Okay.
The floor is yours. You have 20 minutes.
Okay, sir. Thank you very much. Warm regards to all participants, and I would like to thank the organizers of this India-Russian scientific webinar and giving me an opportunity to speak. Since only 20 minutes are given to me, I'll straight go to the presentation and I'll try to stick to the time which is given to me. Basically, in this presentation, I will talk about the advantage of adopting an ecosystem science approach to conserve the biodiversity. And I will talk about a specific case of Gulf of Mannar. In this slide, you can see where is the Gulf of Mannar located and what are the major characteristics of Gulf of Mannar and why its biodiversity of Gulf of Mannar is so important. So you can see this is the Gulf of Mannar, which is in fact located between India and Sri Lanka. And you can see towards the western side, this Gulf of Mannar is connected to the southeastern Arabian Sea. And to the east, it is connected to the Bay of Bengal through Pak Bay, which is a very shallow area. So the northern sector of the Gulf of Mannar, in fact, this region of approximately 10,500 square kilometers is India's first marine biosphere reserve, which has astonishing biodiversity, especially the faunal diversity compared to the adjacent regions. If you see the diversity, how diverse it is, around 4,000 faunal assemblages are there in the Gulf of Mannar. Many of the species are listed in the schedules of Wildlife Protection Act of Government of India. So one can assume how much important the biodiversity is in the Gulf of Mannar. So it's a very ecologically very sensitive area. All the researchers knows about it. But what is not known is that what are the mechanisms, oceanographic mechanisms, which makes the Gulf of Mannar an unusually rich and diverse regions, which was in fact, it is a mystifying feature. And this mystifying feature, in fact, we have studied in detail, and in this presentation, I am just introducing those aspects. So here you can see in terms of faunal diversity, how much important it is. You can see from this left-hand side, different groups of faunal groups from Protista, and you can see up to mammals, and the number of species which are living in those areas. It is numerous in numbers. And more importantly, if you see to the right-hand side of this table, you can see many of them are actually listed as endangered or threatened group of species. So no doubt that this area is very, very important with respect to the biodiversity conservation. Since the time is very limited, 20 minutes, I may not be able to go into every details of this particular research results. But those who are interested may please visit this publication, which is actually a recently published study Basically, this publication deals why the Gulf of Mannar is a marine biological paradise and why it is so important to conserve the biodiversity of the region. Now you can see this picture. It's very, very important to to understand the actual content of scientific content of this presentation. So here I emphasize what is the importance of adopting an ecosystem approach to understand the biodiversity. So basically, this presentation will tell you to what are the ecosystem causes that facilitate an unusually rich and diverse fauna in the Gulf of Mannar, which is this area you can see. So basically, this is based on a seasonal study. 3 seasons we have studied: pre-monsoon, southwest monsoon, and northeast monsoon. Pre-monsoon is March to May here. June to September is southwest monsoon, and November to February is the northeast monsoon. In this picture, top panel, you can see the seasonal water circulation in this area. During June to September southwest monsoon time, this Arabian Sea water flows into the Bay of Bengal. And during the northeast monsoon time, if you see November to February time, you can see the Bay of Bengal waters are actually coming to the Arabian Sea. So there's a seasonal circulation existing, connecting between Arabian Sea and Bay of Bengal. And also you can see this is a major circulation that is actually happening south of the Sri Lanka, but at the same time, there is a feeble circulation. You can see not as strong as the basin scale circulation, but still there is a seasonal exchange of water masses between the Arabian Sea and Bay of Bengal through the Gulf of Mannar. So in this study, we have used all kinds of data sets in the sense that there are in situ data sets which covers physical, chemical, and biological aspects of the ocean. And there are satellite remote sensing data sets. And also we have used some of the numerical modeling applications to verify the observations in the in situ, whether it is getting reproduced in these modeling projections. So in situ data, if you see, we have used 2 data sets. First one was collected in 2010-11 period, southwest monsoon time. You can see these are the location red colored circles represent 2010-11 data set and 2018 data sets. You can see these white circles from the western boundary of the Gulf of Mannar and towards the Arabian Sea. So you can see many numbers of locations in each transect up to a 30-meter depth from the coast. So this is 2010-11 and this is 2018 data sets. And you can see what are the major observations in this particular study. And this is just some of the surface meteorological characteristics of the region. There you can see these are the surface wind. Every month, March, April, May, up to February, you can see very strong winds during southwest monsoon time, June, July, August. And more importantly, you can see the strongest winds are existing in this area. Compared to rest of the region, the strongest wind exists between in the Gulf of Mannar between India and Sri Lanka. Same is the case during northeast monsoon as well. December, January, February, you can see very strong or strongest winds in this area, which makes that region very turbulent. And if you the sea surface temperature, you can see March, April, May. March, April is the warming time, warm waters here. You can see surface waters, but by the beginning of June, July, August, September, there is a considerable cooling, especially very close to the coastline, which is a representation of the coast upwelling, which is prevailing in that region, which brings subsurface cool waters to the surface, and it is getting advected towards south. And it will even advect towards the Gulf of Mannar as well. That point I just wanted to highlight in this particular study. During northeast monsoon, if you see at the same way, there is a cool water mass that is coming from the Bay of Bengal to the Gulf of Mannar through the Parkway. So there is a clear signature in the sea surface temperature, how the seasonal exchange of water happens in this in the Gulf of Mannar region. this is another one measure of turbidity. You can see this is the Gulf of Mannar region. All through the year, the Gulf of Mannar waters are clear compared to the adjacent Parkway. Parkway regions are relatively turbid. Even along the southwest coast of India also, you would be able to see certain level of turbidity, but the Gulf of Mannar is almost very clear water. Gulf of Mannar has almost very clear waters throughout the year. So this particular ocean process, I am very sure that all are familiar. The most important biologically significant physical process that happens along the west coast of India is coastal upwelling, which happens during southwest monsoon period. So coastal upwelling has a positive as well as a negative effect on the biota. The positive effect is that it significantly enhances plankton protection. But the negative effect is that this upwelled waters, which are normally oxygen deficient, less than 2 milligram per liter, cause oxygen stress and seasonal wipeout of fauna in these areas. And there are cases of dead zones which are being formed along the southwest coast of India during the southwest monsoon time due to oxygen deficiency associated with coastal upwelling. So what is the common response of fauna to oxygen deficiency in this region? It is very interesting. There are 2-3 mechanisms or responses organisms can undergo. One is that those vulnerable organisms may die. Another option is that many of them may even attempt to adapt to the oxygen deficient situations, but it is not possible for all organisms to adapt. So many of the mobile organisms, fauna, they avoid and escape from oxygen deficiency. These are some of the responses of organisms, fauna, not only in Indian coast, but it happens world over, wherever coastal upwelling is prevalent. But what is unknown with respect to Gulf of Mannar is that there is no mention or there is no understanding about how this coastal upwelling scenario happens in the Gulf of Mannar, or is there a coastal upwelling existing in the Gulf of Mannar, or what is its implication in the Gulf of Mannar? So we wanted to understand really, first objective was to understand whether coastal upwelling is existing in the Gulf of Mannar or not. You can see this is a 2012 data, 11-12 data. This red colored spots. So these are vertical profiles of temperature collected with the CTDs. So this is the ideal normal upwelled water temperature, less than 23 degrees. You can see marked here. And you can see in all transects, in all these transects, the temperature vertical water column was much warmer than the expected temperature of the coastal upwelling. It conveys that there is no evident coastal upwelling that is occurring in this Gulf of Mannar. But please remember, this is an area from the literature. It is very clear that coastal upwelling happens all along the southwest coast of India up to Tuticorin. But further to the east, whether it is happening or not was really unknown. So this data shows that there is no clear cooling of a water column during southwest monsoon in the Gulf of Mannar, implying that coastal upwelling is not prevalent in the Gulf of Mannar. We did not stop there. In fact, we tried to understand the next indicator, which is the oxygen concentration, dissolved oxygen concentration. So same region, you can see here. This is surface and bottom waters, surface, middle and bottom waters, and less than 2 milligram. Normally we consider as oxygen deficient waters. And you can see nowhere in these locations during southwest monsoon, oxygen decreases less than 2 milligram. Or you can say that nearly saturated waters are there in Gulf of Mannar during southwest monsoon. Again, it supports the view that coastal upwelling is not existing in the Gulf of Mannar. Now, this is the data set in 2018, which we collected, represent these white spots. You can see white circles in all those transects. SP1, SP2, SP3, SP4, and SP5. You can see SP1, 2, SP5 here. This is 10-meter depth locations. This is 25-meter location. Then this is 45-meter locations. This is temperature profiles. And this is dissolved oxygen profiles. You can very clearly see that coastal upwelling is very prevalent along the southwest coast of India up to up to maybe not really up to the Tuticorin, but just before that it dissipates. When it approaches Tuticorin, you can see this surfacing of surfacing of cool waters as well as surfacing of oxygen deficient waters dissipates. So it clearly says that coastal upwelling is dissipating somewhere here, and there is no real coastal upwelling existing in the Gulf of Mannar. Now the question is, what is its implication? Before that, I will show you this figure. This is a satellite imagery of chlorophyll, which will give you a synoptic picture how the ocean physics behaves there and what is its implication on the biology. You can see during the southwest monsoon time, June, July, August, there is very high concentration of chlorophyll in this area, which is truly indicates the strong coastal upwelling existing in that area. Even this high chlorophyll patch is existing even in the Gulf of Mannar. So in the previous slides, I told you that there is no coastal upwelling, but chlorophyll shows very high concentration in those areas. So this just because the reason that there is a lot of advected waters, upward waters advected from this west coast of India, is getting advected and going through this area to the Bay of Bengal. So this is getting a lot of phytoplankton stock from the southwest coast of India as part of the seasonal circulation. The same way during the northeast monsoon time, November time, you can see a plume from Bay of Bengal is actually coming and occupying the Gulf of Mannar area. Essentially, it says that as a part of the seasonal circulation, there is enough supply of phytoplankton stock in the Gulf of Mannar. Okay. Now we can see what is the implication of lack of or absence of coastal upwelling in this area. This is a really geographically protected area from oxygen deficiency and coastal upwelling. So what happens is that you can see here, this is just a magnified view. You can see a lot of diversity. There are 21 very small coral islands are here. And what happens during southwest monsoon, the entire west coast of India, southwest coast of India is under the grip of oxygen stress due to coastal upwelling. But this is the region which is aerated, region with water column really aerated. So the advantage is that the organisms, fauna living here will have more diversity and richness naturally because they are undergoing a life which is under less stressful situation. So also, since there is an avoidance mechanisms, behavior is there for many of the fauna to hypoxia. Naturally, during southwest monsoon time, many of these mobile organisms which escape from hypoxia also may be contributing to the high faunal diversity and richness that is existing in the Gulf of Mannar. So what we stated in this publication is that the geographically sheltered environment in the Gulf of Mannar provide conducive environmental conditions to support high faunal richness and diversity compared to the adjacent areas. So also the Gulf of Mannar may be acting as a refuge of migrant fauna during southwest monsoon because many mobile organisms may be taking shelter in the Gulf of Mannar area to avoid very strong upwelling and associated oxygen deficiency along the southwest coast of India. So I'll just conclude this presentation. These are the major observations on this ecosystem-based study on Gulf of Mannar. He conclude that primarily, unlike the adjacent Indian southwestern shelf, oxygen deficiency associated with coastal upwelling imparting physiological stress to marine fauna does not occur in the Gulf of Mannar. Secondly, the Gulf of Mannar along the Indian coastline receives an adequate amount of primary food because plankton are the basic food for higher level organisms from the Arabian Sea and the Bay of Bengal through the advected water masses associated with the seasonally reversing surface currents. Thirdly, the Gulf of Mannar water has high transparency, aerated sandy seafloor, which are conducive environment for the growth of diverse corals and much sensitive fauna. And the astonishingly rich and diverse aquatic fauna in the Gulf of Mannar, we conclusively state that is a biological manifestation of a very friendly and supportive environment existing in the Gulf of Mannar. It is a fact that this Gulf of Mannar region has always been a sensitive geographical area for researchers and ecological conservationists. But basically, if you ask those people why this area becomes so diverse and rich, there was no clear-cut answer. So some may say that it is a transition zone between Arabian Sea and Bay of Bengal. So that is a major factor creating so diverse and rich fauna. And many others may say that this is the ecotone of Arabian Sea and Bay of Bengal. So those are something like very arbitrary observations because some of the imaginary conclusions. But this study clearly states that there are scientific processes which makes the Bay of Bengal, make the Gulf of Mannar very unique and support huge biodiversity and richness. So we conclude by saying that the Gulf of Mannar is a truly marine biological reserve in the region which need to be protected, and any major environmental modifications planning in the future should take this particular oceanographic aspect into consideration very seriously. So that's all about this publication. Thank you very much.
Thank you very much for your interesting presentation and about this ecologically important area of India. And they have time for one question. So if someone would like to ask a question, the floor is open. No questions. I don't see any questions. So thank you very much for your presentation again. And let's move to the no questions. Let's move to next speaker. And I would like to ask the IT team to provide us the profile of the next speaker, please.
Thank you very much. Thank you.
Sorry. They have to make a collective photo. So before they move into the next speaker, I'm sorry, I'm misleading you again. So please, everyone, make your video on so they can see your faces. And yes, now they can see the faces of everyone. And I will ask the organizers in Moscow to make a video, please. Okay.
Organizers, can you enable the videos of everyone, Inna?
Sorry, could you repeat, please?
They have blocked some videos, so kindly request the IT team to enable all the videos.
Операторы, есть кто-то заблокированный у вас? Пожалуйста, отключите блокировку.
Коллеги, я заблокирована. Чернова.
Еще раз?
Чернова. У меня звук есть, а видео нет. Заблокировал оператор.
Just hold on, please, everybody.
Yes.
Маренко Ирина.
So, okay, so I think now they can do the screenshot. So please, organizers, can you make a screenshot of all the participants? Or still some people are blocked? I don't see Dr. Pitruk again, and I think he still experienced problems with. Okay, one more time. Okay, thank you very much, everyone. So let's move to the first presentation, in fact, but I'm not sure if Dr. Pitruk is still able to connect with us. Dr. Pitruk, can you hear us? Can you start your screen? Can you start your presentation, Dr. Pitruk? I think he still experiences problems. I'm sorry for these technical problems. So let's move to the third speaker. In fact, Dr. Julia Zograf, please show us profile.
Hello, everyone. Can you see my screen?
Yes, they can see and they can hear you.
Okay.
Yeah,
Okay.
please.
Once again, hello, everyone. During the last decades, facts of numerous changes in marine ecosystems have been registered. Such changes caused by both natural fluctuations and anthropogenic influence. These changes lead to increasing of risk of ecological crises and catastrophes. In this regard, the ability to quickly obtain scientifically based accurate assessments of the current state and forecasts of possible development of situation is becoming increasingly important and relevant for managing the state of marine ecosystems. Obtaining such data and forecast is possible only with a detailed understanding of the organization and functioning of ecosystems at various spatial and temporal levels. Nematodes are the most successful metazoan of higher taxa in deep sea sediments, representing often more than 90% of the total multicellular animals abundance. Several thousand individuals and several tens of nematode species per 10 square centimeter of bottom sediments have been previously found. According to some authors, the high abundance and fast life cycle of nematodes determines their significant contribution to energy and material flows in benthic communities. The role of nematodes in the conditioning of bottom sediments is particularly important, and they participate in the formation of sediment texture, changes in the drainage regime, the amount of interstitial water, and dissolved gases. Nematodes are characterized by a wide variety of morphologies, life histories, and feeding strategies. Usually, they are small thread-like whitish organisms. However, some representatives are characterized by very deviating body shapes, sizes, and colors. And living nematodes can be bright and colorful. Many species of Desmoscalycidum have a strongly annulated appearance. Specimens of Draconematide usually have S-shaped forms, and Epsilonematide have an epsilon-shaped forms. The adult body size of free living marine nematodes can be less than 100 microns and more than 4,000 microns. The adults of Benthimermitide and Marimyrmitide can reach from 15 to 17 centimeters. However, typically free living deep sea nematodes are smaller than 1 millimeters and are considered in studies of the Meiabenthos fauna. At the same time, deep-sea nematodes are often found in the macrobenthic size fraction, and there they often dominate in numbers and are characterized by high species richness. Most marine nematodes have endobenthic lifestyle. However, some are associated with macroalgae, wood, and dead and decaying animals. Moreover, numerous non-parasitic associations between nematodes and aquatic multicellular animals are known. Nematodes from different taxonomic groups are found in sponges, bryozoans, and bivalve mollusks, as well as in actively moving animals like annelids, crustaceans, gastropods, and whales. Representatives of benthimermetidae and marimyrmetidae parasites the body cavity and internal organs of marine invertebrates hosts as juveniles and emerge in sediments as adults. Nematode can also parasitize some other nematodes and even unicellular organisms such as foraminifers. Despite of more than 90-year history of studying deep-sea nematodes, our knowledge about their dispersal, distribution, biogeography, ecology, their role in benthic communities remain limited, and the Northwestern Pacific is one of the least studied regions. Until 17th, only one species have been described for far eastern seas of Russia. During the next 30 years of studies, this number was increased and reached 257 species. It seems like a great number, but if we compare this number with Europe, we may see that only from the relatively small Adriatic Sea, 281 species were described. And only for Belgian part, this little one, Belgian part of North Sea, 445 species were described. Talking about the deep sea fauna of North Pacific, only one species out of 638 was known for Northwest Pacific. It is quite obvious that despite of many years of investigations, we are only at the beginning of the description of the real diversity of nematofauna, and we can expect to see a large number of descriptions of new species in future. Since 2010, joint Russian-German deep-sea expeditions were organized. And here you can see the spots where the samples were taken. It includes the Sea of Japan, the Kuril-Kamchatka Trench, and the Northwest Pacific deep-sea waters. As a result, a row of new species and genera were described. This fact of very poorly studied fauna often complicates the investigation of ecology and biogeography of nematodes. Increasing of the total number of samples, studying of new areas such as deep sea, shelf, hydrothermal community, systems under high anthropogenic pressure, using of new methods lead to appearance of significant amount of new information. An example of such area is the submarine Peep volcano, where the hydrothermal vents are situated. Here we discovered and described two new genera of free-living marine nematodes that were new for science. One of the genera belongs to family Oncolamidae and characterized by unusual mouth armature, where oncs have complicated structure with crown-shaped basement. Another genus belongs to family Ironida and characterized by the unusual copulatory system with post-glacial tubular supplements that is described for nematodes for the first time. This is just an example how discovering of new habitats brings expanding of our ideas of real biodiversity of nematodes. And now we have some questions that we have to answer in future. What is the true nematode diversity in the sea ocean? What are the mechanisms of their settlement? Is there any presence and influence of their geographic barriers on nematode diversity? What is functional role and significance of nematodes for ecosystem? And is there any introduction of new species of nematodes and the true scale and consequences of it? That's all. Thank you for your attention.
Thank you, Dr. Zograf, for your interesting presentation. I just would like to mention that the nematodes are a very important component of the meobenthic organisms and play an important role in the ecosystems and widely distributed in the world ocean. So they have time for several questions. Are there any questions from the audience? Questions? Questions, please? No one. I don't see any questions.
Thank you.
So thank you again. And unfortunately, I cannot connect with Dr. Petruk, and I would like to ask him again if he would be able to show his presentation. Dr. Petruk, can you hear us? Can you start your presentation? No, again, and I cannot connect with him. So let's move to next speaker according to our program. So please show us profile of the next speaker, Dr. Mishra.
I think I'm audible. Yeah, I'm Dr. Mishra.
Okay, you can start.
Sound you want to no, not sound. This one's here. Sure. It's okay? Am I audible?
Yes, they can see your presentation and they can hear you. So please start.
Okay, thank you.
You have 20 minutes.
Okay, so this is my topic is today on assessment of marine litter and microplastic pollution along the Indian coast. I am Prabhakar Mishra, working at VR Center of National Center for Coastal Research under the Ministry of Earth Science, and we are actually located at Chennai in the southern part of India. And this particular work started, I can say, that after as we know that Jembeck of US, he wrote a paper in 2015 in Science. Since then, this microplastics and marine litter issues, contaminations studies became very, got a very main attention. And since then, a lot of papers have been published, and all these estimates and a lot of calculations have been made how much marine litter or how much plastics are entering to the oceans. And in 2018, our Prime Minister signed India signed for coastal clean seas. And since then, we are given a responsibility to look into to make some of the research activities on marine litter and microplastics. And so as we know that in their earlier papers, they have indicated that basically the mismanagement of plastics, major river systems transport the plastic, and also the Southeast Asian countries are mostly dumping the plastics to the ocean. So But later, a paper recently published which says that most of the more than 1,000 rivers account for 80% of global riverine plastic emission into the ocean. So there is a change in the in the computation or the statistics or estimation of the data. So all these are based on not real-time data, based on the plastic production of a country, based on the management, how much is getting to recycle and how much is the probability that is entering to the ocean. But to contest or to have some data for each country, it is very important to understand exactly how we are going to, how this plastic problem is going to be more severe with time. And why we are so much concerned, as we know that major litter is, most of them are plastics. And this is what most marine litter is plastic, and that contains cigarette butts, food wrappers, straws, all are SUP, single use of plastics. And once it enters to our ocean, the thing is the plastic bottles I'm throwing today, it can remain to 10 to 12 generations. So this particular picture I've taken from the Trust Travels site, and it says that a plastic grocery bag or whatever it remains, it can go to the ocean environment and it can stay for a long time, and ultimately it will go to the, with the environment, and it will become microplastic from megaplastic to microplastics. And all this are, as we know, there are a lot of concern to our entire biodiversity, and a lot of the ghost net, the nets that are being abandoned by the fishing industry, that can become a ghost net. It can kill a number of animals. And so these are some of even the footprints of microplastic has been seen in the Mariana Trench, the deepest part of the ocean, in the tallest mountain, that is on the Mount Everest, even in the wind system, the atmosphere, even in some of the table salts, what we are using, that also contain a lot of microplastic, and even in the human placenta. These are some of the studies that have been done at different globally, and that the footprints of microplastic pollution is everywhere. And the major polluters, like contributors of these, are some of the corporate, means very high, world's most offenders from the all these big companies like Coca-Cola, Pepsi, and Nestle, so whatnot. And every 20,000 per second are being produced the drink bottles, and that can turn into some percentage are turned into new bottles, and a lot of plastics that is entering to the ocean, that we know. And if you look at the data of ICC, International Coastal Cleanup Report, you can see that cigarette butts, the single use of plastic is highly abundantly available in the beaches. And so once these plastics are getting released, there are two ways that we are producing directly the plastic pellets used in personal care products, that is primary plastics, and the secondary, what is getting fragmented from the megaplastic to macroplastic to mesoplastic to microplastic, and even now that is called nanoplastic, less than 100 nanometer, and the less than 5 millimeter to 100 nanometers are the microplastics. So all these are, we know that the sources are the rivers. So Indian contribution to the oceanic, to the plastic pollution, as per the Mazur et al. Paper, these are all estimated, but Indian rivers, small rivers, some of the rivers are contributing, they are in the position of 3rd or 15th or 9th or 25th rank. If you look at the rank, and we have a lot of river system, and it is very important for the India to look into our recycling system, our plastic problem system. No doubt, India is, in some of the slides I can show you, that India is also recycling major part of the almost 60 to 70% of the plastic waste. But ultimately, what usually happening, if you look at the statistics, if you look at the calculations, the national average plastic waste generation and plastic waste recycling, and you can look at that, something like, if we say that plastic waste littered by coastal districts of the India is something like 0.2 million tons per annum, And if you take the range, suppose 15 to 40% as per the GEMBEX 2015, the calculation that 30,000 to 0.08 million tons of plastic can enter to ocean. So after having said that, with the time in India, the plastic related to the plastic contamination studies has increased since most of the publication after 2018 or 19 has gone up. And most of them, they are focusing on microplastic studies in the beach, coastal, and different environment. And also looking at, if I consolidate the entire research on MP pollution on different environmental components, there are some studies which I can say that if you look at the global also, the contamination in some cases are comparable with the global values. And many of the plastics, like you can see that sediments are having something like 87.4 items per kg on average, and similarly water. So these are some of the studies that has been done earlier by Indian scientists, and I have just consolidated about our environment. And most of the sources are from the river runoff and multiple sources, like multiple activities like fishing activities or beach tourism activities. And mostly the plastic, microplastic, what is coming from the single use of plastics and polyvinyl chloride. And 2018, since 2018, on the NCCR, the National Center for Coastal Research, has started a number of, activities that basically to quantify from the source to sink, and how this microplastic is going to interact with the microplastic, going to interact with different types of biota, and also some of the modeling activities. And recently we have started also using the remote sensing, GIS image analysis, and application of artificial intelligence and machine learning. And we have also initiated a lot of collaboration as there was a talk of collaboration. So we have already initiated collaboration with the UK, with the Arctic, for the Arctic Ocean, for the Norway, with Japan, Jamstick. And also recently we have also, the talks are going on, the workshops are going on IPOI. So, and at the same time, we also initiated a lot of education awareness program along the coastal clean sea campaigns, International Coastal Cleanup Day, how to bring low-cost technology. And these are some of the snapshots. So what the work we are doing, we are having here, the place where we are sitting, we are having 3 vessels. And we are equipped with some of we go regularly, we monitor the beaches, we monitor the coastal waters, and we collect the samples. And this gives an idea to, that means we have observed that the highest concentration of MPs are getting closer to the river mouths. And season on the seasonal basis, if you look at that, most of the time the rainy season, or which is called the monsoon season in India. So there the plastic is more, the microplastic abundance is more. And similarly, the most observed plastic, as I mentioned, polyamide and polyethylene are some of the, and coming mostly from the fishing activities and riverine inputs. And similarly, these are some of the self-sediments and surface water. If you look at also the highest, how we are getting something like near to the river bed or near to the lagoon environment, where a lot of fishing activities takes place, which are being the plastic nets that are being thrown or that are being disintegrated and later become microplastics and remain in the and settles in the sediment. And very interestingly, we observed that in both the cases, water and sediment, the lower size of particles are higher in numbers than the larger size particles. That means the disintegration is going on, and this is significant in the sense that will adverse effect on organisms, because microplastics are dominant. That is more than 50% in both the surface water and sediment. And also we looked at the fish species as well as some of the benthic invertebrates in different places of Cochin in South India, as well as some of the bivalve and penna viridis, what we observed in Chennai. We got there are indications of polystyrene particles of thread-like fibers in benthic fauna, as well as polystyrene in polymers in soft tissues of the bivalves, of the Pannaviridis, and each means some of the 7 species and some of the fish species, we found that there are 3 to 6 numbers of different sizes of microplastics are present. And even on the west coast also, we also had studied seeing the floating plastic debris in the Arabian Sea, and these are some of the results which I'm showing that it has come plastic debris when compared to other studies in the Indian Ocean, but it is the abundance of the occurrence is low. And even in the deepest part of the ocean, like at 10,000 meter, at 1,000 meter depth, if you compare at 200 meter, we had samples from different depths of the ocean, and we also found there are the particles present which are origin from the different polythene bags, or there is maybe the fishing nets or laundry effluents and weathering of larger particles. And what happens usually after a flood, this is one of the scenario along the beach, entire beach gets flooded with the debris, marine litters. And we saw that debris are coming from the land. So it is very important that management part on the land is very important, how we are going to manage our discarded plastics. And some of the activities we have taken along the International Coastal Cleanup Day, and just like finding out different beaches, how much garbage that is being collected, that is being abandoned, and they're making use and raising awareness among the school children, college students, involving them in cleanup activities. So all these observations we saw at different places when you conduct that, the recent data looks like there were 62 to 65, means 50 to 70% of plastic litter. Plastic is the main source of littering on the beaches. And similarly, some of the low-cost scrap that are being used, like in Chennai or in another city called Visakhapatnam, and they have found that almost 2,000 metric tons of plastics in each. Means in case of Chennai, we found that 2,000 metric tons of plastics are out of 20,000 metric tons of litter in 1 year they could trap. So these barriers that can, by using this type of traps, there is an attempt. These barriers can be trapped with the nil cost of energy, and that can be also recycled. So though India has a number of legislation, no doubt since 1986 we have Environmental Protection Act, and since there is a 99, there is a plastic manufacturer sales and uses rules, but still this management, the plastic in the environment is coming as a mismanaged due to mismanagement, and ultimately in 2022, India has taken a pledge that will completely ban the single use of plastics. Though the implementation part comes with another ministry called Ministry of Environment, Forest and Climate Change, but we have to see the after effect after 22 whether we will be able to actually contain this single use of plastics or not. And so the solutions are very clear from this, that whatever the segregation of the waste to the source, responsible to the manufacturer or seller, adoption of beaches by different organizations or to clean up activities and educate, and means putting the citizen, that which is called citizen science, involving the citizens, and the deployment of low-cost traps and a biodegradable material we can introduce, and we can change, stop using the single use of plastic completely, and ultimately one marine litter policy which we are now trying to draft, that is very important. And also because it involves a lot of circular economy, it is very important that we have to look the opportunities for a circular plastic economy. And the current status in India, that plastic management, as I said, that almost these are all managed mostly by the very poor section of people, as well as there are no doubt there are also a lot of industries are also involved. but the 2022 such as plates, cups, cutleries are going to ban, and we can say that how it is that can be reflected or not, and the municipal solid waste management system, how we are going to improve, all these things has to be looked into. And these are some of the recommendations that we have made for the short term, medium term, and long term. And these are nothing the same thing like effective follow-up of extended producer responsibility, implementation of anti-litter regulation laws and their efficient enforcement, ban on or regulate the use dangerous activities during plastic production, encourage purchase of products manufactured using recycled materials, build internal reprocessing capacity to minimize dependence on global markets, make funds available to start working on developing materials alternative to plastics. So, so these are some of the suggestions we are making to the, we are putting in our draft. So this is very important for the management of the plastic in the country like ours. And we have also initiated the Ministry of Earth Sciences already initiated a Sagar Swachhata or Clean Ocean Mission involving different organizations under the ministry. So the objective is to take the different areas like beach, river, sea surface, sea force, and try to the approaches beach adoption or regular cleaning or floating traps or engaging fishermen for litter collections, like to clean our oceans. And though already government of India started in many beaches, the regular cleaning activities have been initiated, but is very importantly how we are going to generate an awareness among the public, as well as how the more involvement from the technical tools and technology involvement can be done to clean our oceans and rivers. That is one of the biggest challenges, so that we can really make the blue economy also successful, and also will achieve the clean sea emission. This is our team. Thank you all.
Thank you, Dr. Mishra, for your interesting presentation. This is really important problem for all countries now, the plastic pollution and microplastics, and the increase of the number of publications you have shown in the beginning. I think it is very characteristics for all countries involved in this in these problems. Unfortunately, they don't have time for questions, and I'm still trying to understand if they will be able to hear the presentation from Dr. Petruk, but no information as yet. So I would like to ask the IT team to show the profile of the next presenter. Good morning and good afternoon and good evening, everyone, again. So it's now time for my presentation, and I would like to tell you today about the important issue as related to the global change and global climatic and environmental change. That is the ongoing and expected changes of marine biotas, mostly on the example of mollusks in the Sea of Japan. But you know, the past is the key to the future. So for that reason, I would operate in my presentation, my data and data of other researchers about the recent changes, Holocene changes and past migrations of biota in the Sea of Japan. So this is an outline of my presentation. I would briefly introduce you about the biogeography of this area and Holocene history of the molluscan fauna, global warming and biotic migrations. And finally, I would talk a little bit about the expected ecofaunal changes in this area. So for those who are not familiar with that area, they call it Northeast Asia. I would like to tell you this is the Sea of Japan is surrounded by 3 countries, Russia, Korea, and Japan. It's kind of Mediterranean Sea for us in Northeast Asia, and also you know, to be politically correct, that it is called the East Sea in Korea. So sometimes you can see in regional publications both names, Sea of Japan and East Sea. So the Sea of Japan is very special in its general characteristics. It's a marginal and deep sea basin enclosed by an island arc. It is rather isolated by shallow straits. The maximum depth of 200 meters. That means that in the geological past, it was really isolated during the glacial period. And also that the maximum depth of the sea is over 3,000 meters. That means that it is quite deep sea and it has a real deep sea fauna. But the most peculiar characteristics of the sea that they have strong ocean currents. They have currents in the form of ocean currents. That means they are very strong and unusually strong as compared to other marginal seas. So there, to be brief, I would like just to tell you that this sea is very special also in the surface water temperature. The northwestern part of the sea is approaching to the Arctic conditions in winter, because in February it is observed the formation of pack ice and ice in some semi-enclosed bays, while in summer it is quite warm. And the southern part of the sea or southeastern part of the sea is quite warm. It is washed by the warm Tsushima current, and it has nearly subtropical fauna. So this very special area in the sea that in the world, like in the south of the sea, they can see nearly subtropical conditions, and in the north of the sea, of the same sea, they can observe the nearly Arctic conditions, at least in winter. So as for species richness of bivalve mollusks in this area, I just split it in 4 areas, which is this is Russian part called Mid and North Primoria. This is name of the province here. Southern Primoria and South Korea. As for North Korea, they have very limited data. And they can see there is increase of species richness or number of species in this area, from northern part of the sea to southern part of the sea, reaching over 300 species. So if we look at the distribution of warm water species, namely subtropical species, they see also very clear increase of a number of warm water species from north, from 9 species to over 100 species. And if they look at the distribution of tropical, subtropical species, even warm water as compared to subtropical species, they can observe that in the south part of the sea, as I just told you, there are nearly 100 warm water species living, while in the northern area, only 1 species may be of doubtful identification. So if they look at different localities or different areas of the sea, they can see that the species diversity varies between less than 100 species to over 200 species. But at the same time, as I told you, these contrasting conditions between the winter and summer makes the fauna is very rich, in fact. So if they take together southern and northern part, this is Amurski Bay near Vladivostok city, and you see in winter, this is big bay completely covered by coastal ice with a thickness of up to 1 meter, and the temperatures go down to minus 2 centigrade degree. This is the same area in summer, while in summer, the sea surface temperature in this area can warm up to 28 degree in August. So that makes the subtropical conditions sufficient for reproduction of these species. So it's just, you know, the normal landscape in this area, in Russian area of the Sea of Japan. So if you look in 2 parts of the sea, western part or continental part and eastern part, they can see the difference between these 2 areas. But this is a number of families, but currently if we combine this data for both parts of the sea, they can estimate the bivalve species richness in the entire Sea of Japan or East Sea at a level about 600 species. You know, it is very high because if they compare the some richest areas in the world for the in terms of biodiversity of mollusks, it's China and Japan. It's nearly half of species richness living in China and Japan. So there are, as I just mentioned, there are discrepancies or differences between eastern and western part. The eastern sector is more species rich, and also the degree of the penetration of tropical, subtropical, and subtropical species to the north is higher due to the warming effect of the Tsushima current. This illustration shows you the species richness in some of the most biodiversity rich areas in the world. For example, this is the Golden Triangle, you know this area very well, or Western Australia, Eastern Australia. So the 600 species is very high. Even in these tropical areas around India, they have less species than living in the entire East Sea. So now I move to the Holocene migration of mollusks because this data can provide us some key or model for future changes. So the Holocene period, it includes the last 10,000 or 11,000 years, and it was a time of profound climatic and environmental changes. Of course, this is reflected in modification of faunal assemblages. I would like to take as example one of the most important period in the Holocene, so-called climatic optimum of the Holocene. In fact, there were several climatic optimums or several maxima of warming of climate in the Holocene, and it is like a series of warmings. But in fact, the climatic optimum of the Holocene, it is 5,000 or 6,000 years ago. It was most important. But the Holocene, you know, it's a very tiny epoch covering just last 10,000 years as compared to the entire geological scale. So at that time, at climatic optimum, the climatic estimates shows that this period was characterized in the Russian Far East by the annual average temperature excess of 2 to 6 degrees centigrade and more humid conditions. And of course, the sea surface temperature in the Sea of Japan were higher as compared to the present. So this time is a perfect model for the future changes. This is several comparison of different data for different areas of the world. So, but anyway, it is showing that this time is climatic optimum, the temperature was higher as compared to the present one. So then the consider the changes in the fauna. They mostly talk about the environmental faunas because, you know, the environmental faunas or faunas living in semi-enclosed bays in environments, it is very sensitive to climatic changes. And also in temperate latitudes, the environments often show a significant discrepancy between summer and winter temperature. So they it makes possible to coexist species of different origin. And you see there are many bays in this area, and so they served, in fact, as refugia for the warm water species during the coolings. In fact, this is two big bays around Vladivostok cities. You see there can be subdivided into outer part and inner part, and inner part is more warm water, in fact. So there are many coastal plains. In fact, in these coastal plains, they represent in the past, they're just ancient bays. And then they look at the, you know, some fossil data. They see many subfossil shells they can find. And dating of these shells shows they belong to the climatic optimum of the Holocene. So this is a scheme of the present day system of currents in the Sea of Japan. And they see there is a number of cold currents flowing from the north, and there is a warm water Tsushima currents with several branches. So they expect that in the Holocene, these currents shift to the north, warm water currents shift to the north. That was possible to bring more warm water species to the northern areas. So in some areas, like in Russian-Korean border, they can observe the storm gravelly deposits with abundant shells of some extinct species, of course, locally extinct species. So these species lived here during the climatic optimum, but now became extinct. There are at least 3 species in this area became extinct in the late Holocene coolings. There are some species of blood cockles extinct, and also there are some common species of bivalve mollusks from the mid-Holocene deposits in this area. They are mostly warm water species, and they are now can be considered as a relict or immigrant species in this area. And these species are now very characteristic for tidal flats in south and west coasts of Korea. If they look at this map, so they can see the changes of the biogeographical composition of the molluscan fauna along the Japanese coast, and they can see that there are many warm water tropical subtropical species in the south and less species living in Hokkaido. So radiocarbon dating of these Holocene deposits show mostly the mid-Holocene age. So they can see that there was a shift of the northern boundaries of distributional ranges of warm water mollusks in the Pacific coast of Japan. And these species lived in the Holocene in Hokkaido. They now became extinct and shifted to the south due to the cooling. There is a distribution of some warm water bivalve mollusks. And you see these areas, the distributional range in the Holocene, and now they became extinct. So then new warming happens, so they can expect that these species could penetrate to this area. There are some differences in local distribution of these species, and there are many records of these warm water species in Peter the Great Bay, both fossil and recent. So again, this area between the Russia and Korea, near the Russian-Korean border, and they can easily see that there is a series of lagoon or lakes here, some fresh water, some saltish water, brackish water. But before it was the big sea area with normal salinity, and so there are some data from boreholes, and they could reconstruct the changes of the fauna in this area. So usually in the beginning, this area was alluvial stage without any marine animals, and then was formation of shallow water bay with muddy bottoms and small gastropods, and usually formation after that came formation of oyster beds, and then the sea water lowering, there was disappearance of oyster beds, and again the transformation of the area into lagoonal and estuarian environments and the fauna. So the process of the changes of marine fauna or marine biota includes also marine invasive species. This is the ongoing changes because the biological invasions in marine environmental is very important process in many countries. Also the bioinvasions create so-called new ecosystems, novel ecosystems, and they contain the new combination of species, and they cannot predict how they would act in future. So the area of Peter the Great Bay, northwestern part of the Sea of Japan, they have many ports, so they identify and detected the pathways of introduction of marine invasive species in this area, mostly through the shipping and ballast waters. They don't have invasive species in aquaculture, no intentional introductions, but many intentional introductions in China, and also due to the climatic changes. So there are vectors of introduction of these species from Asian countries, also the trans-Pacific vectors of introduction of marine invasive species from tropical areas, from the United States. So over 16,000 ships enter ports and harbors in Peter the Great Bay in this area I'm talking about. And many of them operate the international lines. A majority of ships go to Vladivostok port. And so this massive traffic, of course, it favors the introduction of alien species through fouling communities or release of ballast water. Another port is Port Vostochny in Nakhodka Bay in eastern part of Peter the Great Bay in this area. It's also quite international. So they can expect there is a lot of release of ballast water in this area. So several years ago, they have published a book about the marine invasive species in this area as a joint effort of Russian, Chinese, Japanese, and Korean researchers. And as for mollusks, I would like to tell you that at least there are two imported immigrants or alien species introduced into the Russian waters in 1970s and 1990s. And Mediterranean mussel is very well-known species living throughout the world. It first was identified in Peter the Great Bay, but later it spread into the northern areas of the Sea of Japan and even reached the Kuril Islands. There are some marine invasive marine bivalves in the eastern Korea, including Mytilus galliprovincialis, Mediterranean mussel, and some scallops. And you see this is a Vladivostok and Vladivostok port. So the number, over 50 species they have identified as non-indigenous. And you see the diversity of richness of this fauna is decreasing and increasing then the approach to the port. And the same port area, Nakhodka, you can see the same trend in this area. Because this area is very well taxonomically studied, so they have many historical data accumulated for last 100 years for different group of animals. They have a very detailed catalogs for this area, and they studied in detail marine fouling in this area. So they can easily identify the new species arrived in this area as immigrants. And so among them are very important barnacles, like Amphibalянus improvisus. You see the biofouling of the ship hulls. And also these species became very abundant in this area in the intertidal area. And you see it can be found nearly everywhere in Amurski Bay. So in this slide, there are many, you can see the many larvae they have found in ballast water. Of course, it doesn't mean that every larva can settle and can grow in this area, but anyway, this is big potential for them to become established in this area. Also, there are some examples of invasions of subtropical gastropods in this area, Opista branch, new species, as fish fauna is very well studied in this area. So they have already over 50 species of subtropical fishes arrived to this area. Jellyfish, like this Ropilema, a jellyfish was first recorded along the coast of our province in 1999, and then became an object of commercial harvesting. But its natural distributional range is located in tropical and subtropical waters. Also, there are some sea reptiles coming here in summer in this area. So based on this data, based on the historical data from the Holocene migrations, and based on the ongoing changes due to the human activity, due to the climatic changes, they can expect or they can predict of some biotic changes in this area. So they can find out and they can name, in fact, the potential or expected marine invasive species of this area. Like there is prediction about subtropical fish area due to the intensification of warm currents in the Sea of Japan. And there are some prediction about the potential invasive astridians, barnacles, bivalve mollusks, because these species were already introduced into the coastal waters of Japan. And of course, based on the Holocene data, they can predict some invasive species of mollusks. So that is, some species became extinct after the Holocene warmings, and with the ongoing warming of the coastal waters in the Sea of Japan, they can predict, they can reappear, they can again settle in the same areas, in fact. So there are some ecological and biological consequences. They are not only related with immigration of species, but also with the expansion of habitat area and increasing of population density of a number of already established warm water subtropical species. And they are getting the new dominance in bottom communities. For example, oyster beds are likely to become more flourishing. So the oyster community occupies a major part of Peter the Great Bay, but they expect that these species would more flourishing in the future in this area. Based on the data, based on the presented data, they can expect that more than 10 species of bivalve mollusks would immigrate to this area, to the northern Sea of Japan from the south. And the maximum shift of northern boundaries of distributional ranges of these species would not exceed 1,000 kilometers. At that, ladies and gentlemen, and I'm afraid they don't have time for questions. So let's move to the, maybe for one question. They have time for one question, in fact. Any questions? No questions. Unfortunately, they still don't have any news from Dr. Petruk. I'm sorry they missed the important presentation. So let's move to the next speaker of today's session. And first, please show us profile. Hey, Dr. Kubelio, they can't hear you. I'm sorry, but they can't hear you. I see that your microphone is on, but they can't hear you. Maybe there are some technical problems with your computer.
Dr. Shireen, without wasting much time, if you can re-log in again, just log out and log in again, please.
I'm sorry, but they still can't hear you. No, they can't hear you. There's some technical problems. Maybe you can try to fix your problem, and while they move to the next speaker, or you can try once again. Okay. Dr. Diana Solovyova, you can start your presentation. Okay, you have 20 minutes.
Well, good evening, everybody. And I am inviting you to the frozen Arctic from the nice warm seas of India and actually nice warm. It is a great bay. And I will talk about Chaon Bay of East Siberian Sea and how its ecosystem supports the birds of conservation concern, and a little bit about our application for marine protected area if needed. And where is the Chaon Bay? You can see the sign, this red sign in the circumpolar projection map. And you can see the map of Chaon Bay in the left lower corner. And the bay is pretty close. For the long time, the bay was considered as a refuge of warm aquatic flora and fauna. And it was considered that this some strange, unusual warm piece of the Arctic is remained from warm episodes of the past. But just the last year, the mystery of high biodiversity, unusually high biodiversity of this place was discovered. And I download this image from the Illichov Pacific Oceanological Institute, and I'm so happy that I would hear the presentation of Dr. Alexander Charkin from this institute, who actually discovered, I believe, the quite an interesting phenomena of submarine groundwater discharge, the hydrotherms in the Chown Bay. And these hydrotherms located here, and with the current, they distribute the warm water in the East Siberian Sea. And so the mystery of the high biodiversity, usually southern biodiversity, is solved. But you can see that still the bottom temperature is quite low because it's an Arctic. And Chown Bay special, what the bay is known for. It's a western margin of laminaria of brown alga. West of this area, there's no laminaria yet, but we still have the blue laminaria. The tones of blue mussels, Meteorocedulus and Trochilus, is in the Chown Bay, nothing like this west of us. And the Chown Bay famous fish, Dolly Varden trout, Cervelinus malma, is among the largest in the world. And you can see this fish is very important for the local people, for their consumption, for the survival. And this local man is carrying the fish, which is very fatty, very delicious, and which is like 11 kilos. And going up to by the food web to the birds who consume this fish. This is the result of our ground surveys on the west coast of Chown Bay. And here is the distribution of 4 loon species, starting from yellow-billed loon, Pacific loon, red-throated loon, and the Arctic loon. All 4 present in this small area, and it's a unique place in the world where you can see such a density and where you can see all the 4 species of loons. And one of them, the yellow-billed loon, is considered as nearly threatened by IUCN, and it's also protected by Russian law, by Russian Red Data Book, and it's actually an endangered bird. And if you look at the maps, here is a map of aerial survey done along the Arctic coast of Russia by our colleagues from Severtsov Institute of Ecology and Evolution in the last 3 years. And they reported the density of this yellow-billed loon as 0.0002 individual per kilometer. And in the Chown Bay, in Katit Peninsula and Ion Island, they reported the density which is 10 times higher. So it's actually a refugium for this species. And it's a place very important for it. Other birds of seabirds, sea ducks of conservation concern are eiders. The common eider, also considered as nearly threatened by IUCN. And the Chown Bay population is about 5% of total Asian population, despite its very small place, actually. And the spectacle eider, protected both in Russia and United States, has a breeding site here. And all 3 eiders can be found in this yellow square, including the king eider. So it's another important place, interesting place to study niche differentiation in 3 closely related species of the same genera. And other birds of conservation concern are brants, and the extremely endangered population migrating to Japan is using this place for stopover. It's been convinced by Japanese colleagues by VLAT tracking, and up to 15,000 of brants gathering during the autumn migration, autumn staging in the west coast of Chown Bay. And this ecosystem, which is salt marsh, is important for brant to feed. And what is the potential threat to the ecosystem, to this quite a unique, everyone is telling about unique ecosystem, but this ecosystem is quite a unique actually, is a proposal for building a new port for uploading the copper ore. The new port is planned here. This red square shows the proposal facilities of the port. It's like 5 kilometers per year, and then dredging because the Chaon Bay is rather shallow at this place, and the storage of the ground after dredging, and year-round traffic with ice breakers. And I was preparing the ecological assessment to not build this port and use existing ports of Barkumey, Abandon, and Pivek city to upload the ore. But if the company would not pay attention to our application, we suggest some protected area to conserve the species of concern, especially bird species, but also a number of invertebrates and floral species, including Iona Island, which is already a nature monument, including the Ibichana Island, which is a game refuge, and including important staging site of Lesser Chaon Strait, of Katik and Ion Peninsula for yellow-billed loon, and the Raucho River, known for the largest breeding population of lesser white-fronted goose, endangered and also protected. But if we can convince the company, which is Kaz Minerals company, Kazakhstan registered, to change their mind and not to build a new port in the Chaon Bay, nothing of this would be needed, because already this area is well protected by complete remoteness and quite neglectable human populations. It's less than 1,000 people living around this bay, and it's well protected, and it doesn't need any special attention and protection unless the company decide to build a port. So I'm seeking the support from Russian Academy of Science to our application to the company do not destroy the ecosystem of Chaon Bay. And thank you very much for listening of my short presentation.
Thank you, Dr. Solovyova, for this presentation, and they have plenty time for questions. So if there are some questions from audience, please show me. Okay. I don't see any questions. Thank you so much again. This very interesting environmental problems you discussed. And so let's come back to the previous speaker. Dr. Sherin, can you hear us?
Yeah, I can hear, whether you can hear me?
Yes, I can hear you, and I hope that the rest of the participants can hear you too.
Yes, yes. I'm very sorry for the technical problem we had.
That's fine, that's fine.
Yeah, so now I'm putting in the presentation mode.
Mm-hmm. Okay, you have 20 minutes.
Yeah, yeah. So first of all, I would like to thank the organizers of this scientific webinar for giving me this opportunity. And I extend my warm wishes to all the colleagues from Russia and India. So the topic of my today's presentation is marine living resources research in India, and the status and the trends with particular reference to deep sea resources and the related research activities that is being carried out in my institute, the Center for Marine Living Resources and Ecology, which is under the Ministry of Earth Science. And this center is situated in the southernmost part of the country, that is Kerala. You can see in the slide. Okay, so as you know, the center, which is under the Ministry of Earth Science, is dedicated to study the marine living resources of the country. Thus, next slide. Yeah. So formally, the vision or the mission of the ministry is to provide services for weather, climate, ocean and coastal state, hydrology, seismology and natural hazards, to explore and harness the marine living and non-living resources in a sustainable manner for the country, and to explore the three poles of the earth, Arctic, Antarctic and Himalayas. Thus, to further the advancement of the research in ocean, atmosphere and land, we have dedicated institutes undertaking relevant research programs. So coming to our center, which was established at Kochi in 2001 by upgrading the erstwhile Sagar Sambada cell with exclusive facilities for implementing the marine resources program. The center has the following mandate: to develop management strategies for marine living resources through ecosystem monitoring and modeling, evolving, coordinating and implementing the national as well as regional R&D program in the field of marine living resources and ecology through the effective utilization of our vessel 4RV Sagar Sambada, and strengthening the research on the marine living resources and ecology, including the establishment of a referral center, particularly for the deep sea resources, and also coordinating the national program relating to southern living resources, Southern Ocean living resources. Now in this slide, you can see a historical account of the marine living resources studies in India. As you know, the modern oceanography began with expeditions in HMS Challenger, which navigated the which took part circumnavigation around the globe and even to Antarctica. And during these expeditions, they gathered temperature, current, water chemistry, all the information about all these parameters, and studied in depth about the ocean floor deposits. And also the result of this expedition, which has come out, has around 30,000 pages of reports, which has took almost more than 25 years under the great leadership of Sir John Murray. And the discoveries during these expeditions also paved way to understand more about, Mariana Trench, then Mid-Atlantic Ridge. And these exciting expeditions have encouraged the other countries also to take many oceanographic expeditions. And you see in Indian Ocean also during the 18th century itself, more and many expeditions were conducted in the Sea of Lakshadweep, Bay of Bengal, Andamans, and Arabian coast and Gulf of Aden in the vessel Mabahis. And further following these expeditions in the Indian Ocean, International Indian Ocean Expedition took place. This has encouraged many countries, almost 13 countries took part in the expedition with where 40 research vessels have joined. And it is very encouraging to state that the results of these expeditions, to study more on the results of these expeditions, institutes such as institute which is solely exclusively for the marine living resources, many marine institutes have been established in India. Now, let me see what is the significance of marine biodiversity inventory in a national, both in national and international perspective. You see, the biodiversity information is being gathered from the primary data as well as from the secondary data. Primary data means it is from the in situ survey using our own trawling, our own vessel, and also from the diving operations. And the secondary data we collect from the global database and other published literatures. And these georeference data of marine biodiversity records, that is, georeference data, two things are very important. That is physical sampling and systematic experts. So spatially and taxonomically resolved data of marine organisms are thus collected very systematically. And this may help or assist not only the researchers in the field, but also the policy makers who may assess the biodiversity changes, to understand its changes, and also in the perspective of knowing the biodiversity loss. Then it goes, the information will goes to the people who are involved in the making assessments on the environmental impact assessment, then invasion of alien species, marine protected areas, whether the species is, their geographical range, whether there is an extension or there is a shift in the species regime, and there is any variation in the composition or diversity assemblage pattern. So all these informations are very important to be gathered. That is, documentation and inventarization of the marine biodiversity records are very important, and this will go to a global data sets in a very systematic manner in an internationally accepted format. It goes to biodiversity information portal. And from here, again, these informations find importance in different national and international instruments, such as World Ocean Assessment, then Convention of Biological Diversity, then other effective area-based management, such as Nagoya Protocol, where access and benefit sharing is dealt. And again, this Convention on Biodiversity facilitate, or they calls these information to be systematically arranged, because it facilitates the attainment or benefits for their goals, to attain the goals such as Sustainable Development Goals, SDG 14, which directly deals with the theme life below the ocean. So it is very important that we inventory and document the marine species, the occurrence records, and also the information about the species. So now in this slide, you can see the research that is being carried, the Marine Living Resource Program, or the monitoring program that is being conducted in CMLRE. Right from 1919 onwards, we have started this coloring says the amount of the number of visits we have done in each station. We have prefixed stations all along the Indian EEZ. So this coloring says the number of visits we have done to survey and monitor our EEZ. And this is our floating platform, or the research platform. We have 4RV Sagar Sambada, which is a 70-meter Danish-built vessel, which is capable of which is having an endurance of almost 25 to 30 days. 30 days to be in the ocean with all the scientific facilities. So this is considered as our floating lab of CMLR. Now when you see this, you know, to understand or to study or to model our ecosystem, we have to have a significant, or it is necessary to capture, you know, complex interactions between the atmosphere and the marine ecosystems, and also various physical process, biogeochemical process, biological process. So, as you know, the living organisms, they respond very dynamically to the change in the marine ecosystem. Here we have also a program which we monitor and survey the harmful algal blooms, and we could identify the hotspots of these HABs along the Indian EEZ, and plotting the sentinel values or sentinel changes in this harmful algal bloom. We could identify the hotspots along the Indian EEZ, and we could understand there is a change in the pattern and occurrence of these algal blooms. And here also in this slide, you can see how the frequency in the frequency of increase in the coastal HABs, which has led to many severe negative impacts like mortality along the Indian coast, which, you know, at the end having a negative impact for the livelihood of the people also. So here also you can see the severe mortality in core anoxic and broader regions. Such events generally occur when spatial and vertical oxygenated habitat is steeply reduced. So we had a serious problem of mass mortality. The reason is that as you go towards the north, there is a strong reduction in the OMZ. The OMZ is very strong as we go to the north. So this has caused a very significant problem in our marine living resources. And you know, as you know, oxygen is the main limiting factor for any organisms in the marine environment. And in a well-oxygenated region, you see there is, you know, the vertical migration of the copepods taking place, and fish abundance are there in its respective niche. But as you come to the moderately deoxygenated layer, you can see the duration or the habitat for the vertical migration of the copepod is reduced. And thus again, when there is an intense deoxygenation, these fishes which are available in the column water are pushed out of the water, and other unconventional fishery or other marine resources occupy that area. So this has got a serious, biogeochemical adverse geochemical problem, and affecting enhanced production of greenhouse gas. Here also again you see how the dissolved oxygen is affecting the benthic organism. Now you see the relation between the benthic fauna and the dissolved oxygen. As you see from from south to north, abundance and biomass of macrofauna towards the north, where there is an influence of Arabian Sea, OMZ, oxygen minimum zone is strong. The biomass and abundance of this macrofauna is getting slowly reduced. Again, we have observed, or we have monitored some of the epifaunal communities, how the oxygen level is influenced on their communities. For example, in the group of echinodermata, as you see, the diversity and the abundance is affected. More diversity and more abundance are seen in the southernmost, and as you go towards the north, their abundance and the diversity is found to be very less. Same with the case of they also have a influence. The sediments also have an influence on the distribution pattern of the epifaunal communities, where in the southern region the sediment seems to be little sandy, so the abundance is more. But as you progress towards the north, there is a change in sediment, and it is reflected in their abundance, in their diversity and the abundance pattern. We have also observed the decadal changes in marine benthos. As you know, marine benthos constitute an important component in the marine ecosystem. They play a very important role in the mineralization and availability of nutrients by recycling the nutrients. So during '98, we have made a comprehensive survey has done by dedicated cruises on board our vessel Sagar Sambada, and we have gathered both qualitatively and quantitative information about marine benthos, especially the polychaetes and crustaceans. And after 10 years, we visited the same locality, and we found that the density of polychaetes increased and crustaceans comes to be decreased in most of the sites. And this variation is, or this trend seems to be not a good trend for a healthy ecosystem. And as you know, because of this, in many states in India, we have close season to rejuvenate, the concentration or the composition of the benthic organism, which will have an important role to sustain the fisheries. So we give time for them to rejuvenate, and then directly, indirectly, they can contribute for the next, food web mechanisms. We have also observed there is a change in the geographical extent in the regime shift, or there is an extension in the geographical range of the pelagic fisheries. Mainly, in India, the two major pelagic fishery resources are oil sardine and Indian mackerels. And almost a decade back, this oil sardine was found to occupy only till 14 degree latitude. But now, during our survey, we have seen that there is a range extension of these resources. Also, it was not available in the East Coast. Now we have seen it is available both in East Coast and West Coast, and the range has extended almost up to 21 degree north latitude. Same in the case of Indian mackerel. It is as you know, it is a pelagic fish, but it is now seen even in the demersal trawling operation. The fishermen are saying they are getting a lot of catches of mackerels. So now inshore fishery alone cannot sustain the growing demand of fish availability in India. So we need to go into the deeper layers to know what is actually available, which can be harnessed in a very sustainable manner. So the first step was, we have to map the resources. So as part of this exercise, we have delineated the distribution pattern of almost 600 species of deep sea resources, mainly the deep sea fishes, and potential fishing grounds for deep sea fishes were identified. And you can see in the graph, from 50 to 300 meter range, the CPU was more in that depth range. And again, the CPU was very less. And then there was a slight increase in the CPU in the deeper zone, like 500 to 600. So these patterns, these distributional patterns, where the resources are available, these informations were gathered by our research vessel, which is the only deep sea vessel available for the research for the deep sea areas. We conducted these cruises, exploratory dedicated cruises all along the Indian East to get the information about the deep sea fishery resources. Now, apart from the deep sea resources, there are also non-conventional or unconventional resources available, which has the potential to which has a potential for other value-added products. So in this exercise, it is known that this myctophytes, which is available in the Central Indian Ocean, almost 100. The US Global has estimated almost around 100 million tons of these resources are available, and they are the lifespan of these resources is only 1 year. So these resources are just they are wasted without proper utilization, proper harnessing. So the first step towards this exercise was that we need to map these resources and see the composition, see the species composition. So we made a pilot study where these resources are available in the Central Indian Ocean, and some of the species we have found. So you can see all those resources which we have mapped and studied the biology, including the biology of these species. So,
Victoria, you have 2 minutes to finish.
Yes, I will try to, finish it fast. So this is the trawlable ground which we have identified, and during our exercise, exploratory cruises, we found that roughly about 4.6% of the EEZ is found to suitable for the demersal trawl operations in the depth range between 50 to more than 50 meters. Now, another one important aspect which I would like to share is our coverage of marine biodiversity inventarization of Northern Indian Ocean. As you know, CMLRE is a regional node of OBIS, Ocean Biogeography Information System, and we collect spatially and taxonomically resolved marine species records and their occurrence records, their distributional records. We collect it in a very systematic manner, and this is being put in this portal, and physically in our reference center, it is newly established in our center. We have almost more than 1 lakh of deep sea samples which is available for referral for the future referral use, and this is going to be a hub not only in India, to the world itself. And I am very happy to share this information that almost more than around 3,000 numbers of voucher specimens we have stored, and holotypes, that is a new species which we have described, around more than a dozen of new species which are in the different groups, right from the lower invertebrate to the Pisces. Around more than 2 dozens of holotypes are maintained in this referral center. And this we have made an exercise referring to the different literatures to see the checklist, to know the checklist of marine species, and almost 15,000 marine species are present in the Northern Indian Ocean. So so we have a lot of commitments to the international bodies. Now, as per the commitment, as per the Convention of Biodiversity, they insist at least MPA. So to conserve, we should know what is available in our. So as this exercise is, in this connection, during our survey and during our exploratory cruises, we have described almost during this period of time, almost more than 150 or 187 new species has been described to the science, and you can see in the right side the contribution of CMLR in describing the new species is almost 20% compared to other institutes in India. And this Anomuran crab, this is a very small Anomuran crab from the Lakshadweep waters at a depth of 1,000 meters. And it is been named after our vessel Sambade. And again, Axidian shrimp, which is considered to be, an intermediate species, intermediate group between the shrimps and the deep sea lobsters. This is Gynocaris keralam, which is named after our own state, Kerala. This is from the southernmost part of our state. So these are some of the new species which we have described to science, and some of the new records which these species are reported elsewhere, but for the Indian Ocean, this is new records. And you see in the right side, Diroid Sievdeki, then this is considered to be very rare species. Only 3 species of this in the family is being reported in the world over. And many, based on this, many taxonomic keys and catalogs have been brought out. And we have also an active program on mapping the marine mammals. As you know, marine mammals are important indicator, which indicate the health of the marine environment. So we have an active program on marine mammals. And another one, our goal is to see the biodiversity hotspots along the Indian East. As you know, the hotspots, the regions which are biologically rich, at the same time, it shows endemism and rareness. So here we have narrowed down to 3 areas. Like Terrace of Trivandrum, we have seen many organisms which are very unique and rare. And some of these organisms are found to be, to exhibit gigantism. For example, this isopod which we have got from Terrace of Trivandrum, which is an area of almost 25,000 square kilometers in the southernmost tip of Kerala. It is very gigantic in size. And then deep sea coral of the Mangalore coast. Then coming to the eastern part in the Visakhapatnam, we have seen polychaete beds. And coming to Andaman,
I'm sorry, but your time is over.
Yes, I'm finishing. So this is the hotspots which we have identified in the Andaman and Nicobar area, and some of the exploratory activity which we have conducted in Angria Bank. So we have made an inventory of the species which is available in this. So with this, I come to an end of this talk, and thank you very much for this opportunity. And yes, thank you very much.
Thank you very much for this. Is very impressive talk, highlighting the high biodiversity of India, and also I was very impressed by the deep studies of deep sea biodiversity. They are recently conducting many deep sea research in the Northern Pacific. So maybe in future there would be opportunity to collaborate between different Russian and Indian institutions, but I'm sorry they don't have time for questions, and we have to move to the next speaker, Dr. Nair. So please show profile.
Is it, my presentation is seen?
Yes, they can see your presentation and they can hear you. Please go ahead. You have 20 minutes.
Thank you very much, Chair. So I'm very happy, and I'm very thankful to the organizers for giving us a chance to give this presentation, which is very important for the blue economic growth of any nation. So pardon with me, actually, this is not a one location or one specific study I'm doing. I am making a presentation for how to develop an ocean information system for the blue economy. So it encompasses all the branches of oceanography, marine science, as well as the technology, remote sensing, and the ICT, information and communication technology. So this is our institute. It is called Indian National Center for Ocean Information Service, which is meant to transform the scientific knowledge to the benefit of the society, industry, government agencies, and scientific community. And in that way, it is very important for India and especially the Indian Ocean region to give a kind of information services to cater the blue economy growth of each country. And we have our broad spectrum of users, starting from the poor fishermen to the multi-million, the oil and shipping industries. So you know very well that I think we are actually going for the blue economy policies, and the blue economic growth is actually, we are expecting a great improved livelihood and jobs and the improvement in the ocean ecosystem health. So I think there is so many components in the blue economy is concerned. I am giving a small introduction. So it includes the renewable energy, tourism, and also the climate change to have the sustainability and the maritime transport and the waste management. So this blue economy, actually, it is 7th biggest economy. It is equivalent to the Indian economy, but there's a lot of issues regarding this. As you know, the increase in the intensity of the cyclone, the oxygen minimum zone, ocean acidification. These are the things actually the adversely affecting this growth of the blue economy. So the blue economy, because it's I have seen it's mainly the scientific community. I think the blue economy is actually defined by many nations. For India, it's a blue economy can be defined as a subset of the national economy, but it has got a clear the connection with the maritime and onshore and coastal zones. And it is mainly the growth of the ocean economy and mainly the offshore industries. It says if you go down to the Australia also similar way, but if you see that there is a commonality, so it's actually marine industries and it's a sustainable marine ecosystem, and as such it is a growth. So the Indian Ocean Rim Association actually identified 6 pillars for the blue economy. So the fisheries, start from the fisheries, aquaculture to the coastal tourism. So it is most important that all these pillars, we require a ocean information services that actually from the short term, the hindcast to the climatic projection. So I'll be just, I'm not giving a detail. So this, if you see left side, you have the all the pillars of the blue economy, and then center you have the ocean information service or what has to be derived, and then you have the stakeholders. So these are the actually this information generation include a lot of stages, from the observation to the modeling to the validation to dissemination to the end users. So basically, ocean information for the blue economy comprises of the science-based products and services, and the marine integrated management, ecological management of ecological environment. So we have different kind of information is required for the informed decision making for the blue economy stakeholders. It starts from the hindcast analysis and the climate projection to short-term forecast of 2 to 3 days. And the information should be user-specific, impact-based. And I'll be focusing on 3 that kind of information we have developed, and we are disseminating to the entire Indian Ocean countries, mainly on disaster prevention, fishery and ecosystem, and customized product for the marine industries or blue economy stakeholders. So I was telling actually how this ocean information or system for the blue economy is derived. It starts from the monitoring the oceans. You can see in the upper panel, you have a number of the observation systems, including the satellites. So it will be going to the, this real-time data will be putting into the numerical models or computer models. Then we make a ocean information and the advisory services and it's a multi-mode dissemination. It will go to the end users. So we have the hazard-based products actually to make the industries actually to reduce their loss of their life and properties, and customized products for actually to improve their yield. And for the biology or ecology, we have the fishery and the ecosystem services. So this is actually nutshell of, I'm not going to much science on this. I'm telling an overall view where how we will make an ocean information system. So I'll go one by one. So the generation of the services, which include mainly the early warning services. Early warning services are must for the marine industries, because the investment is much larger and the ocean is so chaotic. It is actually to require a kind of a early warning to save the life and property. So this early warning can be from the short-term scale to the meso scale to the seasonal scale. So this will be doing using the approved standard operating procedures. So these are the services, especially tsunami warning services, storm services, and sulfur services, and the perigeal tide flooding. These are some of the services actually we have developed, and it is actually catering to the blue economy users in the Indian Ocean region. So if you need, in COIS is a tsunami warning center for the entire Indian Ocean region. So I think this is actually the product which is going to all the blue economy stakeholders, especially the threat map, travel time map, directivity map, and the earthquake map for the to the end users, especially the industries, marine industries. So another service what we are providing to the marine industries, storm surge, because this is very important, because we have almost 13% of the global cyclones are coming in the Bay of Bengal and the Arabian Sea, and it is very important actually to give this forecast in much ahead, so that the marine industries and the coastal population will be safe. So this is actually we are doing using the computer models, using the where we will be giving the height, surge height, and the extent up to which area it will be going. So this is very important for the all the people who and industries which use the ocean. Same way it's ocean the wave forecast, because most of the marine industries or fishermen depends upon this the forecast to make a decision whether to go to sea or where to keep at the shore, because this kind of information is very important on daily basis to the all the stakeholders of the maritime industry. So another information what we are giving is a soil surge. That is kind of a small tsunami-like event because of the high period soils, which is coming and flooding most of the area. So this is one of the thing which is largely affecting the coastal the tourism, where this area during the fair weather season, when the tourism is high, this kind of events used to there. So so this is what I told for early warning services, but there is a service which is required for the marine industries for the day-to-day activity. This is for the ocean forecast, mainly the parameters of waves, currents, winds, and other areas. So each the industry have their own use of this information to enhance their productivity or safeguard or manage or plan their operations. So then coming to the fishery services, so there is two things we have to keep in mind. One is actually increase the productivity or increase the catch of the fisherman fish catch, and other is actually to giving a alert on the harmful algal blooms or jellyfish. This adversely affect these the coastal this kind of fishing activities. So we do this PFZ, tuna, and Hilsa forecast, that is species-wise the fishery forecast using the data from the satellites, especially SST, chlorophyll, KD 490, etc. This algal bloom services also we provide using the remote sensing satellite, especially in the visible spectra satellite. Like OceanSat or MODIS like that. So this is actually the information which is going on daily basis. So this is one example what I am telling. This is the forecast we are giving for fishermen, where we are telling this is the area the fish will be available, and you can go and fish the fish. So it is actually getting more catch and reducing the search time. Reducing the search time indirectly mean that it is actually you are saving the fuel, and you are actually you have the less emissions. So another service, what our data we are giving is actually the harmful algal blooms. This is very important for the not only the people, but also the fish, because as such, if there is a kind of a algal bloom thing, there is a depletion of oxygen, and we have a lot of fish mortalities seen in our coast. So this is another information system we develop for the blue economy stakeholders. So see the blue economy stakeholders and the marine industries also, they require the long-term maps or long-term vulnerability map, which are the area actually it is safe from all this the hazards what I told, like tsunami, cyclone, storm surge, sea level rise, coastal erosion, etc. So this is one of the work we have developed, we have undertaken and made a vulnerability atlas. So this is very important for the fish, the coastal industries to make a long-term planning for their operations and building the infrastructure. So another the similar kind of work is actually giving a kind of water quality forecasting and monitoring system, where this is very important for the many of the beach tourism, where you may be knowing very well that the blue flag concept is going, where we have to be a clear idea about the pollution level, clear idea about this all other bathing quality of the water. So this is one information system we are developing for the Indian Ocean to give a water quality forecast of all the the parameters like oxygen, nutrients, the pollution aspects, coliform bacteria, etc. Lot of research is going on this, and I'm not focusing, but I'm ready to discuss with many of the things with who are interested in this. So another developed system is actually with the increase of the blue economy, you know that it is polluted by the oil spill. So we require a oil spill disaster management, and for that we require a prediction for oil spill trajectory, where it is going to the oil is going to drift, and how much oil is actually going to be remain there, and what kind of a measures you have to see that. So we develop the oil spill trajectory prediction system. This is the online available with all the users, including all the users in the Indian Ocean. So similar way for the coastal, the wave induced the coastal erosion, also very important for the coastal industries as well as the coastal population. So we also develop the beach erosion forecast, especially during the onset of southwestern monsoon. You know that most of the areas are getting flooded, eroded, and there is a lot of problem for the people who are living in the coastal area. So this is a kind of a seasonal forecast we are developed to say which are the areas affected by the monsoon waves. So the another customized product is there once any kind of a disasters happen, I think, or the people off board happen in the sea, they will be drifted because of the existing currents, the waves and the wind. So it is very immediately we should actually go and search. This is actually the one product we develop fishermen or any other mishap happens in the sea, where people will be drifted, where this software will tell us actually where to go and search. This is effectively used by many of our the Coast Guard and the port and harbors and the Indian Navy. So coming to other customized product, we have many products for the shipping industry, mainly the optimal ship routing. We have come with the forecast for the wind energy and the wave energy, and for the maritime boats for their operations and port and harbors. So this is one of the thing actually extended the short-term prediction and the extended prediction we are doing for our oil industry, and India is coming with Invoice is coming with a big understanding or MOU with the all marine industries in the Indian Ocean region to give a kind of a impact-based and the customized services, ocean services for their daily operations. So this is something we have developed for the marine energy sector, where we identify the area where to go for the offshore wind farms. And actually we will also give the forecast for the offshore wind farms. Similar way it is coming for the wave energy also. So this is another product for the dynamic inland vessel limit, where what we are doing for our maritime ports. So where according to our forecast, they will manage or they regulate the movement of the ship. So it is actually, instead of blanket ban on the fall season, they will we will find out the windows that during those windows, that the weather windows for the week, they can do the business and they can make the money. So this is the forecast along the shipping routes. We have for standard routes, and we're also coming with the optimal ship route software. So this information we are giving to daily basis now. Presently for Indian Ocean countries, Maldives, Sri Lanka, Seychelles, Comoros, Mozambique, and there is an ample chance for India and Russia to collaborate, because I have seen actually that I have touch with many of the Russian scientists who are very good in this numerical modeling and making the numerical model setup for the information services. So of course, this has got a lot of benefit for the maritime nations like India. For example, actually the ocean forecast services, what I have told you, it is actually earning around 3.7 trillion Indian rupees actually for India, and the carbon credit as such we are getting for the PFs, it is around 36,200 crores. 1 crore means I think 10 million. So these are the benefit. This it is really benefit, because we have made a survey with this economic institute that is called that National Center for Applied Economic Research. So this is actually the how much this is actually giving this information, giving in terms of money to the Indian GDP. So you can see that this is also, it is not our work, it is our based, the cost of our information is actually estimated by another, the Economic Institute. So you can see that benefit by no go ahead mission, if you are giving to our Navy, it is actually 4,161 crore rupees. Actually, the overall economic benefit is 3.7 trillion, as I told. The oil spill, whatever I was telling, oil spill advisory, it is around, I think, 89 crores only in 2015. So the what is the way forward? So the information now presently we are giving a kind of analysis or kind of a nowcasting, but on a short-term forecast. But the marine industry, as our discussions with this many of the marine industries, we should go with the medium, this extended range and long range, as well as the climatic forecast to improve our our resources, living and non-living resources. So for example, the optimum ship routing, they require a kind of a long-term forecast. The oil industry require a AD forecast, subsurface currents. Tourism require the rip currents. So many things. We are also next Monday. We are again having a workshop with our the marine industries from the entire Indian area, and they are coming with their revised requirements. So actually, for the marine industries, we have to go. We have to develop a kind of a ready, set to go information system based upon the seasonal forecast, sub-seasonal forecast, and short-term forecast. So this is the most important for the marine industries. I think we'll be coming with this system within 1 or 2 years. Similar way for the fisheries and the ecosystem, we should develop a kind of a seasonal system to predicting the habitat suitability of this. Yes.
Dr. Nair, I'm sorry, but please try to conclude.
Yes, yes. And also the maritime site suitability will also will come with a kind of a system, the long-term prediction system. So thank you very much.
Thank you so much for your presentation, and the interesting system of ocean information services seems very well developed in India. I'm sorry they don't have time for questions, and I would like to...
Dr. Lutakmo,
yes.
We are 5 minutes ahead of schedule, and this is a very, very important presentation. He is one of the only speakers talking on the ocean information services. So I have a question to you that which organization in Russia would be appropriate for them to collaborate for numerical modeling and other projects which he talked about?
Yeah, I'm not very sure about that. When we have some meeting on the WMO meeting,
I
asked moderator to advise us.
I'm sorry.
No, and then possibly you can also speak up.
This question for me, you know, I'm not very aware of such information systems in Russia, and I think maybe tomorrow there's some scientists from the other institute, Pacific Institute of Oceanology, they would reply in detail, but I'm not sure if they have this developed system.
Right. Professor Bala, you wanted to say something, yes?
Yes, Dr. Shishir, actually, we have a lot of discussions. Earlier also with the Russian Academy of Science, we have some discussions, but nothing actually moved forward, because I have seen that many of the leading scientists actually working in this area, especially this require a kind of a development of a computer model. So I think you know that many of the Russian institutes, they are very good in the computer, the mathematical model. So I think there is a lot of institutes, but they are working not towards really this kind of system, but actually they are very good in this area. That I know, because I have a lot of interaction between scientists in WMO and the UNESCO meetings. Thank you.
So you are right. Actually, Moscow State University is hosting another parallel conference on these dates, and we will try to find some of these scientists. People from our scientists from CDAC also. They yes. Contacted these people for high performance computing, and they are running some projects. And similarly, they are developing some projects on ocean information services also. So you are right, absolutely. There are a lot of publications, there's a lot of work. So we'll try to connect you.
Okay, thank you. Thank you very
much. Okay, thank you. I think that they have here in the National Center of Marine Biology some monitoring system for harmful algal blooms, but it's very limited. It's not the kind of integrated system of monitoring of different biological parameters. So I have seen Dr. Petruk appeared on the screen, so maybe they may ask him. Dr. Petruk, can you hear us? Can you join to the meeting now with your presentation? Please turn on the microphone. Your microphone is off. Включите микрофон, пожалуйста. Микрофон выключен. Dr. Petro, can you turn on the microphone? Your microphone is off. Включите микрофон, пожалуйста.
Вроде все.
Yes, now they can hear you. So can you present now?
Yes, of course.
So please, the IT team, please show the profile of Dr. Petro. This is the first presentation of this session.
Yes, the moment. Yes.
Yes, just so Dr. Dmitry Petro, please go on. You have 20 minutes.
Yes. Yes, a moment, my presentation is.
Включите демонстрацию экрана внизу.
Дальше не загрузилась.
Сначала презентацию, потом демонстрация экрана.
Я знаю.
У вас презентация не запускается или как?
Да.
Или что?
Да. Тут у меня с презентацией. На этом компьютере не было PowerPoint, его поставили, но он глючит, потому что...
Ну, давайте тогда, чтобы времени...
Через одну, сейчас другого кого-нибудь.
I'm sorry, ladies and gentlemen. Again, there are some technical problems. So please show the profile. The next speaker, Professor Yuri Kartatsov. Dr. Kartatsov, you have 20 minutes. Please start your presentation. Now they can see your presentation.
Dear colleagues, I'd like to present this talk here. As you see, it will be devoted to molecular problems and some general problems and some
Юрий
applied
Федорович, разверните презентацию во весь экран.
On this slide, you see field of activities, eyeball and Rose-Ball initiatives, worldwide initiatives, and the cloud of different quite impressive activities are shown here. There are a wide variety of problems that exist currently in general biology and general genetics. For example, why DNA barcoding is successful. Is it so indeed? What is theoretic basement for this, if any? Currently, strict contradictions are announced between experimental genetic data and general biology paradigm. Are they real? Such kind things as monophily versus polyphily, bifurcative versus reticulative net-like evolution prevalent. What is the typical pattern? In general, how frequent is one speciation mode among others? Are the differences present? The above main goals may be shown like this, three of them. Hybrid occurrence and genetic integration, kind of signals from gene tree obtained, to data on molecular diversity in lineages fit modern basic concepts, neo-Darwinism, or equal synthetic theory of evolution, and biological species concept, STE plus base C. 2. Tree topology reticulation testing by mtDNA and nDNA markers, what it is evidence for. And 3. Applied point, food fraud. What are main outcomes from marine food mislabeling research? I'll talk primarily about animal taxa, but most ideas are applicable to other taxa too, including unicellular organisms. To talk about the matter, I'd like to just stress for audience, maybe which far from the topic, what is markers we can use and where they are suitable for. Such markers as spacers are usually applicable to intraspecies level and sometimes for wider aims. Mitochondrial DNA markers are quite far, maybe used in a far scopes, starting from intraspecies level and even in the order level, they're very applicable. And nuclear markers and rDNA markers are applicable for more higher rank taxa. How to measure the signals from DNA? That is a simple example how to we can measure nucleotide composition along sequences of different species. Here is the example from one gene, CO1, for flounders. As you see, some nucleotides are same for all sequences, but in other cases, they may be different positions occur. That is measuring, that's why measuring such differences, we can estimate genetic distances among sequences, and so we may judge about the evolutionary signal. More details we'll discuss a little bit later. Framework for the job. I will not comment all the items here, but it is important, as in any biology sampling, then species determination in our scope, we try to use experience of experts in taxa. Then we try to collect different information about specimens and organisms, creating database, and all specimens with DNA and images of alive organisms go to the databases. And in the end, we have sequencing of nucleotides and try to analyze by different techniques the appropriate signal, specimen identification by different techniques like this. And in the end, we have built gene trees, sometimes species trees, and this signal may be useful in evolutionary genetics and evolutionary biology in general. So we may go to goal one, hybrid occurrence and genetic introgression, and how they applicable to the modern species and biological concept. Recent evidence in molecular phylogenetics, phylogeography, and comparative evolutionary genomics form the basis for mentioned challenges to the synthetic theory of evolution, STE. There are three kinds of such evidence. One, genetic introgression across species boundaries. Two, reticulation in gene and species trees. And three, genome mosaicism. I will touch in my talk about first 2 points. This slide shows data on hybrid occurrence in the mussel species, Mytilus spp complex. On the left image, you see the frequency of hybrids, and in the middle, and invasive species in our waters, and local species, Pacific mussel. As you see, 3 different types of genetic markers used, and the signal is similar for all of them. Local genotypes prevail, hybrids occur, but most of these hybrids, which kind of hybrids? Here it is not clear from this image. On the right graph, you can see the frequency of hybrids of different types for different localities in our area of Sea of Japan. In blue, general types of hybrids shown the occurrence. On the gray color, that is F1 hybrids frequency. And as you see, most hybrids in nature for this species belong to hybrids of first generation. And only few of them are back crosses or other kind of hybrid. That is very important clarification and distinction between different types of hybrid and their importance for the genetic introgression, which is raised in literature very widely, and this data interpreted as very harmful for the nature and destroying the species borders by big gene flow. What is the real situation currently? I try to analyze and made analysis, meta-analysis, more than 20,000 literature sources. And this data summarized on these 2 histograms. Above histogram shows just the frequency of hybrids in nature. As you see here, it is frequency varies from nearly 100% to very low levels. And if talking about the score of real genetic introgression, it is quite low, really, as data shows. Here I show it in a yellow color, this data. Hybrid frequency in general is about 25-26%. And genetic flow is much lower. It is about 5.7% with standard error close to nil. So we can consider further data on the interpretation of the phylogenetic molecular signal in a to have more general picture on the data. On this tree, that is gene 3 for CO1 gene sequences for flatfish species. Quite simple tree. And we see the signal occur from 2 points of view, from the topology of the tree on the highest level as the different families of Rhonectiformes, and then on more level, lower level hierarchy, like sequences of the single order, single genus, and next level specimens that belong to the same species. As you see, this level has single root. Tree has single roots on this level, and this level. So they're well supported statistically, these levels, and the signal is obvious. Topology has monophyletic mode, and the genetic distances, as we see by the scale and the length of the branches of this tree, increase. Genetic distances increase with the rank of taxa. That is a simple example, but maybe other data will show different. On this slide, I try to present for you more informative tree on the mitochondrial genome and for day species, fish species. And sometimes we see the generally topology of tree also bifurcative. Most branches are monophyletic, but sometimes we have different pattern, like this on a part of our Pseudospinini and Leuciscine subfamily. And you see that along with one genus, some other specimens occur. That is a kind of paraphily. So how frequent this type of not monophily, paraphily, or polyphily occur? There is unfortunately no general way to investigate how frequent these deviations occur. We try to apply our own approach to measure taxa level and genetic differentiation. That is, on the axis, you see genetic distances squares, and here on x-axis comparison group show. The lowest level of genetic distances has for interspecies level, then interspecies for single genus, then intergenera between the families, and the highest is for families of the same order. As you see, this data shows near linear increase of genetic distances with taxa rank. That is mitochondrial genome data, U1 for complete metagenome data, and that is data for protein genes of the mitochondrial genome, close to that data for other group of organisms, fish organisms occur. So on the question in the beginning that I raised, do data on molecular diversity in lineages fit modern basic concepts, STE with BSC? The answer is yes, they are well fit. So we can go now to the goal number 2, tree topology and reticulation testing by mtDNA and nDNA markers. What the evidence? They have evidence for. Technique, as I show, may be different, but no unification exists currently. So, for example, you may analyze topology of gene trees empirically, have some theoretical application, or use some fair tools to analyze. For example, PC simulations exist, empiric and soft data analysis, our own data, empirical and analysis below. biodiversity quantification have several steps, and we try to apply our own original approach. We try to use VLAT data system, that is barcoding basic data system that currently include mostly data on CO1 mitochondrial DNA barcode records. We compare 3 different taxa levels: intraspecies, intragenus, and intrafamily, and analyze so-called bean scores. That is bean, that is barcoding index number, and we can measure concordant classification and discordant classification according to the place existing in the VLAT database zoological record. So we can identify is molecular classification concordant well with empirical data or not well. As you see on this y-axis, a level of concordance is around about 80%. And that is similar for all 3 levels that is underlined here, as I explained. This is a quite big data of different researchers and research groups that placed their data in VLAT system, including our own data here. So most gene trees, we can conclude, at genera and family level show low reticulation pattern, if any, because topology measured by morphology and molecular data are not deviate significantly. We try to apply not just comparing average, but made regression comparison of 2 former projects on the left and other project on fish. For convenience, we use this data. And you see again, barcoding index number data well correlate and good concordant with the log bean or scale variation. That is scale of morphological variation, and that is independent molecular variation data. So they're very well connected, and the coefficient of determination for left graph is over 98%. Here it's a bit smaller, but in general, coefficient of determination is higher than 84%, correspondingly for 2 analyses. So finalizing these records and using our former record for 2 genes, CO1 and cytochrome B, for very big data set for more than 23,000 animal species, we see that there is nearly linear pattern of connection of genetic distances with comparison group. Here we have 5 comparison groups, and we can conclude accordingly. First, species identification at single gene CO1, like CO1 CB base, is highly effective and unambiguous, 82–98% precision, because low intraspecies here and high interspecies here, diversity at these markers, and as we see before, on complete mitochondrial genome. Conclusion 2, positive and proportional association between distance score and taxa rank supported the idea that speciation in most cases goes by geographic mode, D1 type, and that at least in animals, the phyletic evolution prevails. This data empirically prove basics of PSC and neo-Darwinian interpretation of speciation and evolution. If other speciation mode will be frequent then and comparable in the frequencies than D1 type, the relationship should be inflated, and low correlation between native reasons and tax around, we will see. And three, there are alternative modes of speciation, of course. These 2.1, 2T4, etc. They are rare in nature. This is certainly does not mean that they are absent or less important. This may mean known things. Darwinian evolution may prevail. However, rare drastic genetic transformation would produce principal novelty. And few words on the goal number 3. What are main outcomes from marine food mislabeling research? The number-crediting methods have been efficiently applied for validating different fish products. In Canada, USA, Tasmania, Egypt, South Africa, and other countries, including our own research in our supermarket fish products. On this round three, you see a good example about mislabeling in our fish markets. Atlantic halibut here and Pacific halibut here has obvious differences and different clusters occur on molecular data for CO1 marker. But Pacific halibut is very tasty and quite expensive product. But in the supermarket, they sell on the same single label. So we have customers that lost a lot of money, sometimes buying different food, different not so good food as they expected. Similar
Dr. Kartavtsov, please try to finish your presentation. Your time is over.
Single label that about 7 different species are actually present at the market. General picture shown for the world market shows that different for different groups, but about 25 and sometimes even more % of foods, fish, and other products, marine food, are labeled. Thank you for your attention.
Thank you very much, Professor Kartavtsov, for your interesting presentation. Unfortunately, they don't have time for questions. And I would like to try again to invite to present Dr. Dmitry Petruk, and I hope this attempt would be successful now. So, Dr. Petruk, can you present now?
I hope.
Okay, so they can hear you. So try to start your presentation.
Thank you very much.
I have no sound.
Can you hear me?
I can hear you. Yes, I can.
Great. Okay. Dear colleagues, I would like to have.
Please start your presentation. They don't see your presentation.
Really?
They can see only you, but they can't see your presentation.
Just a moment. I try to start.
Внизу демонстрация экрана.
Я нажал на нее.
А сама презентация запущена?
Запущена. Сейчас она загружается. Как-то она корява. Здесь...
Во весь экран ее, вот демонстрация, наверное, где у вас?
Can you please start the presentation? Choose the full screen mode. In the upper part of the screen, there is a Start Presentation button.
Выше демонстратор.
In the upper corner, I've pressed it, but to no avail. I can see that it says that you have started the presentation, but it doesn't show.
Да, выше вот стрелку. Вот мы же видим ваш экран.
Да, тут уже выше нет, еще здесь новая демонстрация. Не видно ее?
So you still cannot see it. You should go to the upper corner of your screen and unfold the presentation. Maybe I have a different version of the program. Yes, and now move the arrow to the upper part of the screen.
А вы можете стрелочку выше поднять?
Да.
Can you move the arrow to the upper part where there are tabs File, Insert, and so on? Look, I cannot put it to the upper part. Please press F5 to refresh the browser. No, nothing happens.
Вам не видно. Показать вам.
Дмитрий Леонидович, посмотри командную строку. Она у тебя вторая сверху.
Which is the second tab. I have a different version of the program. Yeah, we can see your screen now. Yes, just... Yes. No, you mean the new demonstration. Is that what I'm supposed to press? No, choose file, editing, go, choose it.
Yeah.
Okay, so let's go for that. Will you be able to change the slides on your own? Maybe you will be able to close the presentation and then open it again. It doesn't show in the right way for some reason.
Включите демонстрацию экрана, разверните.
Can you please turn off the screen demonstration and then turn it on again?
Some problems. Please hold on.
Вот сейчас наверху.
Now go above. Please choose the show slides option and start it, where you can see the 85% figure. Show the icon then. Okay, so now we can see it.
We can see, so ladies and gentlemen, sorry, so you have 20 minutes to present.
Okay.
Please go on.
Colleagues, I'm sorry for these technical problems, but nevertheless, I am very glad to have this unique possibility to introduce you in our facility, Primorский Aquarium, and part of our activity. Primorский Aquarium is not only situated at the coast of Ruski Island, but it is situated on quite small peninsula, Zhytkova Peninsula. So the aquarium is surrounded by waters of Peter the Great Bay, Sea of Japan, or Eastern Sea. In Russian, aquarium is oceanarium, so ocean inside, and as you see, ocean outside. Building of our aquarium has a very unique form, like a shell at the sea coast. There are 9 exhibits in our aquarium. All these exhibits devoted to different zones of world ocean, from Arctic to Antarctic. More than 500 species and more than 15,000 species of animals. They are in our aquarium. Total volume of water is about 25,000 cubic meters. Total number of tanks in the exposition of aquarium is 118. near to main building, their science and adaptation facility. The main task of this facility, quarantine of hydrobiomes before placing into exhibition tank, cultivation of some hydrobiomes, and conducting research. The main goal of Primursky Aquarium is to distribute knowledge on the North Pacific biota, because North Pacific biota is most interesting for our investigators and for public also very interesting. Of 113 tanks, more than third are placed in the Far East Seas exhibit. Fish collection of Far East Seas exhibit consists of more than 1,200 specimens belonging to 62 species. Like all aquariums in the world, the Primorsky Aquarium replenished its life collections mainly by buying and catching animals in right. The use of different way of getting animals help us to get Far Eastern marine fishes on early really ontogenic stages, and this possibility make possible investigation ontogenesis of these fishes, and we can get fishes on early stage of ontogenesis to our aquariums. This is help us to increase fish species diversity in its position of the Primursky Aquarium, because adult fishes quite difficult to get in result of different reasons. Ways of getting Far Eastern marine fishes at early stages of ontogenesis. This is hatching from eggs collected in nature. Now we have 24 species from 9 countries getting by this way, spawning in the aquarium 7 species, and the cultivation of fish larvae. 50 species from 16 ways. This fish is this Paris agassiz. Eggs of this fish you get eggs and larva. You got 2 years ago, and we were very surprised when to autumn this fish got quite big size, almost 400 millimeters, and this is both family and females, and the males were mature. This is unusual situation for only one season, from May to September, from larva to absolutely adult specimens. We explain this situation by quite high temperature and abundant food. For this fish, food and temperature, this is most important matter. And all fishes that we get in our area show good result in growing and development. And we hope that biologists will have unique possibility to investigate fish, and not fish only, but other marine animals. For example, jellyfish. Jellyfish is quite difficult to investigate in nature, but in tank in our facility, this is very easy. Now there are approximately 8 species in our facility of jellyfish, and we, our specialists, can reproduce these animals. Now, 4 animals you see name of this jellyfish, and we hope in future increase this number. And octopus and Virostrattas spawned in our aquarium, and we hope to have good result in growing in future. Now with octopus, unfortunately, we could not get good result because there are too many problems and necessary to get experience. Not only animals, but algae. So it's quite important to grow in our facility because there are problem to have possibility to get algae in all the season. But in aquarium, we try to get this possibility and make a good condition for sea algae. Tropical sea exhibit, this is largest in our aquarium in volume of water and in number of species. This is more colorful exhibit, and we also get animals from tropical area, but now we have good result in program of reproducing these fishes. It's very important because in the world there is a big problem in the merger of coral reef in result of catching of fishes with using different toxic methods and using different kinds of nets. So this is one method destroyed coral reefs, and another one when coral reef fish number of coral reef fish decrease. There is situation at coral reef change because coral reef fishes eat algae. Without fish, or when fish become less, algae growing more effectively, and coral reef has some depression. So, ornamental fish, this is very interesting subject for reproduction in artificial conditions. And we have all base for this job. We have fish stock, we have brood stock, and we have animal food supply department who supply us with necessary feed for fishes from phytoplankton to brine streams and others. And of course, due to situated of our aquarium at the sea coast, we can use good sea water heated and clean. As a model for testing known aquaculture techniques, we use tomato clownfish, amphiprion natus, and now we have good result of this job. And using this information, we try to reproduce another species. Live feeds, this is one from most important matter for this job. And now our special department can reproduce 10 species of this animals and microalgae. So from microalgae to done worm, this is very wide spectrum of food that we can use for our fish. A live feed is very important matter to growing animals, because some animals, some fish eat only alive feed, and during growing of this feed, this matter is very important. Okay, this is some tropical fishes that we reproduced in our facility to this time, and this is not finished for our job, but I see beginning only, because we have would start, we have understanding of this situation. And
Dr. Petru, please try to finish your presentation in 1 minute.
Okay, okay. So this is one from unique fishes that we got last year. These animals also was got in our aquarium, like this larva seal. All these animals were born in our aquarium, and this beluga whale also. This is unique in the world, and this picture you can see, you saw Japanese sea lion, but only small light moving, but animal image disappear, and in this way species also can extinct due a very small effect by human. So it's necessary join our facility to save our ocean, to save our macrocosm. So thank you. Thank you for your attention.
Thank you very much, Dr. Petruk. Well, I'm sorry they don't have time for questions, but I just would like to comment that this is the biggest aquarium in Russia, and it's not just aquarium for public. It's a branch of the National Scientific Center of Marine Biology, and this aquarium have research programs and educational programs, and this is one of the largest in the world. Thank you so much. They still have 4 speakers for today's session. Let's move to the next speaker, Dr. Baban. Please show profile. Thank you.
Hello. Hello.
Hello.
Can you see my presentation?
They can hear you, but still can't see your presentation. Please start demonstration of the screen.
Yeah, I have just opened it.
Yeah, sure.
Let me just check it. Is it visible or not?
Still I can't see. Не понял, что он делает?
Hello. Is it visible? My screen is visible or not?
They don't see your presentation. Perhaps you have to start your presentation first and then start your presentation. Yes, they can see yourself, but start presentation and then start demonstration of the screen.
Okay. Is it visible or not? I have started already presentation.
You have to share the screen. Have you shared the screen, Dr. Baban?
Yeah, I have put it on the sharing.
Okay, so there are two types of share. One is share screen or the tab.
I have put it on the, let me see it once again. Share entire screen, I have put it, and I could.
No, when you go to share, yes.
Is it visible now?
No, they see another screen. So you have to show your screen, the presentation. Okay, now just back to the last screen. Yes, now they can see your screen. Okay.
Is it okay?
Yeah, just enlarge it. Enlarge.
Okay, it is.
Oh yeah, fine.
Yeah.
Okay, now they can see. So please start your presentation. You have 20 minutes.
Thank you. First, I would like to thank the organizer, both from India and Russia, giving me this opportunity to present myself and the work I conducted at National Institute of Oceanography, Goa. And presently, I am at the National Center for Ocean Research at Ministry of Earth Sciences, also in Goa. This is part of the work which we have conducted under the Deep Sea Biodiversity Program. As we all know, the Indian Ocean is is one of the least sampled from the biological point of view. Also, it is it has a different habitat as far as the ecological habitats are concerned, particularly from the deep sea, because I'm focusing mainly on the deep sea. And as you can see that we have the Bay of Bengal, Arabian Sea, here is the Chagos Ridge, you have the 90° East Ridge, and then you have Carlsberg Ridge, Indian Ocean Ridge, the Southwest Indian Ridge, and the Southeast Indian Ridge. So these are the different complex ecological habitat provides good opportunity for benthic biology for develop and flourish into this region. So this particularly if you talk about mid-ocean ridges, which is spreading somewhere like 18,000 kilometers in length, and it comprises over around 28% of the global MOR systems with various spreading rates and so far around 11 active hydrothermal vent fields have been discovered. These are some of those areas, red stars and red, where the active hydrothermal sites have been identified. Indian Ocean is also the world's second largest high-grade manganese nodule deposit area. This is after the equatorial nodule belt deposits in the North Pacific, which is also called the Karaoke zone, fracture zone area. In the last 40 years of the deep sea explorations globally, we have scientists have discovered over 90 active hydrothermal fields in different areas. There are over 700 hydrothermal sites on the MOR systems, and also they have discovered more than 500 new species directly associated or these active vents. So what is the current situation globally if you look at the deep sea biodiversity? Deep sea biodiversity, as you know that our deep sea area, if you talk about it, it is 50% of the oceanic area, which is more than 3,000 meter water depth. So it is 50% of global ocean, it is constituted the deep sea area. Also, if you look at it, only 5% of that area has been explored with different type of equipments. And if you go from the sampling point of view, less than 0.0001% of this deep sea floor samples area have been sampled physically. As far as Indian Ocean is concerned, Indian Ocean is one of the least sampled, particularly from biological point of view, region compared to all other deep sea areas. We have been exploring the deep sea area for last 4 decades around both east and west coast of India, particularly in the deeper region. When you go, these are some of the vessels which we have used in the NIO. There were many more also, but these were specifically we have used it, in the deep sea. These are the equipments which were used for deep sea sampling: box cores, multiple cores, penguin grabs, Peterson grabs, dredges, and beam trawl. And the samples were, standard techniques were used, processing on board and in the laboratory. When we talk about the deep sea biodiversity, benthic biodiversity, globally it has been accepted that there are, based on the size fraction, we have divided that. This has facilitated the researchers more easily. So therefore, microbenthos, meobenthos, macrobenthos, and megabenthos, and these are the size groups they have been defined with. If you look at the meofauna composition, particularly the deep sea, and that too I'm talking about the abyssal deep sea, both from northern Indian Ocean and the central Indian Ocean. If you look at more or less, if you have meofauna, they will be of this type of forms. You will have it dominated by some of those, sometimes polychaetes, smaller size, because we are using smaller ships, but most of the time it is the nematodes which is the most dominant in the, and these are the major groups of the meofauna. Macrobenthos, when you talk about it, as you can see it more clearly, they are the one more than 0.5 millimeter in size, and the groups dominants are, again, we have polychaetes, crustacean, and mollusks. These are the major groups dominant in the soft sediment, as we can see that. And there are more than 25 or 23 taxon groups. I'm not talking about the species. There will be more than 300 species what so far we have been identified. I belong to the 15 phyla. And there is a clear relationship with the water depth and the sedimentary habitats. I will talk a little bit more in. As you can see that the distribution within the abyssal region, Central Indian Basin, where we have done the extensive sampling, and this was directly related to some of the programs about the deep sea mining environmental impact assessment, particularly for the polymetallic nodule. This study was related to that. I'll talk a little bit about that also. And as you can see that here is the surface distribution of the abundance, macrobenthic abundance. Here is, as you can see, that the overall distribution, latitude and longitude, and here is the abundance per meter square. Here is a biomass, again, milligram per meter square overall in the Central Indian Basin. Here is the water depth, and you can see that the biomass and density, and as water depth increases, the abundance decreases sharply. But in the coastal area or the shallow area, as you can see, around 3,500, you have got the higher abundance, and then as you go down, it will be reduced drastically. An important message from this is that macrobenthic abundance is very patchy, both even meo but macro specifically, patchily distributed in the soft sediment, both density and biomass. And these peaks, as you can see, the biomass peaks and the abundance peaks are directly related to the biogenic material available in that area, or we can say that directly related to the food material available. And as you can see in the macrobenthic vertical distribution in the sediment, we have studied with the box core up to 50 centimeter, and Here is the vertical distribution of macrofauna. As you can see that they group wise, and here is the family wise distribution of some of those forms we have presented here. Here is the same box score. We have got organic carbon vertically distribution, and here you have got the labile organic matter, protein, carbohydrates, how they are distributed. And as you can see that here, there is a direct relationship between the food availability at the deep sea and the particle flux, which is from the surface. These are some of the familial identification of a dominant group, as we have polychaetes, nematodes, ctenidians, herpetoids, and you can see the latitudinal distribution of some of those species. And in the dominant groups, particularly polychaetes, you can see ctenids, herpetoids. Here you can see it clearly as it was shown in the density and abundance. Here also there is a patchily distributed. You can see there is a patch where you have got high diversity. Again, you have got the high diversity around here. I would like to mention it very clearly here. Patch distribution, one is what we have studied with respect to the organic material and sediment texture. But most important also is to be pointed out here is we do have it more samples in some of those areas compared to the other areas. So we can't directly say that it is only related. Also, this little bit of our sampling bias certainly is there, because this study was focused with the polymetallic nodule environmental impact assessment in the abyssal region, and therefore more sampling in the limited area was, or the small area was done. What is the message from here is one of the group which we did it up to species level identification, and that is nematode. So deep sea nematode assemblies from the commercially important polymetallic nodule area in the Central Indian Basin. Here we have seen that there were around 284 morphotypes, different morphotypes, not completely identified up to the species level, but there are different genera, more than 113 different nematode genera belong to 30 families were identified. Average density in CIB area, what we have got it around 27 individual per 10 centimeters square, which is comparable with Pacific Ocean. But overall, if you see that there is a lower density in the CIB compared to the CCF or Pacific Ocean. Another important component is there. We also did the rarefaction, species rarefaction plot here, and which clearly indicate that it is not showing it has reached the straight line or asymptotic, which shows that more samples would definitely yield more species, and therefore to see that what is the maximum species available in that area, we may require more samples to conclude with that. Even though our sampling, as I mentioned it, that were spread in very large geographical area in the CIB, the composition of both macro and meiofauna, especially the dominant nematodes and polychaetes, are comparable in generally within the CIB area. We also have used, as I told, that northern Indian Ocean, both Bay of Bengal and Arabian Sea sample, deep Arabian Sea particularly, both continental shelf slope and the abyssal region have sampled, and based on the sedimentary habitat, the coastal basins are divided, particularly with the sedimentary habitat. And as you can see that there are those, here is a Mumbai basin is there, Konkan Kerala region is here, then you do have KG basin, Mahanadi basin, Bengal, and Andaman Nicobar basin is there, Kaveri basin. And in terms of the species richness or the diversity of this basin area or basin scale area, if it is there, dominant groups are there: gastropods, bivalves, and polychaetes. And species wise, we have around 1,400, more than 1,400 gastropod species, belong to 142 families, more than almost 500 genera. Bivalves, around 582 species, belong to 57 families. Then polychaetes were 65 species. And if you look at the basin wise, Kaveri basin and Andaman basin are the one which are rich in the richness or species diversity. Okay, considering those basin level and there are some specific area with respect to the oxygen minimum zone, and we have sampled both east and west coast oxygen minimum zone, particularly with respect to the west coast or Arabian Sea oxygen minimum zone. We have an opportunity to sample it repetitively in some of the transects. As you can see that this one of the transect we sampled in around 2000 or 8-9, and we found that there is oxygen minimum zone in the depth range of 200 to 1,500 meter. The transect is from shallow around 47 meter to 3,200 meter area, and the highest, as you can see that the highest macrobenthic density was in the self region around 3,700 per individual per meter square, and lowest was in the basin, down basin area, around 326 individual per meter square in 3,000 or more than 3,000. What is interesting again here is about the biomass spectra. We do have it high biomass, as I told you, that in some of the areas where in the slope region. However, here you can see that high biomass in the slope region, and size spectra, if you see that 300 micron to the 2000 micron. So we have used sieves to separate size, and the maximum we have got it, as you can see that in the slope. So the composition of the fauna was from this size group, like 500 to 1000 micron, and that was most in the slope region. And as you can see that this is the OMG region where you have got the higher biomass. Recently, one of our colleagues, Dr. Sabyasachi Sautia and his group have sampled again around the same area in the Arabian Sea, and they found it very interesting. Our earlier study was more focused on the macrofauna. Here they have focused on the meiofauna and found more or less similar type of conclusions. Like what, for example, you have got the continental margin, you have got the slope, and then a basal plan, and they have differentiated type of fauna available in different area. And they found that distribution on meiophil taxa along the gradient, you do have it high abundance in the shallow region, but you can find sometimes higher abundance even or biomass for that matter in the non-OMG area. And this was the OMG area, as earlier also I have mentioned about it. So OMG area, you do have it, lower abundance and biomass, and then again you get in, if you go to the abyssal region or the slope region, you have got the higher both density and biomass, both in terms of meiofauna and macrofauna. About the functioning of both benthic ecosystem functioning, we studied it in detail about that. The same transect earlier, what was I mentioned it about the macrofauna in the Arabian Sea, and we found that one of the group, again nematode, we did it more detailed, and we found around 110 nematode species belong to 24 families along this transect what we have studied. Most importantly, again here what we have got it is dominant with the deposit feeders, as you can see that, and these were again differentiated based on the body structures, morphological structure. So most of the benthic ecologists, they know that both dominant meiofauna, nematodes, and macrofauna polychaetes, we differentiate them on the functioning. They are functioning based on their mouth parts. Or the mouth, how is the mouth structures, and in these terms, the feeding habits. That is what is there. So we have nematodes, deposit feedings were the most dominant, predatory forms were the second dominant in this region, and epigote feeders were the third one, which were available. Interestingly, there were even the predatory forms, the feeding on nematodes itself. And this is what we have found it in the some of the areas. The nematodes were actually having the young nematodes inside their gut. So they're feeding some of those, they were carnivorous species. So naturally they were feeding whatever is available in the area. If you look at it, the abundance, particularly abundance and diversity, shelf, slope, and basin region. So abundance is high in the shelf region. The food availability is very good there. However, the diversity, if you see that, is also high, not only the shelf region, but even in the basin region. So this is the contrasting, except, OMG area where you have the lower diversity, but again, even the basin area, we have higher diversity of meat. Now, looking at the functioning again, as I told you, we looked at the dominant macrobenthic groups from the OMG area. What is the adaptations? And one of the things which we found that the branchia of these, most of these polychaete species, the filter feeding, they are there, most of them there. And then they expand their branchias in the OMG area, probably for storing and rationing the oxygen during the oxygen minimum season. And that's how they adapt to those areas. And because there is a high food availability, but oxygen is low, and then therefore they exploit that region where there is a less competition, abundant food available, but low oxygen, and therefore the adaptation help them to survive in such area and flourish and use the resources for the community development. So these are the, already I talked meofauna and macrofauna. This is overall about the both northern Arabian Sea and northern Indian Ocean and the abyssal. Now we are working, recently we started working on more details on the mid-ocean reef systems. And as we have found that apart from the abyssal region, there are different habitats which are hard substrate habitats, and those hard substrate habitats are very rich in for megafauna, particularly sessile fauna.
Dr. Baban, please try to finish in 1 minute.
Yeah, sure, I will. And then the dominant species groups are the sponges. We have collected recently large number of samples, and we are working more detail on the taxonomy of this one. There are the new species, no other species reach, but it is not been sampled. And as you can find out, if you go for the detailed sampling, almost 50% possibility of 50% or more new species is there, particularly from the ridge area. I can say it. We studied it both from the habitat mapping point of view as well as the physical sampling. These are the new samples which are not yet completely identified. New fauna which we have found it. There are new species. This is what I was just talking about, the proposed nodal mining area in the CIB area. This is the Central Indian Basin where we have got rich nodal deposits. We have studied the environmental impact assessment, and most importantly is the fauna associated with these nodules is also quite, and here is, if you have more surface area, you have more abundance. So it is directly associated with the nodule. Now coming to the hard substrate or the hydrothermal vent areas. So these are some of the areas where we have been sampling. We have the 4 areas which is Indian Central Indian ridge area, and this is one where these are some of the dominant species which found from the hydrothermal vent, active hydrothermal vent. Around 11 are reported so far in the literature. So all the data collected from the literature, Southwest Indian ridge and Central Indian ridge, around 125 species are directly associated from the active hydrothermal vent have been identified. Of that, almost 85% are the new to the science. And also, not only the hydrothermal vent, we do have it coal seeps in the Bay of Bengal, particularly in the KG basin, as you can see that have been sampled and studied in detail by National Institute of Oceanography. Recently, they have published it. Interestingly, what is important here is we have hydrothermal vent, we have cold ships, and there is a direct relationship between these cold ships community and hydrothermal, because both are, chemosynthetic habitat, both are different in terms of the temperature. However, there are the species which can tolerate high range, and there are some species which are similar to the hydrothermal vent, which are found in the cold ships region. So what is important here is the genomic study of these cold ship communities and the hydrothermal vent communities need to be done, and comparison with vent communities along with the detailed water sampling also is required. Water column sampling in biology is required. There, it will help in identifying and understanding the genetical connectivity of these deep sea species which are associated with different habitat, but what do they have the genetical connectivity not only within the Indian Ocean different habitat, but the other ocean also. We do have some information about the environmental impact assessment which has been studied in detail and also published. Now finally, I would like to just suggest, recently there's a paper published in Nature which indicates that if you are looking for the climate solution, please protect the more ocean, particularly the benthic habitat, because among the surprising finding, what the paper indicates that trawling, deep sea trawling releases as much as planet warming carbon each year, which is equivalent to global aviation emitted carbon. So that is how it is important to protect the deep sea also from different anthropogenic possible future impacts. Again, I would like to thank once again to my parent organization, National Institute of Oceanography, CSR, and NCPR, Ministry of Earth Sciences, to give me the opportunity, and all of you for listening. Thank you.
Thank you, Dr. Baban, for this very impressive presentation, and was very impressed with the significant progress in deep sea studies, deep sea biodiversity in India. And as you told that there's still not enough sampling effort done, so I think that the international collaboration can improve, this situation in studying of the Indian Ocean deep sea biodiversity. I'm sorry, but we don't have time for questions, and I suggest to move to the next speaker. Please show profile, Dr. Timofey Malerenko. Dr. Malerenko, can you hear us?
Yeah, I hear you.
Okay, so please please share your screen and start your presentation.
Do you see my presentation?
No, they can't. They can hear you, but they don't see presentation. Please start the sharing the screen, демонстрация экрана.
Вы видео включите, видео.
Please turn on the video mode.
Now they can see yourself.
Дело в том, что я не с ноутбука, а с телефона.
Well, unfortunately, I will not be able to do that from my phone. It says that you have low level of connectivity on your phone. Probably you are using Wi-Fi. Yes, I do. Okay, so then we will not be able to show your screen due to poor quality of connection.
But you can see presentation. Снова написано низкая пропускная способность.
Again, it says that you have poor quality of your connection. So probably our IT experts could look into that. Maybe they could assist Mr. Malorenko.
Я на нее нажимаю. Сейчас, секундочку.
So I'm pressing the button. So what I can see is that your connection quality is poor and you're highlighted with red. So
Ну, тогда
докладчику.
okay, so maybe we can move on. Maybe we can move on to the next speaker. Yes, I think so too. Then, sorry, we will not be able to show your expert.
keep this presentation due to technical problems, and they move to the next speaker, Dr. Bijay Kumar. So please show profile. IT team, they are waiting for profile. Thank you, Dr. Kumar. You have 20 minutes. Please share your screen and start presentation.
Thank you. Hope it's okay.
Yes, they can see your presentation and they can hear you.
Yeah. Good evening to all. The topic of my presentation is sustainable marine fisheries and marine biodiversity conservation in blue economy paradigms. And in this presentation, I'm going to give you an overview about the global and Indian marine fisheries production and the increasing role of aquaculture and the blue growth initiatives and international commitments such as UN Sustainable Development Goals and the primary conservation threats and how we can bring in the sustainability element when we think about blue economy and blue growth, and then blue growth and sustainable fisheries initiatives in India, and then what is the role of networking internationally to achieve the goals. And if you look at the global marine fisheries production, we can always see that aquaculture accounted for, in the last few years, the aquaculture has a prominence in the global marine fisheries production. And if you look at the number of people, remain connected with the marine fisheries as far as livelihood is concerned, this is over 3 billion people. And out of that, 12% of the world's population depend upon fisheries, and also more than 90% are small-scale fisheries. And that speaks about the importance of marine fisheries, mainly for catering to the need of the people in the developing nations. And again, the importance is actually the women employment, almost 14% globally on an average. But the number increases when you look at the people, the women involved in the value chain process as well. And you can see that the 40% of the coastal fisheries are responsible for nearly 40% of the world's wild-caught seafood. And also, when you look at the future of the UN Sustainable Development Goals, and when we plan a world without hunger and malnutrition, and definitely we have to sustain the fisheries, and over half of the catch in developing countries is produced by the small-scale subsector, and the 90 to 95% of the small-scale landing are destined for local food consumption. And that clearly indicates that the small-scale fisheries also ensure nutritional security and food security of the people in many parts of the world, especially in the third world nations. And when we look at the sustainability of the Indian marine fisheries, we can see that India is the second largest producer of fish in the world, employing over 14 million people in fishing as well as aquaculture. And when we look at the production scenario in the last few years, we can see that gradually the total fish production in India is gradually increasing from 10.76 million metric tons in 2015-16 to 14.16 in 2019-20. And more importantly, when you look here, then you understand that the trend of the percentage of growth of GVA of the fishery sector, and you compare it with that of the agricultural sector, and this is almost 5 times more. And that means fishery sector is very, very important in terms of the delivery of the bioresources as well as the export value as far as India is concerned. But when you look at sustainability, and if you compare the marine fisheries landing as well as inland fisheries landing, what you can see is actually it's almost plateauing. The marine fisheries production from the natural water bodies is almost plateauing. At the same time, the total fish production in India is increasing primarily with the contribution of inland sector, and that too from the aquaculture side. Okay, so if you look at so that is the reason why when we think about blue growth and blue economy, we need to have a stronger orientation and focus to sustaining the fishery resources, especially from the oceans here. And this is the production trend of the marine fish landings as far as India is concerned. And you can see that there is almost a plateauing here, and there is a possibility of increasing deep sea fisheries. And also you can see that major part, almost 83% of the landing is contributed by the mechanized sector. And also the marine fishery resources contribute considerably to the foreign exchange earning of India. And in 2021, the contribution was almost 5.96 billion US dollar. Okay, despite the fact that there is a slight decline of 11% due to pandemic and the restrictions imposed by that. And here also you can see that the capture fisheries contribution reduced from 56% to in terms of to 36% over the last few years. That means year after year, the contribution of aquaculture sector is increasing. At the same time, the contribution of the capture fishery sector is declining. Again, reinforcing the fact that there should be a kind of sustainability in marine fisheries production. And you can see that the gradual way in which the aquaculture is contributing considerably, and 2030 projections indicate that there will be 62% contribution from the aquaculture sector, and that indicates the requirement for sustainability again. And if you look at the Indian government initiatives, as you are aware, now the government of India has launched Pradhan Mantri Matsya Sampada Yojana, and this Sampada Yojana clearly indicate or with a stronger focus on enhancing fish production to 20 lakh million ton by 2024-25. And for this, already more than 3,000 Sagar Mitras have been employed in the coastal villages, and they will look for the strengthening the marketing network as well as enhancing aquaculture production, so that India can excel in the years to come. And then look at the international commitments for India. You look at the Aichi targets 2020, which we rather failed to achieve. And also look at the UN Sustainable Development Goals, where the goal number 14 is actually the conservation of life below water. So here the basic question, also you look at the United Nations Sustainable Development Goals, and the first one and second one is linked with the no poverty and zero hunger, where the small scale fishery sector or the capture fishery sector can contribute considerably. And in addition to that, many other the developmental goals are linked with the biodiversity or bioresources. And 14 is exclusively dedicated for the conservation of life below water. And look at the COVID-19 scenario, and the COVID-19 scenario reiterate the fact that the addressing poverty, addressing malnutrition is one of the major portfolios for the governments as well, and which indicate a more thrust towards nature-based solutions, as well as looking at the ways in which we can invest more on the aquaculture or fishery sector. And when we think about blue growth and blue economy, and the paradigms are very clear, and the indication is for the enhancing fisheries, enhancing aquaculture, as well as the other resources, non-living resources also from the ocean. And then the question is that, throughout the world, there is a strong focus on blue growth initiative. And like the green economy principles that preceded it, FAO's blue growth initiative emphasizes the 3 pillars of sustainable development: economic, environmental, and social development. And also this is inevitable for attaining the 2030 United Nations Sustainable Development Goals. And here the question, when we look at the blue economy and blue growth, and then you look at in the perspective of the conservation, and then how will you address the issue of conservation paradigms, especially use of sea and marine resource for sustainable economic development. And this is a point of discussion in the coming slides. And the major reason, when you look at the major threats to sustaining marine fisheries, we can see that there is actually the increasing fishing pressure, and that is evident in India as well. And then we have to address the issues, posed by climate change as well. If you prioritize the issues one by one. And then climate issues are actually multifold, and the impacts of climate variability on fisheries is actually multifold. It impacts fisheries, ecosystem, and also the sustainable livelihood of the people. And also the impacts are much stronger, and like the IPCC latest report revealed, the impact will be much severe, especially in the Arabian Sea, which was actually cooler, and now it's becoming hotter, and the number of incidences of cyclones are also increasing, which indirectly impact the fishermen's livelihood, as well as it also damaged their gear and increased the risk of the sea, and also flooding in the coastal communities. Sea erosion is also a stronger issue in Indian coast, and that means we also have to remain adapted with the system of climate change, and that means we have to invest in that scenario as well. And the major fish which contributed to the marine fishery of India is the oil sardine and the longiceps, and there are indications that there is northward migration of this species because of the sea surface temperature enhancement, and also coupled with there are other issues like the development of oxygen minimum zones across this zone of the Indian Ocean. And finally, there is also issue of heavy fishing pressure on the juveniles along this region. And that is the reason why this is a typical case of environmental influences on the production of a pelagic resource in Indian coast, a drastic decline in production in the last few years in Indian coast. And also we need to address, in order to ensure the availability of resources, we also ensure the sustainable ways in which the fishermen live in the coast. And there are large number of major coastal areas which are under threat because of the sea level changes which is happening now and which will happen in future as well. And you can see that the Wildlife Protection Act of India enlists large number of marine species, including fishes, which are in the Schedule 1 and Schedule 1 to 4 of the Wildlife Protection Act of India, which means they are all protected because of a large number of reasons. And typical case is as that of the seahorses or the syngnathids in general, because of overexploitation and the fishing pressure, and also illegal export, they are on decline. And also we need to have a stronger strategy in managing larger fishes, predatory fishes, including the sawfish, which are much rarer than the tiger in India. And also there is a growing concern of marine debris, and there are increasing evidences in India which show that marine debris is a major problem, and the microplastic and nano particles have become a serious issue to deal with, especially when you look at the resource availability, also the safety of the marine resources available. And the details of the marine invasive species is not studied in detail, and this is a typical case of the marine invasive species, bivalve mussel called the charo mussel, Mytella strigata, gradually spreading into all the estuaries along the west coast of India, and it cover the clam habitats where the clam mining and as well as the fishery was prominent, and also it's impacted the green a mussel fishery along the estuarine regions of the southwest coast of India. A typical case of the marine invasive species impacting the livelihood as well as the fishery resources in Indian coast. And now the people have started using it as a resource, one way in which it can be managed, even though it's rather difficult to control it fully as exemplified in other regions. Now, how to address the issue? The primary issue is when you plan about sustaining the fishery, restricting the fishing capacity to sustainable level is one other issue. For example, it is estimated against the recommended fleet size that overall marine fleet size in India is excess by about 125%. It clearly indicates that, in addition to the carbon emission and the carbon footprint, increased carbon footprint, we need to have some kind of a carrying capacity based operation of this fishing vessel so that resource availability can also be managed. In addition, there is actually a need for addressing climate change and preparing for adaptation and mitigation and linking it with the livelihood of the people. So this is one of the challenges. And also how to bring in the local communities, including the fishers, in conservation initiatives, and promoting co-management and community mobilization. This is already happening. That's a positive sign. And many of the coastal villages, the fishermen themselves are coming up with different community-based management plans, including for marine turtles, which are endangered. And also now there is an issue. Another positive thing is that there are few examples of green certification, especially by the Marine Stewardship Council. And the first certified fishery is the Ashtamudi short-neck clam, already certified, which is a fishery in the Kerala state of India. And also the process of certifying the tuna fishery, the pole and line tuna fisheries in Lakshadweep Island is also in full swing. It's almost in the final stage of completion. And with the help of communities, the UNDP has already tried installing the square mesh net in the trawlers. They have installed it in more than 300 trawlers. And this is one way in which we can reduce the bycatch as envisaged by fisheries management strategies. And also in Kerala coast of India, the government, the fisheries department has already imposed a minimum legal size of commercially viable species, another way in which we can manage the fishery sustainable level. And also, when you think about the blue growth and blue economy, there should be actually a focus on the small scale fisheries, particularly because global food security and nutritional security are linked with promoting sustainable fisheries, small scale fisheries at a larger scale. That is identified by FAO as well. And also sustainable development goals clearly indicate this in 14B, which provide access to small scale artisanal fishers to marine resources and markets. Okay. And how do we achieve it? There are a large number of strategies to achieve it, and primarily is actually by meeting the social security objectives. And the social security objectives, particularly the food and nutritional security of the fisher folk, are critical to ensure participation as well as involving them in sustainably managing the resources. And the states have a key role in incentivizing and providing strong support system. And also we have to deal with the issue of subsidies in industrial fisheries. At the same time, we need to provide the subsidies for the fishing community to access the trade and business, so that they can remain equipped in managing the fisheries. And when you look at the conservation and sustainable use of biodiversity, and the principles of a convention on biological diversity rests on three pillars: conservation, sustainable use, and access and benefit sharing. So we need to have a stronger focus on that and identify the key biodiversity areas and increase the area of the marine reserves in Indian coast, and also involve the local people in the coastal protection and climate adaptation so that the fishery availability may be sustained in the long run. So these are the areas where India need to focus. And also a large chunk of the fisheries, marine fisheries, is wasted in the form of bycatch, and we need to promote a kind of utilization of bycatch, as well as not wasting any fish, because small fishes, particularly the from the small fisheries, are rich in nutrients, as exemplified by so many examples by FAO. And we need to promote the use and the creation of value-added products and from these resources in a sustainable manner. And also, there are a large number of examples where the fishermen themselves are employed in promoting tourism. And there are a large number of opportunities as far as managing the resources are concerned. And also it is one of the pillars for supporting blue economy and blue growth. For example, ecotourism, cultural tourism, including academic tourism, where the seminars like this should promote the exchange of personnel, including students between the Russian and the Indian universities. And this is one typical example with which we can promote the whole concept of blue growth. And also, when you look at the financing sector and how the ocean economic sectors perform, and there are some critical roles where the countries like India and Russia can collaborate. And one is actually the developing knowledge and creativity. And the creativity play a very, very important role, especially how to use the technology for bettering the blue growth and blue economy, and also how we can use the natural capital to sustain the income in the years to come. And the policymakers must engage researchers and stakeholders to promote evidence-based collaborative planning that ensures that sectors are chosen carefully, that local benefits are prioritized, and that blue economy delivers on its social, environmental, and economic costs. So this is one area where we need to have a stronger focus in the years to come. And Indian government has already brought out a blue economy draft policy, and the thrust on sustainable blue economy and knowledge and natural capital is now very much necessary, in addition to meeting other objectives which is already set in the blue economy paradigms as far as India is concerned. And when you look at the blue economy per se, rather than focusing on the industrial part of that or economic part of that, the original definition clearly indicates that blue economy is a sustainable use of ocean resources for economic growth, improve the livelihood and jobs, and ocean ecosystem health. That means when you look at the economy on one side, on the other conservation side, we need to focus on improved livelihood and jobs as well as ocean ecosystem health. And that is the reason why, sustainable management for the blue economy paradigms are more important in the Indian context as well. Then we have also seen the previous sessions, what is actually the role of genomics in conservation. And we are yet to actually begin in a larger way in India. Particularly these kind of collaborations can play a very, very important role in understanding the role of genomics in conservation, especially when we have a plan to revive and restore and reduce species loss with the genetic rescue plans. And we should have some models, better models in India using the modern technologies like this. And finally, when you look at sustainable blue economy, and the special focus by India should be on, one, the Gandhian approach of balancing economic benefits with sustainability for meeting broader goals of growth, employment generation, equity, and protection of environment. And also, they expand the protected area network as a measure against vagaries of climate change. And India must focus on marine ICTs, information communication tools, and transport and communication services, and creation of a knowledge hub for marine research and development. And creation of a knowledge society is very, very important in meeting these objectives. Okay, and these are the primary recommendations from our side. And when you look at the future, especially the coming 10 years, 2021, 2030, these are the ocean decade on ecosystem restoration, and as well as United Nations decade of ocean science for sustainable development. That means we should have some very clear-cut programs to move ahead, particularly to meet the target of the blue economy and blue growth. And this is also a time when we should have a national policy on marine science, and also a clear strategy to promote biodiversity science per se, where this kind of networking play a very, very important role. And when you compare the biological effect of protection and economic effects of protection, especially when you demand more protected area networks, and it ensures habitat protection as well as increase the supply of the resource. In biological, the protection of marine system will ultimately economically beneficial to the system in some way or other, and this has been proved by examples from throughout the world. Okay. And also, India should be actually a part of many of the international network. At present, they are to some, but we need to expand the collaboration, especially with larger, networks like networking with international initiatives like Marine Biodiversity Observation Network. Because when you look at the future, developing common framework for essential biodiversity variables play a very, very important role, and this will ultimately help in attaining sustainable development goals as well as managing the resources in a sustainable manner for future. Thank you.
Thank you, Dr. Kumar, for this comprehensive review and useful suggestions for the sustainable marine fisheries and conservation of marine biodiversity. I think we have time for one question, if someone would like to ask a question. No questions? Okay, thank you very much again. And let's move to the last but not least speakers, two speakers in fact. Last presentation, please show profile. Окей, я не услышал, что вы сказали. Share your screen and start presentation. You have 20 minutes from now on.
Ген, иди помоги мне. Ну вот здесь как это самое?
Или демонстрация.
Какую кнопочку?
Демонстрация экрана.
Please turn on the screen demonstration, screen sharing. Yes, please press the screen sharing on the bottom and then first launch the presentation. Just launch the PowerPoint and then press screen sharing and choose the tab with the screen sharing.
Так, что-то я...
Да, да. И разверните, пожалуйста, презентацию.
And also please unfold the presentation, please launch it. Please put your presentation into full screen mode. You chose the wrong screen. Choose the square with the presentation. The screen sharing one, right? You can leave the demonstration and launch the presentation. And then press the screen sharing. And it will offer you a choice of different options. So far it's not offering anything, it's offering screen sharing, and that's it. Have you opened the presentation? Yes. Please press screen sharing. Yes, please press the screen demonstration. Screen sharing. I can only see you. Because you chose this screen, you should have a different screen with the presentation to demonstrate it, because you're just demonstrating me. What do I need to press? I only have a screen demonstration. Please choose the one with the presentation. I don't have the word presentation in it. Not presentation, but your presentation which is open. Choose the PowerPoint with the open presentation. I opened the presentation. Can you see it? No. Please try closing different windows. It demonstrates, it shares what you have on your screen. You have to find your presentation to share it. I found it, but here on the screen I cannot find it. It says participants, chat, reactions, settings. No, your file. Try the bottom tab, the bottom line, where you have folders. There. Just open your presentation. Yeah, I'll open the presentation. Do you see it? Yes. And now press again in the lower part. Find the Zoom conference. You'll see this blue thingy with camera, the zoom sign. All of this should be on the bottom, in the bottom chart, in the bottom bar. Do you yourself see your presentation? Yes, I can see it perfectly. Please move your mouse and just go higher up. The bar in the top part of the window, and you should find the demonstration there. No? So what should I open? System, dynamic, microphone. Choose camera. Is this where I should be looking? Here I have webcam and mirroring. It should say demonstration in Russian.
If you can conclude, then we can move on and have this presentation maybe later.
I'm sorry, I cannot hear you clearly. There is not enough internet that shows this low capability of internet.
I'm saying Professor Shadman's problem can be sorted out offline.
No, still can. I still cannot hear you. Just some noise, some sound.
Can you hear now?
Видно,
Да,
нет?
вот сейчас вы запустили, да, правильно.
Yes, you finally launched it. Do you see the presentation itself? Just launch the presentation now and you can start.
Dear colleagues, I want to present you.
Разверните ее.
Please unfold it, launch it. On the bottom there is a sign launch presentation. Where you see the number 70%, next to it is a button to launch the presentation. So it would be full screen.
Так, что-то.
Вы опять выключили демонстрацию экрана. Не надо уходить с этого, не надо выключать ее. Ну, тут еще потренироваться надо было заранее, потому что очень долго.
You turned off the screen demonstration again. We cannot see your presentation again. Can you hear me? Nikolai, can you hear me?
Dr. Shadrin experiences the technical problems. Dr. Shadrin can... I'm sorry, Dr. Shotri, I cannot hear you, and because your internet is very low. Your internet shows very low. I cannot hear you clearly. Only some words. And I'm sorry. I suggest that skip this presentation because it seems like Dr. Shadrin has technical problems and he cannot start his presentation. Cannot launch his presentation. So they have, I think they nearly finished skipping 2 presentations. They nearly finished our session, and they still have in our program the panel discussion. And I wanted to ask Dr. Shrotria maybe to say a few words, but unfortunately I still see that your internet is very low, and also your screen is not very good. I mean, the video is very slow with delays. So maybe, I don't know, is it Wi-Fi? So
it's not Wi-Fi. Are you able to hear me now?
Yes, now I can hear you. So may I ask you to say a few words about this on the panel, in the beginning of the panel discussion?
So thank you very much, Dr. Tenko. It was a wonderful session we had since morning, and all the speakers presented their part. The aim of this webinar is to bring together partners from both sides. As many of the speakers mentioned their work, and they said that there is a lot of possibility for collaborative effort. As you also mentioned, we have had some interactions between FEBRAS and National Institute of Oceanography and a few other institutes. We request that these projects are taken up further. The recording of this webinar have been recorded. They are live on YouTube and they will be further available. So I request that these projects could be formulated. There will be some research calls between India and Asia. 2 research calls are likely to be announced very soon. This was agreed upon during the joint working group meeting between both sides, which was last year. And similarly, there are other multilateral programs which are also available, like BRICS. And some researchers are also participating in mega projects mega science projects of Russia. So that possible also there. Besides that, both sides are also very keen to promote entrepreneurs who are towards solutions and commercializing these technologies. So that partnership is also available, and we have some supportive program for that. One supported by FASI, your Federation for Department of Science and Technology India is already on, and startups and small and medium enterprises working in various fields. The fields which we are discussing, requested to kindly go through the call and formulate their proposal for and election. Request Dr. Constantine to kindly give his concluding remarks and ask other speakers to give their concluding remarks.
Okay, Dr. Shruti, thank you so much for this summary. And before I give my remarks, may I ask some other participants if they may have some other remarks or comments on this session? If anyone would like to say a few words or comments on your impressions.
Yes, can I say something?
Yes, sure. Yes, go ahead, please. Yeah.
Yeah, this is Dr. Baban Ingole from the National Center for Polar and Ocean Research in Goa. Thank you very much. It was wonderful webinar and also good exchange from both India and Russian colleagues. Particularly, I was very much impressed of one of the presentation. Most of them were from the Russian side was on the aquarium, because they have been doing wonderful job of breeding some of the exotic species. It is not easy to maintain it under that temperature and that conditions, and also breeding. It has got huge potential both from commercial fishery point of view, also from the pharmaceutical point of view, and third from the tourism and aquarium keeping point of view. And this probably we can take forward about the techniques and the training of some of the youngsters, if it can be explored and taken forward, because it is immediate and it can be taken at with low inputs, if I can say it from financial point of view or anything. But of course, administratively, it has to be seen it how. But I'm sure it has huge potential both for the both country, and expansion is possible. Thank you very much.
Thank you, Dr. Baban. And maybe someone would like to make more comments or some remarks, final remarks.
Dr. Litvenko, you are the director of that aquarium also. And
I already left this position and come back to the National Center now. So I'm in charge of International Cooperation Department again. They have another director there. But the aquarium is indeed a branch, is a part of the National Scientific Center. And they are trying to develop the research there in aquarium. So it's not just, aquarium for public. And you have seen that there are good examples of aquarium studies for the reproduction of this fish and so on. So it's important research for fisheries, but also they do some other research, especially the aquarium. This aquarium is the only place for keeping the big mammals. So they have mammals for show in this aquarium, and they have mammals for research. So they can do some physiological research and reproduction. And you have seen that they have very good experience in the birth of the beluga whales and so on. So it's one of the potential for collaboration. So in fact, it's a research facility too, as well as education center for the public. So if there's someone, I would like to invite the other participants for the panel discussion. So if you have some comments. Dr. Sherin?
Yeah, yes.
Okay, welcome. You're welcome.
Yeah, thank you very much for the opportunity. And indeed, it was a very good presentation. So at the onset, I would like to say that, CMLR, Center for Marine Living Resources and Ecology, which is in Cochin, is mandated to study the marine living resources of the country, especially from the deep sea. And as I presented, we are in the process of establishing a referral center and a museum for deep sea resources. So now we are also establishing a center. It is like a study center, or a knowledge hub for the marine mammals also. So I would like to get the technology or the know-how how these skeletons of the big marine mammals can be exhibited or displayed for the purpose of knowledge sharing. So I would like to get some technology in that subject. And another one thing, as our secretary, honorable secretary, in the welcome address, what he has mentioned, that government of India is moving ahead with ambitious deep sea mission. So there, we are one of the main player in inventorying and documenting the marine biodiversity of our Indian EEZ, especially in the hotspot areas like Andaman Sea. So now we are looking for, non-invasive method like deploying ROV submersible. So I would like to get some of the specifications or the technology. What is, in Russia, how they are doing all these deep sea sampling without any other destructive practices like trawling. Without that, how best we can make use of the ROVs and submersibles. So I would like to share that specifications, because we have the budget for procurement of ROVs. So these two things, if I am able to get some information from the Russian side, I'll be very happy to collaborate more on that. On behalf of CMLR, I am very happy to collaborate. Thank you very much. So I look forward for this collaboration to become a very fruitful and productive way to see the fruit of our research. Thank you.
Thank you, Dr. Sherin. Yes, I was also impressed by the deep sea research in India and in several presentations. And, in the National Center of Marine Biology here in Vladivostok, 11 years ago, they started a deep sea program. And they have two research institutes. Another one is Shershov Institute of Oceanology in Moscow. So they use the manned submersible and they use unmanned submersible. So they already have some experience. And Dr. Shotri visited us several years ago, and he has seen our submersibles here. So there is a deep sea program jointly the foreign countries, currently mostly with Germany, and they are interested to share this experience. And I'm sure that in future they can find some joint points for research and some, platform to for discussions and also for collaboration. I would like to invite other participants of the meeting to join to the panel discussion if someone have suggestions or a comment. Okay, Professor Balasubramanian, I'm sorry for my pronunciation.
Yeah, so thank you, Chairman. Actually, today's program witnessed a wonderful flow of information on the science of the biodiversity, both in the surface as well as deep sea biodiversity. And all my colleague scientists has given a wonderful flow of information. The Russian side also, we got a amount of information, especially the aquarium is very attractive. And in fact, India is also on the move of constructing aquarium, even under public-private partner mode also. So there is a lot of scope for collaborating between India and Russia on deep sea biodiversity, sustainable utilization, as well as my colleague, Dr. Bijay Kumar, he has made an extensive approach to meet the sustainable goal. So probably we need to work out the strategies, how best we can make it. And coming to your presentation, I was highly impressed. And in the way in which you could be able to construct, being a geologist, you could be able to make out from the shells the change in the habitat. Probably, maybe we can also involve that kind of studies in our Indian waters, because we have a lot of submerged areas, and there we may be able to get plenty of different species. And although we are one among 8 megabiodiversity country, I'm sure considerable effort is needed to document all the biodiversity sources in Indian waters as that of the Russian waters and other waters. Then probably, definitely the bilateral collaboration will prove a better approach for mutual benefit of both the countries. Thanks for the opportunity. Thank you.
Thank you for your comments. And if someone would like to join, okay, Professor Kumar, please, you're welcome.
Yeah, thank you. Thank you for this opportunity. And I think there are, this opened up, the venues for collaboration. And one thing definitely is actually, in terms of, exchange of technologies, which is already mentioned, including an oceanarium. And, this is, I believe, is one of the handicap in furthering the marine education in India and awareness. And one state-of-the-art aquarium or oceanarium is one of the things which is required in India, especially when we look at the coming decade. And second is actually to strengthen the databases and where the tax.
Oops, there is some disconnection, I think. Okay, so if there are some other comments from the floor, so maybe they are going to wrap up our meeting, and I would say several comments too. And I was very, it was exciting meeting for me. And during the last year, they hold 2 seminars in between the Indian scientists and the Russian scientists. And today I learned much more, and I see that these important studies about the monitoring of the marine environment and the biodiversity, and they are progressing is very well, and deep sea biodiversity and sustainable fisheries. So I think there are many there are many points for interaction and collaboration between Russian and Indian scientists. And I'm very pleased that Russian scientists took active part in this meeting, and Indian scientists presented a lot of new information. So I suggest that the Indian embassy would continue their efforts to to develop, the collaboration between the scientists and of our 2 countries. So thank you very much, the Indian embassy, and thank you all agencies involved, and thank you Russian Academy of Sciences and International Department in Moscow of the Russian Academy of Sciences for organization of this meeting. I'm sorry again for some technical and other problems in the beginning of this meeting. This is because the part of the organization team is in Moscow, part in Vladivostok, and it's not easy to sometimes to interact and arrange. And for 2 presentations, we have not been able today to show you. I advise the organization team, the international department in Moscow, to contact these presenters, so maybe they can present later tomorrow or in other meeting. So thank you so much, ladies and gentlemen, and we would be glad to meet you online, and not only online, and face-to-face after the COVID-19 situation will be over. And I hope that this kind of meeting is an important step to go forward in our collaboration. So maybe international department from Moscow, from Russian Academy of Sciences can say some words, can say some comments. Irina?
Yeah, I just want to thank you, Konstantin, for beautiful moderation of the session, and all the participants for their efforts to participate. Sorry, once again, I bring our apologies for the not very successful beginning. But I think that tomorrow we will make the technical part of the conference better. Thank you very much. And I give the floor to Shishir Sharotria. I think that this is time to begin the second session. You're welcome, please.
Thank you, Dr. Ludenko. Thank you, Ms. Sinha from Russian Academy of Science. You have been working very hard to put up this program, and this has really come out well. Now I request the organizers to put up the slide for introduction of our moderator, Dr. G. Dharani. He's from NIOT, Chennai, and he will be taking the moderation for the second session, which is on marine chemistry, marine biotechnology, and marine drugs. A very important session. So may I request the organizers to put up the introductory slide for Dr. Dharani. Dr. Dharani, you are not visible. Could you turn your video on, please? Yes.
Namaskaram, sir.
Namaskar. Dr. Dharani, to take forward the session 2 as the moderator chairperson, please.
Yes, sir. Thank you very much for providing this opportunity, and thank you for giving the proceedings to take over, sir. Already, I think we are slightly running late. So with your kind approval of the organizers and MOS, can we go inside this session, sir? So,
yes, please. Yes, please.
Yes. On behalf of the organizers and Ministry of Earth Science and Indo-Russian embassies, I'm very much thankful for conducting this conference, and I welcome all the participants, the key speakers, and the panel discussion speakers for the session 2 of marine chemistry, marine biotechnology, and marine drugs. I would be glad and would be happy to moderate these sessions, and we have the first talk by Dr. Dimitri Amini, and I request Dr. Dimitri to start his presentation.
Hello, are you hear me?
Yes, you are clear and loud. Sister, can we give the platform to Dr. Dimitri, sir?
Yes, I think he's loading his presentation.
Yes.
Okay. Clear? Okay.
Надо сначала открыть презентацию, а потом демонстрацию экрана нажимать.
Вы меня не видите?
Мы вас видим, и ваш компьютер видим.
Can you see me? Well, we can see you. Just please open the presentation first and then push the screen sharing button. Okay, my presentation is open.
before Dr. Dmitry is going to speak, I
Мы
will just
видим
have
проводник. Надо выбрать другой экран, где
I'll be happy
презентация.
to introduce him. Dr. Dmitry is a member of RAS, head of the Laboratory of Bias and Mechanism of Action of Biological Active Substance, Pacific Institute of Bioorganic Chemistry, FEB, RAS, Vladivostok. And his talk is going to be on systolic heart failures, structural activity relation studies, and 1,2,4 triazole to develop novel cardiac myosin activators.
Okay, I cannot see my presentation on the screen when I going to the screen demonstration. There are not my presentation, but I can
Вы
see.
этот экран закройте. Сейчас уберите
can-
демонстрацию экрана, закройте.
Just close this screen, turn off the screen sharing, and just open the presentation on your computer. Well, it has been opened, but not full screen. And now turn on the screen sharing mode. Okay, I can see this folder. Oh, you need to choose from your screen the presentation mode. Is it that? Just turn it on. Yes, we can see everything perfectly.
Dear colleagues, first of all, I would like to thank organizing committee for the possibility to attend this meeting. Okay, next screen. Okay, I want to introduce you with international research project, which is already applied to Ministry of Science and Technology, India, and Russian Science Foundation. This project entitled Systolic Heart Failure Treatment, Structure Activity Relationship Study of 1,2,4 triazolones to develop novel cardiac myosin activators. There are two principal investigators for this project. First is Dr. Manika Manoj from PSG Institute of Technology and Applied Research, Coimbatore, India, and me. Actually, Dr. Manoj initiated this cooperation and suggested us to participate in this joint investigation. According to the World Health Organization, cardiovascular disease, including heart failure, are the leading cause of death globally. In an estimated around 18 million people died from cardiovascular disease in 2019, a number that is expected to grow to more than 23 million by 2030. Heart failure mortality is similar to or ever higher than that due to various cancers. It's a leading reason of death among other cardiovascular diseases. At least half of the patients suffering from heart failure have a low ejection fraction, 40% or less, thus leading to systolic heart failure. What does it mean? The heart muscle becomes weak and enlarged. The measurement of how much blood to the heart pumps out with each beat is called ejection fraction. In systolic heart failure, the ejection fraction is lower than normal. This can cause blood to back up into the lungs and cause shortness of breath and eventually lung edema. It's a very, very dangerous. This is also called heart failure with reduced ejection fraction. It's still challenging to improve the cardiac function of heart failure patients with reduced ejection fraction. Currently available drugs or inotropes for heart failure cure are, for instance, adrenergic receptor agonists like dobutamine, phosphodiesterase inhibitors, for instance, milrinone, or calcium activators such as levosimendan that increase cardiac contractility at the expense of increased concentration of intracellular calcium and cyclic AMP, contributing to increase heart rate. But those drugs also induce hypotension, arrhythmias, and increase mortality. Cardiac myosin is a motor protein best known for their roles in heart muscle contraction. It's ATP dependent and responsible for actin-based motility and heart beating. Cardiac myosin activators, or inotropes, appear to accelerate the rate-limiting steps, fostering the transition of myosin from weakly filament-bound to the strongly filament-bound state, thus leads to increased force production. Recently, the new drug, omecamtiv mecarbil, or briefly OM, was synthesized. It's a 1,2,4-triazone derivative. It's a selective, first-in-class, and still single cardiac myosin activator. OM activates cardiac myosin and increases the ATP hydrolysis and accelerates the formation of strong actin-myosin complex linked to increased contractile force. This drug is being developed by Amgen and Cytokinetics, USA company, with financial and strategic support from Servier Pharmaceutical Company. OM is currently undergoing phase 3 clinical trials for the treatment of left ventricular systolic heart failure. Those trials are expected to complete in next year before it will be marketed. Also, the clinical study results showed that omecamtiv mecarbil is high effective in relieving symptoms and enhance the quality of life of systolic heart failure patients. A wide range of drugs with a new 1,2,4 triazolone pharmacophore that can improve cardiac function is still required. Dr. Monika Manoj from PSG Institute of Technology and Applied Research now is deeply involved to develop these derivatives as a novel cardiac myosin activators. It's clearly seen from his K publication during the last 5 years, which was published, which we published in high impact journals. His rational design and synthesis of triazole with a weak hydrogen bond acceptor, or HBA, and no hydrogen bond donor, HBD, showed poor cardiac myosin activation, around 33%. Here. However, introducing strong HBA and HBD as represented in 1,2,4 triazone showed patent cardiac myosin activation. At 10 micromole, it's equal around 99%. And this preliminary work is already done. We plan to prepare the combinatory library of new 1,2,4 triazolones, around 70 compounds with a different structure optimization. Some of them you can see on this screen. Overall, India-Russia joint program will focus to develop novel cardiac myosin activators and select a new drug candidates directly targeting heart muscle ATP-ASA enzyme for the treatment of systolic heart failure. Indian team and Russian team have different tasks. Indian team will focus on the design and synthesis of designed molecules. All the synthesized molecules will be screened for cardiac myosin ATP-ASA activation to find a novel lead molecule which can be developed into drug molecule. The enzyme assay will be done by both Indian and Russian experts. Selectivity assay on skeletal myosin and smooth myosin will be performed by the Indian team. Russian team will conduct an in vitro, in vivo, and in silico studies, including compound cytotoxicity studies using rat cardiomyocytes, compound influence on cell viability in hypoxia conditions, compound influence on intracellular calcium concentration, compound influence on cells hypertrophy or contraction, and compound influence on cardiomyocytes mitochondrial membrane potential. The selected molecules from in vitro assay results will be formulated for in vivo studies. What we will do in vivo, we will measure the fractional shortening and ejection fraction measurement by echocardiography technique using experimental rats. And in silico, we plan to carry out molecular modeling, docking, and QSAR studies. It will be done to support the molecular design. What we expect, this cooperation will give both the teams a chance to explore a novel cardiac myosin activators based on 1,2,4 triazolone scaffold as a pharmacophore. This could lead both the teams to extend the collaboration and forward the patent molecule to preclinical and clinical trials and commercialization as a drug for systolic heart failure. Since our session is dedicated to marine chemistry, marine biotechnology, and marine drugs, I would like to say that the ocean are the largest and richest source of biologically active compounds. The number of newly isolated marine substances is steadily growing. Since 2008, about 1,000 new compounds are regularly isolated per year. The total number of known marine natural compounds is currently about 30,000. About 60% of known drugs were created on the basis of natural compounds, their analogs or derivatives. A number of marine natural compounds are distinguished by pronounced and often selective and unique biological activity. Therefore, they are often good molecular scaffold for further synthesis, modification, in order to obtain more active products. It's believed that 1,2,4 triazolones do not exist among natural products. But recently in 2008, a new antibiotic substance was isolated from marine microorganism. It's Streptomyces, collected from sediments in the Mediterranean Sea of the Egypt coast. The compound named Esramycin was assigned as a 1,2,4 triazolopyrimidine, an extremely unusual structure for natural occurring substance. This 1,2,4 triazolone was isolated from the marine organism for the very first time. Therefore, we have a great opportunity to find out the new patent cardiac myosin activators based on 1-4 triazolone scaffold in marine biota. Using our scientific fleet facility, we can travel around whole world, collect marine animals by scuba or trawling, isolate chemical compounds and study their biological activity on board research vessel. And finally, I really hope that someday we will explore the Indian Ocean together with our Indian colleagues in joint scientific expedition to find out new and perspective and patent medical drugs. Thank you for your attention.
Thank you very much. Thank you, Dr. Dimitri, for a very brief and very short and crisp presentations. Thank you very much. And since we are slightly ahead of the time is slightly less, so I think we can call the second speaker, and we can have a discussion during our panel discussions on your presentations. Once again, I thank you, Dr. Dimitri Amni, for your wonderful presentations and your thoughtful insights on the cardioactive substance. And with this, I invite Professor Mohammed Khatta to prepare his presentation. In the meantime, before Professor would upload his documents, Professor, you can upload your documents, your presentations online.
Am I audible, sir?
Yeah, you are clear and loud, sir. You please proceed for
very good.
Yes. Presentations.
Yeah, sure.
And I take this opportunity to introduce Professor Mohammed Khatta. Professor? Yes. Professor Mohammed Khatta is from Department of Marine Biology and Microbiology and Biochemistry, Cochin University of Science and Technology, Kochi. And his topic would be bioprospecting of marine bacteria for novel marine natural product strategy for discovering and development. Welcome you, sir.
Yes, thank you, sir. Shall I go to the full screen and see whether it is working all right?
Yes, sir. It is working. As such, it is okay. Just you can try for the full screen.
As such, it is okay. Fine. Then it is fine. I will not modify anything. Yeah. So here, first of all, I would like to thank Indian Embassy in Moscow, Russia, Ministry of Earth Sciences, Russian Academy of Sciences, and Council of Scientific and Industrial Research, India, for giving me this opportunity to talk about the potential of marine bacteria in the discovery of novel marine natural products. In fact, we are living basically our Earth is not planet Earth, it is planet ocean, because 2/3 is ocean and only 1/3 is Earth. So definitely the ocean provides us with lots of resources, a lot of hidden resources, in fact, because exploring the oceans is definitely a difficult task. The logistics are really not easy to arrange. So it is really remain as unexplored, and it also provides a range of habitats. If you look at the Earth, I mean, the life is originated in the oceans, and the ocean provide moderate to extreme habitats, like you have the black spunkers or these rift valleys, you have sandy beaches, you have seagrass beds, you have coral environment, coral reefs, you have deep sea, you have pelagic, I mean, you have benthic environments, you have halophilic environments, you have, high temperature, low temperature. So it's plenty of variety out there. And the marine microbial diversity, if you look at, and the life is there, it is originated in the oceans. It is there in all these various niche, which I have just mentioned. Plenty of them are there. And still, the cultural diversity, as we know, is hardly 1%, because most remain hidden, or we really do not have the capacity, or we really not understood yet the complexity of the environment where these organisms are growing. So we are unable to provide the right kind of medium, or unable to create the right kind of situation in the medium so that you can grow lots of organisms. So it remains as a challenge, and this challenge is the greatest opportunity it offers. So I'm basically, my research is, I'm basically a microbiologist to look at the diversity. I have samples from the tropical to the polar, both Arctic and Antarctic. I have deep sea sediment samples, estuarine. I have a very good collection of organisms, which we generally look at the diversity and bioprospect and some of the things, selected enzymes or other things which we proceed further. And I'm also very keen to look at the pathogens, especially the mesophilic pathogens, food and waterborne pathogens. And one of my key areas is antimicrobial resistance. How do they spread? How the conditions are being created? How it is, a global issue. So here we really require these marine microbial natural products. I'll come to that on my next slide. So here, if you look at the marine microorganisms, they have been evolving and diversifying for billions of years, in contrast to microorganisms from terrestrial environments, because basically the life started in the ocean. And the oceans offer huge range of habitat, very extreme to moderate to other slightly haline and other meso level barriers. So here, why searching for marine microbial natural products is a very basic question, because at the first step I said it is not easy to go to the marine environment. It is very difficult environment as far as logistics is concerned, and getting a sample out there is tough task. Growing them, mimicking the conditions, and getting them in culture as a prerequisite is definitely very, very difficult. But the hidden or underexplored or unexplored nature of the thing makes it is a bounty for this kind of this kind of marine microbial natural products. For example, since 1970s, over 15,000 structurally diverse natural products with an astounding assortment of bioactivities have been identified from marine environments. And why we need it? Because we have a constant challenge or increasing challenge from multi-drug resistant pathogens. You require novel antibiotics, which are not keeping in pace with the evolution of microorganisms or evolution of pathogens. They are evolving much, much faster than we discover stuff. That's what we are experiencing now. And new and emerging, re-emerging pathogens, because the mutability of many of these pathogens is very fast, and there are lots of horizontal gene transfer mechanisms which give them permutations and combinations which we are unable to deal. They are emerging very virulent. They are emerging very fast spreading. They are emerging. They are mutating very quick. So we always find it difficult. So we need new things to target them: antiviral, antibacterial, and also anti-cancer stuff, and also re-emerging infectious diseases which require new drugs and solutions. So here, if you look at this is the globe as such. Of course, currently everybody has felt the power of the germs or the power of the microorganisms, because corona pandemic or coronavirus driven pandemic, COVID-19, it was a true pandemic in every sense. It has not even spared Antarctica. I have seen that Chile Antarctic Station had several people affected with COVID-19, and it has not spared high seas, the ships, people traveling in the ships, in the small oceanic islands, everywhere it is spread. The kind of spread was, it was a tremendous kind of, we never anticipated that kind of a spread. Apart from that, you can see lots of new viruses, new issues, Ebola there, E. coli, several strains of E. coli. I work mostly on E. coli 0157H7. I have several strains of pathogenic E. coli, very many strains of Salmonella, Aeromonas. So you have now drug-resistant malaria. Now, finally, after 50 years, we have come up with a vaccine against malaria, cryptosporidiasis, then human monkeypox. So many things are out there which is giving a great challenge. So here, what is the market value of, if you look at the commercial side of it, what is it, whether it really offer, you know, some of it we know now in COVID, after COVID-19, how come certain, you know, companies, even in India, like Serum Institute or Indian Biologicals, Indian Immunologicals, how they become, you know, in the limelight within no time, just become great companies, you know, helping lots of people by supplying the required amount of vaccine. So if you look at the market of marine pharmaceuticals, this is up to 2016, it was around 8.6 billion and growing at 12.5% compound average annual growth rate, CAGR of around 12.5%. And it is projected to 26.5 billion in 2020. It is already achieved. I think it has already crossed that one. And, you know, to 38.5 billion, 1 billion by 2027 will be growing at around, you know, compounded annual growth rate of around 8.5%. So that's a huge market to be tapped. We have lots and lots of, you know, upside in this particular, if you look at it as a business opportunity or a market where the requirement of these things. And you have several major players, you know, in this, like Seattle Genetics, DSM, Takeda, BASF, Amway, Pfizer, GSK, Nordic Naturals. There are several of them, and many of them are currently, if you look at the market share, basically nutraceuticals. Many of them, you know, because that doesn't require that kind of a stringent thing as a medicine. So it is, and the largest application is basically in anti-tumor drugs. And if you look at how we can do this stuff, so we are seeing a very optimistic, very future-centric research area where how we go about this. So this is, we need to collect samples, we need to culture and isolate, we look for the bioactivity, then we have to isolate the compound, characterize the compound, confirm the bioactivity. Then you go to the other testing levels, product development and testing, and finally commercialization. These are all very, very important aspects. If it is a medicinal molecule, you need to go through trial 1, 2, 3 different stages, and then finally commercialization. But the COVID-19 scenario, actually what we have seen, the commercialization of the vaccine took place in less than 2 years. So when there is a real need, the barriers are broken. The regulatory control is fastened in such a way that we achieve it at a very faster. Otherwise, it was taking 10 to 20 to 30 years for developing a normal vaccine. So here, why marine microbes? Again, you know, because oceans include greatest extremes of temperature, pressure, and light encountered by life. And life has to evolve, and the bacteria evolve much, much faster than any other organism. They evolve, adapt. If you look at, you know, the conditions which affect life processes, you see a huge range in when it comes to bacteria. You have from anaerobes, obligate anaerobes to obligate aerobes with a large stretch in between. You have got psychrophiles to hyperthermophiles with a large stretch in between. You will see acidophiles to alkalophiles through mesophiles and other range. So plenty of stuff. So here we have a lot of extremophiles. You can encounter them hydrothermal vents or pisophiles from great ocean depths. And, you know, those organisms living in association with, you know, there are several marine natural products coming from sponges or other organisms where many research says that it is a microbial symbiont doing the job, protecting the host. The host is providing the nice environment for the microorganism to survive and produce this stuff and share it with the symbiont, and both of them live happy. And the intertidal region is definitely a very harsh environment, periodic submerging, then, you know, drying, the tidal pools which are above average temperature, and the kind of extremes up and down, extremes rolling between up and down. It is sometimes extreme, sometimes it is getting to normal again. So this is a kind of nice kind of pressure for the organisms to evolve to various situations. And they represent an underexplored reservoir discovery of MMNT, so marine natural microbial natural products, and it is definitely, there is a lot of scope. So here, if you look at this slide, you can see how this marine natural product related papers are published in the PubMed. This is just the PubMed citations alone or published papers alone taken there. You can see marine microbial anti-biofilm compounds growing at this level. But if you look at microbial anti-biofilm compounds, this many papers, so a lot of upside here. And marine microbial natural bioactive compounds, you can see it is picking up pretty fast, and microbial natural bioactive products, all environments, growing pretty good. So here you can also see, in addition to plants and animals, microorganisms are a major reservoir for the discovery of this. You can see nearly out of these 900 plus natural products, nearly 600 of them are coming from bacteria, the antibacterial and antibiotics. And also you will see a lot of anti-cancer stuff now coming from bacteria. Antiviral is coming up, antifungal is coming up, because you are screening for these activities. Very, very intensive search is going on, considering the real need. So you are striking success in a very significant way. So here, how these approaches, how do you approach, to the discovery of marine microbial natural products? Your major, the first step is getting the culture in, getting the bacteria in pure culture is a prerequisite. Then you need, once you have it, you have, comprehensive physiological characterization. What are their properties? What all things they can do? Then you systematically investigate those organisms. So here, the major issue here is low culturability. As you have seen, you're able to culture only 1%, so 99% is hidden. So this is one of our paper based on this culture-based diversity from the high Arctic, very recently published. Another one also on culture independent we have published. So here, so limitations encountered while culturing marine bacteria. One is you really don't know what exactly this, their requirement we are unable to do. We are unable to understand. And the high seas or offshore is oligotrophic. So where you need to, the nutrient high, many of the media which are rich in nutrient unable to support. So you have to use low nutrient filtered seawater. And sometimes some of the organisms fast growing, the competing organisms. So you dilute it to a great extent so that you reduce the number of those competitors. And slow growth, for which many of them are slow growing, we need to give at least 2 to 3 weeks to 1 month sometimes. So you have to actually incubate them long, and poor cell density. So here you can actually highly sensitive screening like epifluorescence microscopy, flow cytometry could be used. And then some of them remain dormant. Again, you come up with the long-term incubation. Sometimes you provide the special needs by providing chemical gradient systems. And sometimes you put, because they will only grow in an attached situation. So you provide in situ colonization carriers. And sometimes they will only grow in association with other groups. So you co-culture them with the helper strains. And the complexity of microbial communities you can understand by high throughput cultivation screening. So here, when low culturability is mimicking the natural environment is very critical to recover the uncultivated organisms. For example, here I put one example like SAR11. This is a marine bacterioplankton clay, which is seawater has been used to culture previously uncultivated microorganisms. Seawater plus agar. You mix, you prepare a media, you grow them. You are seeing something which you have not seen earlier. So this is a good challenge for microbial ecologists to look at how you do those things, how you try to mimic, how best you come up with the solutions to mimic the natural environment. And other innovative cultivation approaches, like you can sometimes put supernatants of cell-free extracts from helper strains, because such extracts help these hidden bacteria sometimes grow by acting as a stimulating factor for the unculturable strains. And also you put them under extended incubation. You don't throw away the plate after 3-4 days or a week. You just extend the incubation time so that you probably see something is growing up, because their generation time is pretty long. So you get the colony, visible colony will appear only after quite some time. And other pre-treatment methods like enrichment, you can do physical dry heat. Sometimes you just heat up the sample before plating and then you plate. Sometimes you provide sucrose gradients in the medium so that some of them will grow nicely. There are several bacteria which have a specific taste for different gradients. So you even or your sample you process in sucrose gradient centrifuge, then take it up and then plate it. So here you can see, for example, Nocardia will be coming in samples which are, from a 20% sucrose layer, while Streptomyces from 30, 40, and 50% sucrose layer, or Micromonospora in 20 to 30% sucrose layer, Actinoplanes from 10% sucrose layer. So these are all, you are subjecting the sample for a density gradient using sucrose, and then taking samples from different layers, and then you plate it, you probably strike success with these kind of organisms. And there's also other pre-treatment for recovery of diverse actinobacteria, like we are actually currently have a good collection of actinobacteria from the fjord environments as well as the marine environments, which we are, some, I think 4 or 5 of them are showing good anti-cancer activity and antiviral activity. The compound is being characterized with the help of LCMS and other different tools to characterize the molecule. Hopefully we get something. And you can also provide a low nutrient medium. So here, marine microbes have specific nutrient requirement for growth. For example, sponge extract. So many of them, as in association with sponge, will basically love the sponge extract, or you can actually give sponge extract to enhance the culturability. Or chemical like cererophores, some of them require iron binding proteins in the medium, or some signal molecules like CAM, which is a very effective inducer, cyclic AMP, which led to cultivation efficiencies of up to 100% of total bacterial counts. Or you give artificial brackish water with the different carbon substrates, or provide electron acceptors, and you manipulate with the medium and see how it is working. And sometimes you change the ionic strength of the medium. Some of them require a high ionic strength, such as these organisms. Some of them require a medium ionic strength, such as these organisms. So these are some of the marine microbial bioactive metabolites which are effective against, MRSA or methicillin-resistant Staph aureus, which is a major problem not in India but in Europe. They really have serious issues, and even US also have serious issues. They then came up with the vancomycin-resistant organisms also, which is a problem. Vancomycin-resistant Staph aureus. This is methicillin-resistant Staph aureus. You see, marinomycin. These are all some of the things in the market: pederin, desferrioxamine, sporolides, then griseohodin, and eptidamnamide. These are all some of the things, marine microbial natural products already available in the market. And sometimes, the conventional like bioactivity guided screening, like you can grow the organism, you take the supernatant, you check for activity like anti-cancer, antibacterial, antiviral, antiparasitic. So the bioactivity is tested, and based on that, you will be going ahead further. If the activity is there, you go on to characterization. You can also use combinatorial chemistry as a very powerful tool for synthesizing, molecule once you identify the structure. And here, sometimes you use culture supernatant or supernatant or extract from the pellet. For example, marinomycin AD from saline culture extract of Marinospora. And this has got a significant activity against multi-drug resistant pathogens and cell lines. A fantastic molecule. And gene-guided activity here, you will be, targeting some of the genes associated with some pathways, like biosynthetic pathways with NP, natural products, biosynthetic pathways. For example, gene fragments between those coding for ketosynthase and methyl malonyl CoA transferase of polychaetous type 1. This set of genes are likely to express a kind of protein which is very likely to have an activity. So this offers great potential in the hunt for new marine microbial natural products. This predictive capability provides a simple and rapid method to avoid the isolation of non-compounds. One is you can avoid isolation of non-compounds, or reinventing the wheel could be avoided. At the same time, you also circumvent the issue related to culturability and other things, or to identify that strains which can produce compounds within the same structural class. So here I just put an image here. So this is your sampling marine environment. So in a gene props based screening, you are looking for the kind of gene which are likely to produce that bioactive substance. So you look for that gene and hybridize that, using a gene prop. You look for the presence of the gene and you actually identify this is a replica plating, and you will identify which are the colonies. So this colony here is having that gene which you are looking for. So this will be your target. You will not take anything from these four, but you will select this one. But bioactivity related, you have the organism, you do it in a fermenter, you produce this stuff, you take the supernatant, you do the bioactive screening based on the supernatant or whatever you want to do. Then you look at what is the activity, what is the characteristic of that particular compound, whether it is a novel, whether it is and you also identify the organism based on its 16S gene. Then you will rRNA gene, then you will see whether it is novel microbe or whether it's an existing microbe. So here, so culture-independent methods, this is gene-targeted methods, basically offering now most people, there are several people looking at metagenomics, which are basically culture-independent. You straight away go to the environment, extract the DNA, environmental DNA, and you try to identify whether you make the gene library, and you probably you screen it. You have different next generation sequencing platforms which will do the sequencing and match with the library and tell you this is something novel, this is something exciting, something new species or whatever. You can also look at the genes. You can put them in a surrogate host and express it and see whether this particular gene which you are looking for, whether producing any new stuff. So there are two approaches to metagenomics. One is sequence driven, one is function driven, right? So here in sequence driven, the DNA from the environment of interest is sequenced and subjected to computational analysis. And the metagenomic sequences are compared to the sequences deposited in publicly available databases such as GenBank. And these genes are then collected into groups similar of a predicted function. In the function driven, you have the DNA and move a piece of them into surrogate host and see what is its function. And for that function must be absent in the surrogate host. You are looking for one particular kind of function. So that particular kind of function should not be there in surrogate host. You fix in that new gene into surrogate host and look at it whether now the surrogate host is producing that property, then you are striking success. So here, this phylogenetic analysis all there. It is we look at taxonomic diversity, which will help maximize novel taxa recovery in culture, because when you know there are so many things hidden, you also have a chance to try to grow them, recover them in culture. This is our couple of recent paper, developed or published based on that research. And functional gene-based approaches, this is a highly powerful tool. And here you will target genes for specific functions of the environmental DNA, and the gene coding for key enzymes in various environmental processes have been studied in the marine environment. And this is the drawback is your database is very limited, so you don't have many information about the functional gene diversity when compared to 16S rRNA, but you will keep adding. People are doing metagenome research in a lot of laboratories world over, and always strengthening the library, and hopefully we'll have a good library support. And lack of accuracy in taxonomic assignment due to lateral gene transfer, because microbes always do this horizontal gene transfer, some of them sometimes fixing some new stuff into the organism which you are looking for, and you have a confusion. So here, linking phylogeneity, this is another very powerful approach, like a stable isotope probing is the first method to be developed in this direction. I mean, actually, the
Can you please consolidate?
Sure. This is you. Yeah, just 1 minute, I'll finish it off. So you label the substrates with stable radioactive isotopes, and whether you see whether the organism is incorporating it in them, right? So that will help us, find whether they are really creating some stuff, some new stuff with your stable isotope incorporated in that, could be screened easily. So this is basically which I have discussed, and this is the two aspects. Function driven and gene driven. So these are some of the success stories for terragenes, violacin, indurubin, turbomycin. These are currently in there, and these are those compounds and the methods, recent methods which they have used to get these things. So thank you, everyone.
Thank you, Dr. Mohammed Atta, for your very nice and elaborate speech. And since as we are running short of time, we will proceed to the next lecture, and we'll have a discussion at the end of the panel.
Yes, sure, sir. Thank you so much.
Thank you very much, sir.
Yes, sir.
May I request the presence of Professor E. Ponnarivu or Professor A. Carson-Nikova?
Поддержка, давайте, пожалуйста, профайл Красильниковой и Пономаревой.
Yes. Thank you, Professor.
Hello, dear colleague. You hear me?
Да, мы вас слышим.
Thank you, спасибо. Dear colleague, let me introduce our report about scientific development in the field of cryopreservation of fish.
I'm sorry, may I ask you to make your presentation full screen? Сделайте на полный экран, пожалуйста. Это рядом со шкалой внизу в правом углу, рядом со шкалой значок полный экран.
Не вижу.
В самом низу, в правом, да, внизу, еще правее, вот здесь рядом. Да.
Go to the bottom.
Dear colleague, let me introduce our report about scientific development in the field of cryopreservation of fish stew. To date, low temperature preservation is one of the most affordable and acceptable methods of low-term storage of cells. Astrakhan State Technical University, together with specialists of the Federal Research Center, Southern Scientific Center of Russian Academy of Science, conduct a long-term scientific research on the development and improvement of method of cryopreservation of reproductive cells of various species of fish of the southern seas of Russia. We conduct research on the cryopreservation and long-term storage of the genetic materials of fish species. The aim of our work is the development of technology of cryopreservation and storage of reproductive cells of fish, as well as the development of techniques of using frozen material in aquaculture. One of the most important issues of low temperature preservation are the speed of freezing and composition of cryoprotective media. The laboratory staff conducted research on optimizing of freezing and the selection of compounds of cryoprotective media of different fish species, such as Russian sturgeon, beluga, steelhead sturgeon, sterlet carp, silver carp, and canoe. In connection with the different of sperm as a particular type, the necessary species-specific selection of composition of cryoprotective media, which can prevent destruction and loss of germ cells by low temperature preservation. Studies have formed the basic for the development of the methods of cryopreservation of sperm of sturgeon, providing electrical stimulation of germ cells that increases the rate of penetration of the treat in the cells of sturgeon. Also, the efficiency of reducing the volume of toxic substance in the composition of cryoprotective media for pentazole of sturgeon, which in turn reduce the toxic effects of the latter on the objects and has led to increased lifetime toward cells. Together with the staff on the Institute of Cell Biophysics, the method has been developed to reduce the low temperature jump during the crystallization of cryoprotective solution, which allows to increase the integrity of toward cells after cryopreservation. It consists in the first that it's a method involving freezing and suspension with biological material in liquid nitrogen to freeze and solutions of cryoprotectors of the cells of living organisms. Remote effect in performed on the freezing of the solution by ultrasonic waves. As a result of scientific research, the dependence of the volume of frozen material of the survival after thawing has been shown. The possibility of freezing similar fluid on grids in the form of thin films has been proposed. The effectiveness of reducing the volume of toxic substance in the cryoprotective medium for sperm of sturgeon species was also established, which in turn reduced the toxic effects of the latter on the objects and led on to an increase in the lifetime of deported cells. In the order to increase the level of heterogeneity of the resulting offspring and eliminate the negative consequence of inbreeding, a scheme for the formation of a broodstock of sturgeon fish in industrial condition using cryopreserved reproductive cells of males preserved in a cryobank is proposed. During the formation and operation of the breeding stocks of fish, the annual use of 10% of males' reproductive cells stored in liquid nitrogen for a long time from the cryobank is recommended, which make it possible to use high quality sperm at any time, eliminating the risk of ultimately maturation of fish and using more females for reproductive purpose. Work has been carried out to obtain offspring and determine the physiological usefulness of juveniles obtained using cryopreserved sperm. The resulting offspring turned out to be able and in turn of the reactivity of central nervous system and the receptor complex, particularly did not differ from the juveniles obtained with traditional technology. Fish obtained using frozen towered sperm had better survival rate, growing rate, fertility of physiological and biochemical parameters compared with those obtained by traditional methods due to the selective effect of low temperature of liquid nitrogen. For several years, Astrakhan State Technical University, together the Southern Scientific Center of Russian Academy of Science, has been worked on the creation of a cryobank, the activity of which is radically different from the exciting experimental collection. Its activity is based on the accumulation, preservation, and use of genetic materials to fill the shortage of produced and correct existing technologies of artificial reproduction of rare and endangered species of the south of Russia. The collection of the material is carried out of fish breeding enterprise of the south of Russia. The preserved genetic materials can be used to fill the shortage of production and correcting the exciting technologies of artificial reproduction of sturgeon fish. Thank you for your attention.
Thank you. Thank you, Professor Alexandria Karsilnikova. Thank you very much for your kind and very short presentations. Thank you very much. And you have completed before the time, and we will save the time and utilize this time for the next presenter. I invite Dr. S.R. Marigowda, scientist, Ministry of Earth Science in India. Professor Dr. Marigowda will be speaking on the ambient water quality standards to regulate the contaminations in the coastal water. Dr. Marigowda, please.
I'll sit with you. So I have to cut like this, paste. I can type paste morning. I went
greetings, sir. Is it viewable, sir?
Yes, your slide is clear and you are clear and audible. Please proceed.
Okay, sir. Thank you. Greetings to the all the panelists, audience, and all the organizers, and I thank for this opportunity. I am here to make a presentation on ambient water quality standards to regulate contaminants in coastal waters. Basically, because the marine pollution is one of the grave problems which needs to be addressed by means of regulatory measures. The regulatory intervention is necessary to conserve the water quality as well as the life in it. So we have an program which is focusing on developing of standards for the nation to deal with the pollution for priority contaminants. We'll do certain kinds of laboratory experiments on various food chain organisms, and as well as we also undertake field studies wherein the experiments are conducted in real environmental scenarios. So one need for a healthy water body, integrity and ecology of water body is very, very important, wherein we have chemical and physical and biological integrity is very, very essential for the ecological functions to carry out and the habitats to be protected. But what happens is in the changing conditions, each of these integral parameters are getting affected. That leads to an unhealthy water body that needs to be protected through the regulatory measures. And this is a background where the coastal pollution happens mainly because of the coastal waters are the ultimate sink for most of the land-based activities, which carry lots of various toxic chemicals, metals, and various other other nutrients which can be added exogenously into this environment, that environment gets contaminated, and that leads to various adverse conditions like depletion of oxygen or lowering of oxygen. That leads to the mass mortality of fishes, and sometimes it may lead to the algal bloom. Sometimes it may lead to various cascading effects in the food chain organisms. So basically, when we look at the sources, these are two sources from the point of regulation and control. So these can be categorized into a point sources and non-point sources. Point sources can be easily regulated by means of controlling the particular parameters as well as the discharge. So in case of non-point sources, it will be very difficult to manage these things. Therefore, these diffused sources needs to be addressed critically in terms of ambient water quality. So ambient water quality, basically they are very unique in terms of physical, chemical, and biological properties, where the industrial discharges are not directly released, which are treated before the release. But any change in these things can lead to an adverse conditions, as I already mentioned, regarding the algal blooms or the foam formation or any other such adverse conditions that leads to an and degradation of this water quality. Ultimately, the change in this water quality is mainly because of the added contaminants that changes the water chemistry, and that will lead to many kinds of impact. Basically, the Clean Water Act of the USA, which was enforced in most of the, which has been adopted in most of the countries, and that has been taken as a benchmark, wherein these parameters, how the water quality standards has to be specified in terms of its specific goals, and how the water body is used, and the stakeholder involvement in defining the standards of these designated uses, and what is the basic purpose of these uses of these waters, that will be taken into account. And basically these elements include the designated uses. The designated uses is the prime use, where we can define whether it is meant for what being traditionally used in that particular water body. That has to be taken into account to define the designated uses. And then we have to define the water quality criteria in terms of parameters. That parameter has to be, particular parameter has to be defined to meet the objective. Then there should be a policy and general policies and policies. Then the law and legal course will take into an action where the policies are involved. Ultimately, we need to protect the water quality. At the same time, once we can ensure the water quality, that can help us in protecting and maintaining good water quality, as well as to support the marine and aquatic life. So mainly the purposes to designate, main uses of this purposes to designate the uses of this water quality is protection of marine life. That is the basic need to maintain the ecological integrity and to protect the marine habitats. Then the recreation, bathing, sports, these are the some of the uses which are primarily used. Apart from this, there are many other uses which can come into different domains, and these all uses has to be categorized and grouped in some classes. Then we have to set the goal. While setting the goals, we have to keep the protection of these organisms and the human health at the most priority, and then the protection of these aquatic resources. So then once these goals are set, we have to monitor from time to time, so that if any kinds of adversaries are happening, then we have to intervene and control these kinds of things from further degradation of this environment. So when we look at the global scenario, use of these safe limits, there are various pollutants which have been prioritized in terms of priority pollutants, non-priority pollutants, and various other organic phosphate and pesticides are also have been determined in various different countries. So ultimately, the objective of these priority pollutants is to limit their concentrations in the ambient environment, so that the life in it can be protected. When we look at the principal method which is involved in developing in this kind of safe limits, is the key point is the toxicity is the basic and functional unit of water quality that has to be developed that has to be conducted, and then we have such kind of data can be used. And these kind of things have been used in US EPA, ANZAC, and European Commissions. So therefore, in Indian context, we do not have much data which has been which can be collected from the secondary sources where there are differences in its size, toxicity, and the chemicals. So therefore, we have to prioritize the chemicals which has to be targeted and for which the standards are essential. So already existing, what can be changed and what amendments can be improved, how best we can protect this environment. So to do that one, basically we are selecting we have adopted the US EPA methodology, wherein the planktons and all food chain organisms, planktons, crustaceans, shrimps, and bivalves and fish fingerlings are used. These are which can represent the representative species of this coastal and marine environment, and that that the experiments, laboratory experiments are conducted like acute and chronic toxicity experiments, acute which are very short term in nature, and chronic experiments are generally longer term experiments, wherein the endpoints like LC50 or IC50, inhibition concentration in case of phytoplanktons, and lethal concentrations in terms of organisms wherever the fishes and other organisms are involved. So these kind of endpoints are derived through the experimentation, and during the chronic experiments, we need to define the no observed effect concentration and lowest observed effect concentration, where there is effect is very lower and where there is a effect is very minimal. So such concentrations are derived to derive a chronic value, and the acute and chronic value together, that gives an acute chronic ratio. Then the ranking will be done to derive two different types of criteria. One is criteria maximum concentration, another one is criteria continuous concentration. Criteria maximum concentration is basically based on the acute and short-term experiments, and the concentrations are little higher than the chronic concentration. Therefore, these kind of standard will be useful for accidental spillage and other accidental events. But CCC or criteria continuous concentrations can be implemented as a safe standard for longer period of time. So these are some of the activities which are involved in deriving these standards. This involves a field activity, then the culturing of these organisms, and then the experimentation. So experimentation, we have the customized system wherein we maintain a chemical reservoir where the desired dose level of concentrations are added, and then these are pumped through the peristaltic pumps in a defined way. Then it will get mixed, and then which is supplied to the organisms. This is a continuous flow through system wherein these organisms are subjected for testing to derive acute or chronic experiments. Once the experiments are done, we have to measure the chemicals, what is the level of its concentrations once we add into that, and that has to be precisely measured by using the standard method. That has to be done to ensure that the quality and the values are reproducible. And here I'm just showing an example where compound like pH, like anthracene, one of the priority chemical under the pH, that how it is going to impact on the phytoplanktons and the copepods. So basically they interfere in their biological functions like chlorophyll growth, as well as reproduction, spawning, breeding. So long-term exposure of these things will lead to certain kinds of derangements. This involves some kind of molecular interventions, interferences, and that leads to an oxidative stress. Oxidative stress is very, very bad, which can also damage the DNA. So therefore, these kind of effects are also seen in case of lower organisms also. So in addition to that, histopathology is one of the traditional method. It is one of the oldest classical method to diagnose what has happened to the cells and to the individual organisms and tissues. So it is one of the diagnostics tool that the data from this histopathology can give us a clear-cut indication that what is happening physiologically to the organism, and that will be taken into account. This kind of data also will help us how the standard which we have derived, how far it is safer. That can be tested by using this kind of histopathological techniques. So in addition to that, we also complement with the biomarker enzymes. Biomarker enzymes basically, once any kind of exogenous chemical, when it is introduced into the media, it will enter into the cellular cell lines as crossing the bilipid membrane. So once it enters into the cell, it will have a cascading impact on the cellular organelles. That cellular organelles will have a molecular mechanism, thereby it affects the molecular mechanism and the physiology of the organisms. So therefore, this kind of data also, say for example, like a key antioxidant enzymes, which can give us a non-specific biomarker where it can also support in deriving the safe limits. So in addition to that, how far our derived standards are relevant and how far these are environmentally relevant in real environmental conditions, that needs to be tested for their applicability, whether they are holds good or not. So for this, we are conducting a small scale mesocosm experiments in open sea waters. And this also will help us in deriving PNEC, predicted no observed effect concentration, CCC, and CMCs. So these concentrations will be tested here and then it will be confirmed. So in addition to that, we may have to monitor the marine outfalls, how far these implemented standards are complying or non-complying, and what actions has to be taken to ensure that marine outfall monitoring is also necessary from time to time, so that we can ensure that these safe limits which we have derived and implemented, that can control the degradation of the water quality. So in addition to that, there is another approach called as SSD, species sensitivity density approach, wherein the same data which we have derived will be used. In addition to the secondary data, there are databases from the US EPA and also from the Australian databases are there. These kind of databases are used along with the endemic and native species to derive these kinds of safe limits. Here, I am showing an example where we have derived by using the SSD of Berlioz, Australian Berlioz software, as well as US EPA softwares. So what we found is the Berlioz software, which softens or smoothens the data, is more appropriate in this case. So ultimately, what we can achieve is once these kind of standards are derived, ultimately that has to get into the policy. So what we have, this kind of exercise shows an example how the laboratory experiments and field experiments and these scientific inputs can get into a policy making, and this has been taken into consideration, and the amendment has been made to the existing standards. So with this, I thank all. And this is our team which is working on these aspects at NCCR, and it is headed by a microbiologist, Dr. KV Sharma. And we have the team of different kinds of specializations like marine biology, coastal aquaculture, marine biotechnology, coastal ecology, and engineering, as well as a chemist. So with this, I thank all the organizers and the panel members and all the audience also. Thank you, sir.
Thank you, Dr. Marigowda, for the very elaborate and talk on the seawater quality and the policies and standards. Thank you very much for your nice talk in a short and sweet time. Thank you very much, Dr. Marigowda. I request Professor Yulia Fedorovsky from Astrakhan State Technical University. Dr. Yulia Fedorovsky will be speaking on alternative source of protein for local raw materials for fish feed. Dr. Yulia, the stage is yours, please.
Can you hear me now?
Yeah, you are here and loud.
Okay, it's nice to greet you. I represent the Astrakhan State Technical University, the Department of Aquaculture and Fishery. And today I want to tell you about some of our investigations in alternative sources of protein from local raw materials of Astrakhan region in fish feeds. Because our department, we kind of famous in the create of different recipes for aquaculture objects. I want to briefly talking about the situation in with the aquaculture feeds in Russia. The volume of global production in industrial compound feeds for aquaculture is steadily growing, and in 2016 reached 45 million tons, according to the FAO forecast. By 2030, the demand of them will increase to 62 million tons. The largest producer of fish feeds is, of course, China. 26 million tons were produced in 2015. Other major producers are Europe, Thailand, Vietnam, Norway, Japan, the USA, and Chile. In Russia, the situation with feed for agriculture facilities is fundamentally different from the state of feed production for the needs of animal husbandry and poultry farming. In 2016, 27 million tons of compound feeds were produced, as a result of which Russia took 7th place in the world ranking of feed producers. In accordance with the focus of the development of these branches of agriculture, it's predicted that in 2020, about 30 million tons of compound feeds will be produced, the basis of which will be feed for chickens and pigs. Conversely, the poor state of fish feed production in Russia, poor quality and limited assortment, and the lack of modern feed biotechnologies hinder the development of aquaculture. After 1991, the volume of domestic fish feeds production decreased fourfold to about 70,000 tons with a demand of 260,000 tons. As a result of the shortage of high-quality domestic fish feeds and the limited assortment, imported feeds predominate in the domestic market, occupying about 2/3 of consumption volumes. The share of imported feeds for valuable aquaculture objects such as Atlantic salmon, rainbow trout is even greater. The main part of fish feeds produced in Russia has a poor quality. They are characterized by the low nutritional value and the balance, insufficient water resistance, and the excessive crumbling of granules. This is a consequence of the use of outdated technologies in production, non-complements with the recipes, and poor quality of feed components, as well as low production culture.
Your slides are not visible. You're not moving your slides.
Yes, I know, because I have this slide, and I want to show you this one. Then I will change the slides. Don't worry. Now, in the diet of fish, there are more than 100 types of feed and feed additives made from waste from food and oil extraction industries, including products of microbiological synthesis, various salts, vitamins, enzymes, amino acids, antibiotics, sorbents, antioxidants, flavorants, etc. In industry, new nutritions cheaper than fish meal, feed components of vegetable and animal origin containing easily digestible protein, valuable carbohydrates and other substances, waste of secondary raw materials of processing industries are emerging. The study of the nutritional value of new high protein feed raw materials makes it possible to solve the problem of replacing the fish meal by developing the new effective starter and production compound feeds for valuable fish species, organize the cultivation of juveniles and commercial fish on an intensive basis, and abandon the use of imported products. And now, the next slide. As I told you, the traditional trends in feed production in the field of aquaculture in recent decades indicate a reduction in the use of fish meal with increase in the content of plant and new alternative protein components. In the future, we can expect a reduction in the use of fish meal in feed by up to 10%, with the increase in vegetable protein to 69% and new components to 10%. Among the most promising areas of technology in the production of feed for aquaculture, experts call the use of the following alternative source of protein:
like no traditional marine sources, for example, like seaweeds, the plants, krill, and etc. Non-marine, non-traditional sources like insects, growing fish for its subsequent use in feed, and innovative technologies for the collection and use of fish processing waste. I want to describe some experiments which were carried out in our Astrakhan State Technical University in some farms and using some industrial conditions like growing fish in tanks. We choose 3 replacement for fish meal. First of all, it was them, which is some kind of sapropel. I explain it further. In fresh salmon, in small rivers, ponds, and specific types of salty sediments with an organic content of more than 15% is formed and is called sapropel. Translated from the Greek, sapropel means rotting dirt. This substances is a biological origin formed under fresh water during bacterial process with a little oxygen access at the bottom of the freshwater reservoirs as a result of accumulation along those mineral substances of the remains of aquatic plants and animal remains. This active biological substrate with a high content of organic substances like amino acids, vitamins, enzymes, hormones, and trace elements in an easily digestible form allows to increase the usefulness of feeding and has a positive effect on the body of organism. This is due to the interaction of 3 components at once: biologically active, mineral, and organic. The organic part of sapropel contains readily available carbohydrates, mainly the protein origin. Sorry.
Please proceed.
Dr. Mariguda, kindly mute yourself. Dr. Mariguda.
Sorry, should I continue?
Please.
The organic part of sapropel contains readily available carbohydrates. Mainly the protein origin of sapropel nitrogen causes the presence of essential amino acids like lysine, methionine, cysteine, tryptophan, etc. Of other bioelements, they include chlorophyll, carotene, vitamins, sterols, hormonal compounds. A number of biologically active substances are produced by the microflora of sapropels. Humic acids are the most important biologically active components of sapropels. Biologically active substances have the antimicrobial action, suppress pathogenic microbia in the body, microflora in the body, activate metabolic process. Feed sapropel in dry form can be used in the production of feed as an organomineral additive and as a filler in the producing of vitamin premixes. At the same time, the feed coefficient of the feed used is significantly reduced, and the diet is enriched with biologically active substances, minerals, and amino acids. The next slide. Also, I want to add that the feed sapropel affects as all physiological process in the body. In particular, it prevents disease of the skeletal skeleton in farm animals, improves the reproductive functions of animals too. The second component which we decided to investigate was amaranth. Among the raw materials of plant origin, the experts of the FAO recognized amaranth as the most important crop of the century for its high nutritional value and productivity. Zelenkov showed that the chemical composition of the leaf part of Amaranthus cruentus is characterized by a high content of soil residue, which in turn indicates rich mineral compositions. You see on the slide. A distinctive feature of amaranth from many forage crops is its biochemical composition, namely the presence of all amino acids necessary for the normal functioning of the body. The total content of minerals in amaranth is higher than in the grain of other cereals. The content of calcium is 7 times higher. Magnesium is 2 times higher than that of wheat and rice. The use of amaranth as a feed additive provides the enrichment of the diet with biologically active substances, vitamins, microelements, which allows not only to increase the average daily growth rate in weight, but also to improve the digestibility of protein, fat, and fiber, the use of nitrogen, calcium, and phosphorus, thereby increasing the yield and improving the quality of agricultural products. The third one, also we in Astrakhan region, we have many problems with such insects as migratory locusts. And that's why we can have them as some kind of components for our feeds. Larvae and imagos of migratory locusts are high quality balanced food for many insectivorous animals. When using invertebrates, including locusts, as a feed additive, the question arises about the effectiveness of this raw material in feed production. According to numerous studies of the total chemical composition of this raw material, the amount of dry matter is 33%, crude protein is 23%, and the ash is 1.4%. That's why we decided to create in our Aqua Biotechnopark. It's some kind of place with a lot of tanks. When we carried out our experiments with fish feeds, with fish breeding, and as the object of our investigations, we choose the carp fingerlings and also adults of tilapia hybrids. There was an object of our research. As new components, we choose the dry mass of amaranth, superpel, and migratory locusts. You can see on the slide the recipe with the nutritional properties of this recipe. Of course, we can show you the percent of each components in the these recipes, but we replace the fish meal by the amaranth in test 2. We replace the fish meal with sapropel in test 1, and we replace the fish meal with locust flour in test 3. As a control, we use the standard feeds for the carp fish and for tilapia. Then I'll show you. What we have as a result of our investigation. As a result, in the course of our research, it was found that the use of feed additives as sapropel, amaranth, and locust, it has a positive effect on the growth rates of carp juveniles, which was expressed in an increase of body weight compared to the control variant by 25, 25.5, and 34 grams, respectively. A decrease in the feed costs by 0.2 and 0.3 units, an increase in the mass accumulation coefficient by 0.4 units. Also, we studied the biochemical compositions. Wait. Oh, I'm sorry. Yes, this was the first experiment with carp. And we have a good results with each of the additives. This is the biochemical composition of the carp body, and it was found that the highest mass fraction of protein was in the group of fish that consumed locust. The second one was sapropel with 13.5%. The highest proportion of fat was in the control group, it was 19%, and the lowest in the individuals who consumed sapropel, the feeds with sapropel and locust. Also, we check the morphological picture of the carp liver of the control and experimental groups, and they did not differ significantly. The microscopic structure of liver hepatocytes indicates the full functioning of its main cellular structure. Compared with the control, it's possible to note a more intensive infiltration by lymphocytes of the aged epithelium of the intestinal microvilli of the fish of the experimental groups, which apparently is associated with their more intensive nutrition. When studying the addition of sapropel to tilapia feeds, it was found that the absolute increase in the experimental group of tilapia exceeded the control by 5 grams. The average daily growth rate increased, and it was also 20% higher in experimental individuals. The weight gain for the amount of feed consumed was also higher in fish that consumed feed with the addition of sapropel. The feed ratio was lower by 0.1 units. The survival rate in the control was lower by 13%. When consuming experimental compound feed, fish had a slight increase in also some hematological figures, which is ESRR within the norm of 2 to 10 millimeters per hour, which indicates a more intensive diet as well as adaptation of fish to a new compound feeds. The differences in hemoglobin concentration between the experimental and control fish were not complete, and we were at the level of 88-90 grams per liter. Lipid metabolism proceeded intensively in both groups, as evidenced by a stable index of total serum lipids of 3.4 and 3.46 grams per liter in both groups. Cholesterol content also did not differ significantly in the experimental and control groups. And as a result, we can recommend some amount of replacement of the fish meal in the production feeds for tilapia and carp, which is the most using aquaculture object in our region. And this recipe can be used as in the fish breeding also. We carried out a lot of experiments with the different recipes, with different additives, and I want just to show you some of them. Thank you for your attention. Thank you. This is all.
Thank you very much, Professor and Dr. Olivia Broski. Indeed, your talk is a very elaborate and detailed talk, and it is very much essential for the present status of fish feed requirement. So the world is increasing aquaculture production. So this is very, very important topic. I think we can have elaborate discussions at the end of the meeting. Thank you very much for making the presentation in time. And I would take this stage for my presentation.
May I request organizers to show the profile slide of Dr. Dharani again? I think the IT team is not prepared.
No issue. I will introduce myself, sir.
Yeah, please.
One moment, sir. Is my slide is visible and my audio is clear and loud?
Yes, absolutely.
Yes, thank you very much, sir. I take this opportunity to thank Ministry of Earth Science and the Embassy of Russia to make this event so wonderful and giving me an opportunity to speak in this forum. And I will be representing Ministry of Earth Science. I'm from National Institute of Dharani, from National Institute of Ocean Technology. We are working on marine biotechnological aspects. And today we are going to discuss our work, some of our works on the deep sea microbes. The title of the topic would be the harnessing the potential of deep sea microbes for the Indian Ocean for biotechnological applications. I don't need to re-emphasize the points again, but still I would like to say the volume of the Earth, because the Earth is already covered. Someone, previous speaker said it is not a planet, it's a water air or ocean air. So it is 71% of the, covers the 71% of the Earth's surface, and the depths are up to 12,000 meters, and still life is found and occupy 99% of the world living space by volume, and mostly still remain unexplored when we compare to the space that we have explored. The life, it is believed that life begins in the sea according to the evolutionary theory, and 80% of the life found in the ocean. And more than hundreds, thousands of components we identify in the sea. This is a presentation to show the volume of the deep sea by the area and space. It is roughly 62% of the deep sea by the area. It occupied by area, and about 78% of the deep ocean occupied by the volume. This shows the area that is occupied by this. The formal expeditions were started in 1872 to 1876. It's the first formal and scientific expedition for this Challenger Deep, which has brought a lot of findings on the sea, and there are animals and there are living organisms inside in the depth of the sea. The Challenger Deep has recorded at least 8,184 meters in the South Pacific between Guam and Parland, and in the Mariana Trench. Of course, they had made the first sound in the Mariana Trench and identified, yes, there are organisms living below the surface of the water, which was interesting to the science at that point of time. And since then, so we are continuing exploring the deep oceans, and we are into the marine microbes. And why the marine microbes ecology? In land or water, we can say the microbes rules the world. And one of the deep sea is one of the earlier biosphere known to the man, and the niches remain to be explored further. Marine microbes, because whatever microbes or living systems, microbes especially lives in the deep sea environment, which has a very high hydrostatic pressure, low temperature, high salinity, and absence of sunlight, and they do not follow any circadian rhythm or any light-related rhythm, and they do their own cycles because the deep seas are entirely has only one phase, it is darkness. The study in the deep sea would throw a lot of light on the understanding on the benthic biochemicals and the biogeochemicals in the machineries. It involves in several crucial biological processes, and that may be a warehouse of multitude of biological active compounds. The deeper part of the water body surrounding Indian Ocean, particularly the extension of Indian Ocean like Bay of Bengal and Arabian Seas, are not thoroughly studied for the microbial studies, except for few institute studies like I never go over. And why this marine microbial ecology is not very new, and it has been a lot of studies, systematic studies has been started since the Census of Marine Life, COML, an integrated ocean drilling program headed by Japan JAMSTEC, the TARA expeditions, the deep sea drilling projects, and international Census of Marine Microbes, and the ocean exploration research, are some of the iconic programs which are aimed to monitor the microbial diversity of the deep oceans and oceans in the deep seas. And our institute, although before we start into the deep oceans, so initially we started a program called Drugs from the Sea program, which is a consolidation of some 12 institutions of Government of India, different institutions from Ministry of Earth Science, CSIR, and other institute participated, where they targeted on, which was guided by the premier institute called Indian Institute of Indian Institute of Drug Technology, who are the responsible party to qualify drugs and to produce new novel compounds. And in that project, we are a part of that project, and we did sampling in Andaman Seas. We collected more than 250 specimens of various species from Andaman Sea, from the shallow water, from the shelf region to about 40-meter depth, purely by scuba diving. We have a very strong team of divers, including myself. And the biological assays resulted in 26 antibacterial and 9 immunomodulatory and 3 antifungal tuberculosis product. And this product are with Indian Institute of Drug Research, and they are carrying out further characterizations and synthesis this part of the compounds. So we are working on the deep sea microbes. Of course, microbes, a lot of speakers spoke about these things. The microbial process involves right from the isolations of water and morphological characterizations, biochemical characterizations, then molecular characterizations. And of course, we do the metagenome, which is the largest part of the microbes are non-cultivable. So we essentially target into two phases. One is in culturable microbes, which we go for, which is supposed to be less than 1% of the total organisms available, less 99% is uncultivable. So the 1% cultivable is also a greater task. As earlier speakers spoke, there are a lot of constraints in development of media, development of proper support systems, and the co-culture systems. So that we do, and we also do the cultivable, uncultivable bacteria by metagenomic approach by environmental DNA. So our National Institute of Ocean Technology do have 4 research vessels, 2 ocean going and 2 coastal research vessels. And this is this is a picture of an IoT research vessel, ORV Sagar Nidhi. It is in technology demonstrations vessels, and it is in polar capability, and we participated in a number of cruises on this vessel, in variety of our vessels, and we continue to collect samples from the deep sea. So these are some of the sampling points which we occupied very recently in Northern Indian Ocean as well as in Indian Ocean. We study the diversity of the deep sea bacteria from the shelf to the deepest part of the oceans. And generally we use very known equipments like a piston corer or a vertical gravity corer and water samples. So one of the biggest tasks in collection and identification of microbial studies is collection of water sample aseptic water samples, because microbes everywhere microbes is there, and you have made a very particular point that yes, we are collecting the microbes from the deepest part, and as we discussed exactly, the water, as the depth increases, the hydrostatic pressure increases and the bacterial community changes, and there are a group of bacteria called physophiles and physotolerant. The physophiles are one which loves the pressure and which will not tolerate any changes in the pressure, and the physotolerant will adopt to the pressure. But all this organism has to be studied together. So we have designed with the Institute of NIOZ, within collaborations with support of NIOZ, we had developed a high pressure water sampler, which has a rating of 6,000 meter bar capacity, and it can operate at up to 6,000 meter depth, 600 bar pressure rated sampler, which can operate at up to 6,000 meter. And this sample can be sterilized and completely autoclaved on the surface, and it can be lowered to the depth and triggered from the ship based on the CTD signals, which will collect samples from the deepest part of the ocean without contaminations, and also it will retain the pressure until we retrieve back to the system. So this is of some of the sampling systems, which has 200 ml jacketed titanium vessels, which we use for collection of water. And this is a pressure difference of the collection of the sample. And this is a system which has all the things. That is, one is the collection systems. This is the collection system. The collection system is a high pressure sampler, and the high pressure sampler comes from the ocean, and again it has been stored in the refrigerated conditions, transported to laboratory to shortest time, and the system goes to a high pressure serial dilution systems, and the high pressure serial dilution system, it goes to a high pressure fermenter, which is again a 350 bar rated, and we can mimic the conditions of the ocean up to 4,000 meter depths. So out of this, in recent past, in the past 3-4 years, we have collected a total of 348 number of barotolerant deep sea microbes, which was isolated from 1,000 to 4,000 meter depth. The total bacteria is approximately 180 isolates, and fungi is 45 isolates, and Actinomyces 123 isolates. And all those isolates have been tested to grow in a high pressure fermenter, as well as has been exposed to atmospheric conditions and studied the pressure tolerance as well as the pressure loving nature. And we have screened this organism production of various compounds, and out of this we have, to name few, we have multiple proteoglycin and proteoglycin molecules from a Streptomyces species, and a Nigrospora, aggressive fulvin and spirobenzofuran compounds from Nigrospora, and pyridine peptidazole compounds from Streptomyces, and sequestering derivatives from Astrotricha, and L-asparagines from Neocardopsis, and lipopeptidic surfactant from Bacillus, cyclic peptidine from fenvensis, antimicrobial peptides from Bacillus, and ectoine enzymes from Bacillus. So what we do this, if we are collecting the pressurized sample, also we study the pressure tolerance of the bacteria. And these are some of the studies showing when the pressure changes from 1 atmosphere to 100 bar. There are a lot of scientific interest in as the pressure changes. Even though the bacteria are some bacteria are barotolerant, but still the morphology, the biochemical productivity changes entirely during the pressure operations. You can see very well the bacteria is normally of 2 micron in diameter, 1 micron in diameter, and 2 micron in length of outside bacteria. If you are subjected to pressure, the divisions they lose the sense of division length, and the divisions exceeded more than 5 micron in length, and they produce terminal spores, and they grow on further. And we have a lot of interesting studies in this. If you study the microbial enzymes, particularly fatty acids, there is a very significant change when you change the pressure from 1 bar pressure to 50 bar pressure. There's a set of fatty acid biosynthesis changes, and it is interesting to note this. The biochemical changes, the pathway changes, and there are a number of opportunities to study these things and make use of this difference in pressure for production of the required compounds. And this is a FTI spectrum, which shows the difference between the 1 atmosphere and the 50 bar pressure rated. Though the signals are very, very clear, showing there's a very different in the production. So these are some of the achievements. We optimized production of extracellular novel glutamase asparaginase from actinobacteria. We have also produced a surfactant and surfactant from the deep sea bacteria, and also an ectoine biosynthetic gene has been produced, and developed a multiplex PCR kit and a novel consortium of microbes, which is very, very specific to eat hydrocarbon, which they consume. Hydrocarbonized and sole carbon has been developed. And to elaborate the system and recombinant L-asparaginase, this is one of the well-known cancer drugs which is in market, but this L-asparaginase is devoid of glycine, which is having an earlier all other old-known compounds has glycine, which is supposed to be toxin. So what we did this L-asparaginase, we have from deep sea actinobacteria Nocarditis alba has been identified, and the gene, the entire complete set of genome has been cloned out in an expression system, and a recombinant DNA, through recombinant DNA technology, the entire clone was expressed in an expression systems, and this is ready to the transfer, and this can produce an L-asparaginase without interference of glycine and have high activity, biological activity. Similarly, biosurfactant producing genes from a deep sea sponge, isolated sponge associated bacteria also been identified, and those and again the biotechnology through biotechnology tool, this complete gene has been transferred to expression systems, and the production has been standardized, and this biosurfactant has been the biosurfactant production system has been established for production of biosurfactant for industrial applications. And a biosynthesis, similarly, ectoine producing gene, and ectoine, you must be knowing, ectoine is a commercial dermocosmeceutics, which is used for formulations of UV screen and so many other cosmetic products, which is coming from the land-based bacteria. The first time we had identified this similar ectoine expression systems in a deep sea bacteria, and we could able to successfully extract the gene and again clone it in an expression system through recombinant DNA technology, and this technology has been patented and been transferred. And similarly, other one, glycine-beaten biosynthetic genes from deep bacteria from spermadurans is also in the way, and the present status it is in an optimization of large-scale expression systems and expression systems in work. And support to the system, we also study the antibiotic properties of cultivar actinobacteria from the deep sea, and we have this is one of the first findings from the actinobacteria from the Barren Island, which is one of the active volcanic islands in India, and we had a group of cyanoformide and lopoformide and nucleoorganose synthetic genes coming from this bacteria. And also we study the deep sea sediment through metagenome. This is metagenomic approaches collected from different regions from northern Indian Ocean like Bay of Bengal as well as Arabian Sea. And we have performed an Illumina long read sequencing, and when we have a huge bacterial database, and we could able to make at least identify at least 25 phylum from 50 class and representing 552 genus, which is a very high bacterial diversity. And the major genus or Microspora and Streptomyces and Salmospora are important species identified from the gene sets of metazoa. So from this analysis, we could able to identify at least 79 operational taxonomic unit from 3 different stations, and the maximum operational taxonomic unit is identified from NAOT-C, NAOT-1, which is from 3,000 meter depth. So what is the problem here? So we are continuously looking for products and supply from marine natural products. One of the biggest problems is identifying marine natural product as such is industrial supply, the total synthesis. Though marine organisms and bacteria hold huge reserve for biomolecules, but the extractions, the scaling up and the extraction of the biomolecule from this resources are limited, because the collection of the resources is difficult from the deep sea, and cultivation of these resources are very high, and productions will be very less. So there is always a difficulty to produce this from the natural resources. And of course, synthetic chemistry has been one of the route to do that, but for to produce to have a retrosynthesis to produce 4 subunit of amino acids, it requires 50 steps in total and a non-linear sequence. And overall yield is only 0.7%, which is very much not cost effective based on this findings. And there are reports says that this ectasin from marine ascidian, which was earlier produced from the marine ascidians, but later on it was identified that the bacteria which is having which is living in this ectasin acidin is producing a key compound. So they are able to first do the fermentation process, and later on the fermentation products also was abandoned and identify the purity genes inside the bacteria, and those bacterial genes has been cloned. And now those cloning systems, this ectasin is coming from the clone library of the bacteria. And this are some of the natural products already reported from tunicates for various bryozoans, sponges, and actinobacteria. And all these sources are now has been replaced from the genetic resources. That means the whole biosynthetic genes from this bacteria or this organism has been identified, and the genes has been transferred vertically through recombinant technology, and this expression system is now is being used for production of commercial scale. So the point which we want to emphasize is, so the the living organisms will give a clue, and it is possible to synthetic to to synthetic pathway, synthetic pathway. In the terms we cannot do that, there is always an opportunity to produce the same kind of enzyme, same kind of compounds by expressions of the genes in several vector systems by employing novel today's recombinant DNA technology. So the new concept of drugs from the marine natural product in the post-genomic era, as described by Fernando D. Carl from a division of Pharma Marine Spain, which is one of the very big company doing in mining of the genomes and mining of the product, and they have a huge number of product, at least 4 to 5 number of product in cancer treatment in market, in identifying from the base, from the raw material to marketing, they have done successfully. And what they say is the marine biodiversity, the whole metagenome, not even the organisms, the whole metagenome and other microbial consortia, the genome mining is been happening, the target genes are identified and identification secondary metabolites and the gene clusters of enzymes. And these new genes are either the silent or they could be cultivated, they could be expressed, can be done to design very novel drug discovery strategies using the marine biotechnological approach. So this is a combination of classical marine microbiology to and molecular biology and genetics, which go on hands in hands, and thanks to the novel DNA technologies nowadays and the long-range sequencing pyrosequencing, which enable us to duplicate the complete whole genome and identify the complete set of genome for production of novel compounds. And parallelly, we are parallelly to identify the metagenome and do the functional-based metagenome and the gene-based analysis to identify beta compounds. And this thing is a very promising studies. And I also felt the number of authors, as number of top speakers has already spoken in this slide from India as well as Russia. We will be very much happy to interact with them and go ahead further to do this research. Thank you very much. And with this thing, may I take the opportunity to invite Professor Olesya Ikubova. I know he has logged in. Olesya Ikubova is a PhD, Senior Lecturer in Department of Technology of the Food and Commodity Research, Astrakhan State Technical University. And today's talk is going to be on scientific foundations and implementation of collagen production from the fish of Volva Caspian Basin. I request Professor Olesya, now stage is yours.
Good evening. Can you hear me?
Yeah, you are clear and loud. If it's possible, you can switch off on your video for a minute.
I'm sorry, it's impossible because I have no web camera on this computer, so I'm sorry.
Thank you. I think you can load your presentation.
Верните на полный экран, пожалуйста. Внизу в правом углу экрана есть значок рядом со
Could you
шкалой.
please return it to the full screen? In the right corner, you have a special sign with the scale. Okay, thank you. Can you hear me? Yes. Yes, we can hear you.
Participants of the International Scientific Conference, and let me introduce myself. My name is Yakubova Olesya. I am the candidate of Technical Sciences, head of the Laboratory of Technology and Technologies of Biopolymers, associate professor of the Department of the Astrakhan State Technical University, head of the Enterprise for the Production of Food and Cosmetic Collagen from Secondary Fish Resources. So collagen is the main protein in the human body, which provides the structural framework, the matrix of the body, due to which the elasticity and strength of the musculoskeletal system, blood vessels, and integumentary tissues are formed.
With the lack of collagen, the aging process of the body are activated. So collagen is called the protein of youth. It increases the elasticity of the skin, thereby reducing wrinkles, promotes the growth of dormant hair follicles, accelerates the growth of healthy nails, restores cartilage tissue, restores freedom of movement. In general, collagen is a preventive agent against aging of the body. Currently, there are a lot of collagen of different origin from the skin of pigs, cattle, and the most valuable collagen from fish raw materials, which is called marine collagen. It's often asked why is marine collagen is the most valuable food. So firstly, collagen from fish is easier to digest due to the structural features of the polypeptide chain. Secondly, it's absolutely safe, eliminates the risk of infection with diseases that can be transmitted to humans. The third is the high content of low molecular weight protein, peptides, and amino acids from which its own collagen are synthesized. It's also important that the quality of collagen is ensured by the absence of discrediting tests and smells. And most importantly, all these characteristics of collagen are ensured by its authenticity. Only collagen with confirmed origin, animal origin without vegetable thickers, carbohydrates, dyes, sweeteners, flavors can be called natural collagen. We produce food and cosmetic collagen from secondary fish raw materials of the Volga-Caspian Basin and commercial aquaculture facilities. The bones, skins, hides, scales, and swim bladders of fish are used for production. The production technology will differ from the type and composition of raw materials. It includes three main stages: separation of high molecular collagen from related components, transfer of collagen to a bioavailable low molecular form, purification of collagen from impurities, and deodorization. High quality collagen doesn't have a discrediting bite and smell. So before the production of collagen was organized, research was conducted at the Astrakhan State Technical University for more than 20 years. And now scientists are continuing to study collagen products and product based on it in different directions: biology, technology, engineering, and medicine. This slide shows part of the scientific potential for introduction into production. Research is supported by federal programs and grants. In 2019, a grant was implemented on the topic technology development and application of biopolymers from secondary fish collagen containing raw materials. Currently, 2 products are being produced: food grade fish collagen with a protein content 10 to 15% in 2 types of packaging: a combined metallized dry pack package with a fitting and transparent polypropylene packaging, and food grade fish gelatin and sheet forming cardboard boxes. Submitted projects are presented at exhibitions and different forums. They were awarded medal at international and national competitions. So in conclusion, I would like to thank the organizers of the Russian-Indian Conference for the invitation to participate and provide the results of our activities. If the submitted projects are interesting, we are ready to cooperate. Contacts, you can see contacts on the slide, on this slide. Thank you for attention.
Thank you, Dr. Alicia Ekbove. Thank you for very short and sweet presentation. And I think it is of a very interest for fish collagen in terms of health benefit and for aquaculture purpose. I think we can complete the last speech by Dr. Suraj Das, and we can discuss in the panel discussion. Thank you very much for your kind time and your nice and short presentation. Thank you.
Thank you. Thank you.
Can you please unshare your slide and mute your mic, please? I take this opportunity on the behalf of the organizers to invite Dr. Suraj Das, Associate Professor, Life Science, National Institute of Technology, Rourkela, and his presentation is going to be on marine bacteria and biofilm and bioremediation environmental pollutants. I request Dr. Suraj Das to take the platform and start your presentation.
Hello, am I audible to all of you?
Yeah, you are audible, you are clear and loud. If it is possible, can you put your slide in a full slide mode, full screen, or presentation mode?
Is this fine?
Yes, yes, absolutely. Okay, thank you.
Well, thank you, and good evening to all of you, respected chairperson, eminent scientists, all the members of this symposium. Being the last talk here of this session and today's, I would not take much time. So my talk is on marine bacteria and biofilm in bioremediation of environmental pollutants. I am Dr. Suraj Das. I am working as associate professor in National Institute of Technology, Rourkela. So here all we know that marine environment ecosystem covers almost around 71% of Earth's surface and responsible for 32% net primary production of this globe. And marine ecosystem, it fundamentally covers vast area including estuarine, coastal, swamps, mangroves, rocky islands, seagrass, and the coral reef. And this environment plays several beneficial ecological roles and provides various economic benefits to mankind. And the ecological, and besides, the economic benefits include medicine, food, natural oil and gas, timber, and other plant products, and so on. So coastal and marine ecosystem are found in 123 countries and are considered among the most productive ecosystems in the world. Well, so here we see around 2.5 lakh marine species have been discovered so far, among which India's contribution is around 7%, which includes more than 18,000 species diversity of flora and fauna. And marine organisms possess some unique features like a tolerance to extreme environment and variation of the pressure, salinity, and various other dynamic conditions in the marine ecosystem. They also produce variety of chemical substances. We find that in the largest chemical ecology. And marine microbial diversity of the marine ecosystem are essential contributor of global biomass and biogeochemical cycle. They occupy all the available niche within the ecosystem, and surprisingly, only less than 0.1% of the ocean microbes have been described till date. So marine ecosystem harbor a huge diversity of bacterial population. So the bacterial community of the marine ecosystem predominantly consists of 5 groups: alpha proteobacteria, gamma proteobacteria, cytophageal bacteria, bacteria, and formicutes. Besides, there are various new genera being discovered over time. And there are different physiological groups of these marine bacteria, such as sulfur-reducing, methanogenic, halophilic, luminous bacteria, nitrifying bacteria. They play different crucial roles in the marine environment and contribute over 50% of the carbon turnover of coastal marine sediments. They also play a role in cycling of minerals and in the marine environment, and contribute in the nutrition of marine organisms at gut microflora. Well, the dynamic changes in the marine environment make the inhabitant bacteria suitably adapted to the adverse conditions, and marine bacteria have their huge diversity, biofilm formation ability, biosurfactant production, and they also have a diverse catabolic genes, and their cell wall helps them to adapt in the dynamic marine conditions. Marine bacteria acquire a strong potential for remediation and restoration of the contaminated environment. So biofilm formation is considered one of the most pronouncing characteristics of marine bacteria with regard to bioremediation. So bacteria within a biofilm possess several advantageous properties than the free-floating plankton cell, such as high cell density, increased availability to nutrients, tolerance to environmental stresses, transfer of the genetic information, intercellular communication, which is named quorum sensing, and production of these extracellular polymeric substances. Our emphasis in my lab is to explore the microbial biofilm, their associated EPS matrix, and the intracellular communication system prevailing in the biofilm environment for enhancement in the bioremediation process. So here, as mentioned, immobilized bacteria or biofilm forming bacteria are of more significant in addressing environmental remediation. Now we have two questions in our mind here. First, how do bacteria sense the surface? Secondly, what are the important developmental steps involved in building a biofilm community? Well, to answer this, we have the papers and outputs from the legendary scientists who are working since long on biofilm. Matthew Patrick described that the Pseudomonas aeruginosa, when it senses the surface and it produces the cyclic adenosine monophosphate, and they are called the surface net cell, and there are other bacteria, planktonic bacteria, which are called the surface continent cell. So here, these planktonic cells exhibit high CMP level, and this increase in increased high CMP level eventually contribute to the progression to the irreversible attachment stage, where cells remain surface adhered and ultimately develop the biofilm. In another leading researcher, a globally leading researcher in the biofilm, George O'Toole. So the paper what he has described is that the biofilm development requires a series of discrete and well-regulated steps. While the exact molecular mechanism may differ from organism to organism or maybe even the genera, but the stages of biofilm development appear to be conserved among wide range of microbes. So in our lab also, we have studied the process of biofilm formation by bacteria and observed that it takes place in a multi-step process that initiates with the attachment of the planktonic cell to the substratum, then cell proliferates to form the monolayer over the surface of substratum. Then the attachment of cell over substratum is supported by many physicochemical interaction and the extracellular protein. And next stage, in the subsequent stages, increase in the cell number results in the synthesis of elevated level of autoinducer, which induces the bacterial cell themselves and produce the extracellular polymeric substance. And with time, progression of biofilm to a mature biofilm with increased resistance to hostile environmental conditions and other factors that will lead to the breakdown of the EPS, and then for the adhered cells will become planktonic to find out suitable substratum to form the new biofilm. Well, so inside this mature biofilm, a number of metabolic processes occur. Well, and now next, what we would like to see is what is the biofilm composed of. It is not composed of alone of the bacterial cell, where we can find the matrix on which the cells are embedded, and this matrix is named as the extracellular polymeric substance. So here the EPS layer of the biofilm provides enhanced protection to the cells of the biofilm from adverse. Since EPS is a polyanionic nature, it readily sequesters the metal ions and solubilizes the hydrophobic substances. If you look at the composition of EPS now, it is a mixture of several macromolecules, and they are, for example, they are polysaccharide followed by protein, nucleic acid, lipid, and the humic substance. And generally, polysaccharides and proteins contribute to the adhesion and aggregation of the bacterial cell, and nucleic acid promote exchange of the genetic information. Lipid helps in the export of cell components, and humic acid contribute to the adhesion. Well, however, what makes the EPS so useful? So if we look at the chemical characterization of this EPS, we found there are various functional groups present. For example, the carboxylic group in polysaccharide, phosphate group in the nucleic acid, amide bonds of proteins, and methyl and ethyl group of lipids. And the interaction of heavy metal ion and polycyclic aromatic organic pollutant with the EPS occurs via these functional groups. So here, as we have understood that the bioremediation, which is considered as the triple corner process, depends on three pillars. One is pollutant, second is the environment, and the microorganisms. And various inorganic and organic pollutants, such as heavy metals, polycyclic aromatic hydrocarbons, released from different natural and anthropogenic sources, can be subjected to bioremediation. And the biofilm EPS or marine bacteria have been widely used as hydrocarbon solubilizer and metal sequestering agents. So the production of EPS by marine bacteria can influence the fate of oil and hydrocarbons in the ocean. to emulsification, degradation, dispersion, aggregation, and sedimentation. We are all aware, so Pseudomonas aeruginosa by Professor Chakraborty, who described how these oil-eating bacteria can be helpful in degradation of the oil spill. And what is done by these bacteria is through these EPS. And what this EPS does, EPS is the function of the EPS is to emulsify and disperse petroleum component and thus enhance their solubility and bioavailability. And it provides a physical structure allowing the microbial community to degrade oil and to provide colloidal and particular trap for petroleum component and subsequent sedimentation. So recently in our from our laboratory, study was carried out to explore the role of bacterial EPS as an emulsifying agent for petroleum hydrocarbon. And in the study, we have reported the emulsification of petrol by using the extracted EPS of biofilm forming marine bacterium, Pseudomonas purukawai. Here we have used the bacterial EPS as bioemulsifier, and heat treatment at 85 degrees centigrade for 50 minutes showed reduction of the emulsion forming capacity of EPS. Well, so next, what inside the natural biofilm? Yeah, so bioemulsifier, I mean this EPS gets accumulated at the interface between the two initial phases of by reducing the surface tension, and that is the mechanism through which we can harness this EPS of biofilm forming bacteria. Well, so next, the microcosm study we have conducted and performed to evaluate the efficacy of EPS in crude oil degradation, and we have obtained almost 89% degradation after 5 days. And this study suggests the involvement of EPS as an associative agent for changing the cell surface characteristics. Well, so in other studies, we have analyzed the interaction of biofilm EPS with the heavy metals. So there are several metals we have and this spectroscopy we have found the EPS of Pseudomonas aeruginosa, bacillus, and Pseudomonas pseudoalkali, that there are various functional groups, and on trapping with this metal with this EPS, there is a shift of the peak, significant peak, and then we can find out the stretching of different chemical bonds in the IR spectra. And most of the functional groups which are involved in the metal binding capacity are usually sulfate, phosphate, hydroxylamide, and carbonyl groups. And these are represented in the metal sequestration. So from all the above studies, we have proved that bacterial EPS can act as the potential adsorbent for different toxic metal ions. Well, so it is not alone the EPS, rather we can say it is an complex, and this is called the biofilm EPS complex, which in combination helps in degradation of the organic pollutants. We have also explored the role of quorum sensing system in biofilm regulation and degradation of pollutants for efficient use in bioremediation purposes. So the figure here we can see on the left panel shows the confocal laser scanning imaging, which is about the natural biofilm architecture, stained with the two different dyes to stain cell as well as the exopolysaccharide. The rod-shaped cells embedded within the dense biofilm matrix you can see. So further, EPS spectroscopy revealed the biofilm EPS presence of the different functional groups, for example, uronic acid and CH vibration, and then NH traits of primary amines, then carbonyl traits for carbohydrate, and phosphate bonds for the DNA matrix. And then when he conducted the degradation study, which is two small aromatic compounds like phenanthrene and pyrene, and we have found that Phenotrophomonas, this marine biofilm mode could degrade up to 86% in case of the phenanthrene, whereas the pyrene degradation was a little slower because of the complexity of this molecule. Well, so next what we found is the accumulation of phenolic intermediate was higher in the phenanthrene culture. It was relatively less for the pyrene, and the enhanced degradation by this Phenotrophomonas marine bacterial strain proved the prospects of marine bacterial biofilm in bioremediation of hydrophobic organic pollutants. Well, so another strain we have isolated from the coastal area of Bay of Bengal of Odisha. So it is Pseudomonas mendocina. See, this strain was studied to explore the role of biofilm density for degradation of polycyclic aromatic hydrocarbon. And so this work was focused on the expanding the role of calcium in biofilm formation and its impact on degradation of phenanthrene. We know calcium is very much abundant in the marine ecosystem. And it was found that calcium plays a triggering role in the onset of the biofilm. And in this study, we found that the biofilm formation ability of bacterium was gradually increased with increase in concentration of calcium up to 20 millimolar. And it was confirmed by confocal microscopy in the right-hand panel, where maximum biofilm density was observed increase with 20 millimolar calcium. Well, so the degradation study was conducted, and it was found that Pseudomonas mendocina marine bacterial strain was grown in different media, and it could able to degrade up to 87% of the phenanthrene in 15 days. So, and next we found the negative correlation between EPS to cell ratio and phenanthrene degradation. Hence, it is evident that biofilm with higher EPS content showed more degradation. Well, so our next work was carried out to explore the role of signaling mechanism. So we know, mentioned earlier, the quorum sensing is the signaling molecule, signaling mechanism, and the molecule is the autoinducer through which the bacterial cell gets themselves induced and form the biofilm. And how these pure signaling molecules are helpful in the degradation of pH, that was what studied here. We exogenously added this molecule, and we found there is a significant increase of this degradation efficiency. And all the autoinducer of the acyl homophene lactones, which are exogenously added, have enhanced the capacity of the bacterial cell to degrade within a shorter time and with a higher efficiency. Well, so we have learned so far that the immobilized bacteria form biofilm on suitable substratum. But next, the question is, which substrate are preferred? Is there any choice of materials? In order to find out that answer, we have studied this year. We have studied and formed biofilm on different substrata, abundantly available substrata like glass, polyethylene, steel, ceramic, and rubber. And this atomic force microscopy was performed, and the architecture of 48-hour-old biofilm was characterized. So among all the substrata, atomic force microscopy revealed maximum biofilm roughness of around 742 nanometer over the ceramic substratum, the highest among the all, which indicates the heterogeneous biofilm. And the maximum biofilm height of around 1480 nanometer was observed over the ceramic substratum with the mean height of 611 nanometer. So the ceramic substratum, was preferred by this bacterium because of the higher ratio, which provided the maximum roughness for the growth of biofilm. Hence, it was evident that the surface roughness is a crucial factor influencing the biofilm formation on the of these bacterium. So, on learning biofilm development of this bacterium, we studied the phenanthrene degradation efficiency in the independent and the co-culture condition with the other bacterial strains isolated from the marine coast. So this Pseudomonas aeruginosa could able to approximately around 74% degradation was observed. You can see here the residual phenanthrene was left here almost around the 26%. So here we found 74% degradation we have achieved, and the degradation was higher in case of co-culture, which consumed around 76% of 100 ppm phenanthrene spike in this experiment. Well, GCMS analysis we have carried out, and the metabolites or the intermediates we have identified. So derivatives of naphthalene and phthalic acid we have obtained, and this suggests the involvement of the multiple pathways for degradation of the phenanthrene by the bacteria. Well, so next, what we wanted to find out, what are the gene networks present. So we sequenced the whole genome of this strain, this marine bacterial Pseudomonas aeruginosa, to explore the mechanism inside of the polycyclic aromatic hydrocarbon metabolism. And next generation sequencing unfolded a genome size of around 6.3 megabase pair, arranged in 94 scaffolds encoding around 5,000 genes. Well, gene annotation and pathway analysis unveiled a total of 145 genes across the genome. And this study provided the deep insight the metabolic capacity of the Pseudomonas marine bacterial strain. So like the organic pollutants, inorganic pollutants also we have addressed in our laboratory, and we have performed this number of studies to find out how these marine bacteria can be able to degrade or detoxify or reduce the toxicity in the laboratory conditions. So here we have found that metals, which are of course vital, but some metals which are non-essential, they are released into the environment through anthropogenic or various other natural settings. For example, the mercury, cadmium, lead, and chromium, and various other heavy metals. And under this heavy metal stress, these bacteria adopt different strategies. that help to reduce the lethal effects of the heavy metal, for example, the flux bomb, entrapment into the metal ions, then formation of the metal cyanine protein, biotransformation to convert into less toxic compounds, for example, in case of mercury from inorganic mercury to Hg0 volatile mercury. And these mechanisms are exhibited by bacteria to resist or detoxify toxic heavy metals to amend the lead. Well, so among various toxic metals, mercury is considered as the topmost one. And to gain more insight into the mercury removal potential of marine bacterial isolates, we actually compared two strains. One is isolated from marine environment, another one from the steel industry waste. So SD43, that was the strain which was isolated from the waste of the steel industry, and the PW05, which was isolated from the coastal ecosystem, which shows we can clearly see the difference of the efficiency, where we can see the marine bacterial strain could able to remove almost 94% mercury. Well, so this mercury resistant bacterium, we have studied its volatilization efficiency. This was named as the Bacillus thuringiensis, PW05. And this foggy area in the figure confirms the mercury volatilization potential up to 10 and 50 ppm mercury supplemented in the medium with the 90% removability. This isolate was further found to volatilize mercury efficiently under different environmental parameters like pH, under different temperature, under different salinity. And the bacteria harbor the gene which is called named as the MAR-A gene, and the expression of MAR-A gene was significantly influenced by the fluctuation of the parameters like different salinity levels and upregulated at 5 ppb saline. And from this study, we concluded that marine bacterium Bacillus thuringiensis is a suitable candidate to remediate inorganic mercury in varied environmental conditions. Well, so we have learned before EPS plays a great role in organic pollutant degradation. Well, now this study was performed to explore the mercury absorption efficiency of extracted EPS. So now here we have taken out the EPS from the biofilm itself, and now we wanted to find out how this EPS can do the sequestration of the metal. Well, so in this study we have found the EPS could be able to absorb around 90% of the inorganic mercury after 2 hours of incubation. And this study confirmed the potential of bacterial EPS for mercury removal from low to high mercury contaminated media. And to decipher the nature of surface functional groups of bacterial EPS in mercury binding, we have characterized the FPR and both XRD. And in this FPR, we have this string EPS and the EPS with the mercury, and then we can find out the changes of the the shift of the peaks due to the binding of these heavy metals. And the diffractogram of crystalline sample confirms its amorphous nature, and the EPS gained a crystalline nature after treatment with the mercury. And this conversion of the amorphous to crystalline nature will be beneficial for mercury leaching process. So with increasing temperature, the Gibbs free energy value becomes less negative, which indicated the binding of EPS to Hg more preferred at room temperature. And the enthalpy change, delta H, was found to be negative, suggesting the exothermic nature of the process. And hence, we conclude that the interaction of mercury with the extracted EPS is an enthalpy-driven process. Well, next metal what we have addressed was the cadmium. Here we explored the cadmium-resistant marine bacterial strain called Pseudomonas aeruginosa, which showed the resistance up to 1000 ppm with an MIC value of 1250 ppm. Well, the bacterial biomass and EPS was examined to reveal their potential in cadmium removal, and we have found this harbors the gene called the CZC-ADC, and we have found the expression of the genes and this strain could be able to use the could be grown into the medium with suitable and optimum pH and other physicochemical parameters. Well, so next metal which we addressed was the lead. And we investigated the interaction of biofilm-associated EPS of marine bacterium with the lead and the raw integrated density. So this was the biofilm in the presence of the different concentration of the lead. So this A panel was 1, 2, 3, 4. These 4 are the pristine one without any kind of lead, and then here this is with the 100 ppm, this is 200 ppm, and then increasing concentration we have found that there is an interaction and there is an inhibition of the biofilm. But this strain actually can reveal the reduction in the aggregation potential and biofilm growth in presence of the high concentration of the lead. And then the interaction was studied with the 3D EM fluorescence imaging, and it depicted the protein-like fluorophore, single protein-like fluorophore containing the tyrosine. And here the study confirmed the presence of the fluorophore as the main substrate available for heavy metal binding in the EPA. So here in our next study, multi-metal resistant strain, by recent paper here we have reported the multi-metal resistant strain of Pseudomonas isolated from Coisa, India, Bengal of and which could able to resist lead, cadmium, and chromium as a ternary metal solution. Well, so the semi-DAX analysis confirmed the binding of the lead, metal, cadmium, and chromium, and it was observed that the optic behavior of bacterial cells in biofilm mode is greater than the planktonic mode, and the Pseudomonas chenguensis could be used for the bioremediation of the sites which are contaminated with the multiple traces of the multiple metals. Well, so our next study was to find out how we can exploit the immobilized biofilm associated with the EPS in the marine bacteria, Pseudomonas aeruginosa, and for the lead sequestration. So in this study, calcium alginate was used as an immobilizing agent, and both for live bacterial cell mass and EPS. 3 types of the absorbent beads like crystalline alginate, EPS alginate, and biofilm alginate was prepared using calcium alginate for removal of the lead ion, and we found almost the EPS alginate bead of lead followed by the more efficiently, and we can find out there is a recovery, and we can reuse the calcium alginate beads for the absorption of these metals. And the maximum bioabsorption of the lead by EPS alginate may be due to the presence of more reactive functional groups on the bacterial EPS, which have the affinity to bind lead ions. Well, so this sort of study gives us the clue that this can be reused for several times. And this confirms a strong affinity of EPS alginate bioabsorbent towards the heavy metal lead. So after revealing the metal absorption potential of immobilized bacterial biofilm EPS, we move to elucidate the metal bioaccumulation behavior of the bacterial strain. So here the protein what is involved is the cysteine-rich metallothionein protein, which are mostly involved in the intracellular sequestration of heavy metals inside the bacterial cell cycle. And these proteins are encoded by the gene named as the BMP. And this biofilm forming marine bacteria, Pseudomonas aeruginosa, N6 is a very potent strain we have isolated from this Bay of Bengal Odisha coast. And this was found to possess the BMTA gene, which conferred resistance towards many heavy metals like lead, cadmium, mercury, chromium, and zinc. And the expression of this metallothionein gene increased significantly, the increase in the lead concentration up to 800 ppm. So here you can see there was an upsurge or upregulation of the gene still up to 800 ppm. And around 11.4-fold increase in the BMTA gene was observed in 48-hour-old biofilm. So we have conducted experiment here with the planktonic cell with the biofilm cell. So planktonic of 24 hours zone, planktonic cell of 48 hours, and biofilm of 24 hours zone and biofilm of 48 hours. So this was found as the optimum one. So here biofilm of 48 hours could, here we can see that BMTA gene was better expressed compared to the other stages. So thus from this we found immense potential of this marine bacteria and biofilm layout for the degradation of various pollutants. Well, so in summary, we can say that marine biotechnology is a multidisciplinary science involving the application of marine organisms and engineering them for product and service for the benefit of mankind. And marine bacterial biofilm and associated EPAs exhibited tremendous potential in bioremediation of various organic and inorganic pollutants. And these marine bacteria may thus contribute immensely towards the world of biotechnology if the resources are explored to their maximum potential. Well, so at this outset, I would like to thank the funding agencies which generally funded my research. And these are all my graduate students, those work which I have presented here. Thank you one and all for your patience here.
Thank you, Dr. Suraj Das, for very classical studies on biofilm formations. And it is taking us to our research and graduation level research in technical institutes. And your talk is really enlightened us, particularly me, and as well as I hope other people could have enjoyed this. And we are also working on the biofilm in terms of biofouling aspects, and hope we have a lot of synergy. We will have kind of discussions in future. Thank you very much. And I think all of the participants who has promised to give a talk, all the 7 has given the talk lecture. I feel that this we can open up the panel for discussions, and I request all the speakers as well as participants to provide the views and provide their take-home points. So as a customary, may I invite Dr. Dimitri Aminin to give his views on today's deliberations.
Dr. Dimitri, I think, is not joined in. Can you just see the participants available and kindly invite them accordingly? Yes.
Yes. I invited Dr. Mohammed Hatta to give his.
Yes, Dr. Dharani sir, I am indeed happy to listen.
His audio is gone bad.
Hello, sir.
Yes, you are clear and loud.
Okay. Thank you so much, Dharani sir. I have listened to all the lectures and all the presentations. It was indeed very heartening to see some of the research, especially the last one from NIT Surathkal, Rajat Das. It was, in fact, we have contributed a book chapter to one of his book on metal bioremediation. Wonderful research. And also I've seen other people, though not much on microbiology side from the Russian team, but I've seen the work which is carried out at NIOT on deep sea microbes also. I mean, the kind of equipment for sampling, the kind of research, which are actually pretty advanced. So hopefully we will, I mean, as we have seen, the ocean is the key for the resources for looking into further I mean, exploitation may not be a very right kind of word, but the proper sustainable utilization, which definitely holds a lot of potential. And from microbiology side, as we know, it's a very hidden majority with a lot of potentialities, which could be utilized, and the collaboration, hopefully, could reveal or strike some very successful joint ventures, which could ultimately lead to product or applications or patents and things like that. So I wish all success. I thank the Ministry of Earth Sciences, CSIR, Indian Embassy in Moscow, and Russian Academy of Sciences for the opportunity. Thank you.
Thank you. Thank you, sir.
You're welcome, sir.
Okay, thank you very much. May I request Professor Dr. Alexander Kaselnikova to give our input? Dr. Alexandra Kaselnikova.
Perhaps she's not with us now, but may I say a few words on behalf of the Department for International Cooperation of the Russian Academy of Sciences. Thank you everybody for your beautiful presentations, And I would like to remind that our program is not over because tomorrow we continue with 2 more sessions. And I hope that we all hope that these 2 sessions will be not less interesting and not less important for those specialists who work in this field. So tomorrow we meet you at 9:50 by schedule in your program, and please, you are welcome.
Thank you. Thank you very much. For sure, we'll participate in tomorrow's session. And I also invite all our colleagues, all our colleague presenters to give the views. Maybe Professor Olia Fedorowski.
I'm
Yes,
sorry,
please.
for me, the questions for me?
Yeah, you can share your experience and you can give inputs on today's deliberation.
Yes, I want to thank everyone who gives their interesting reports, especially the foreign for me foreign scientists, because it's very important to share with the results of their experience with their investigations, and I hope that in future we will have some connections. Thank you very much. Thank you for the opportunity to participate in this conference.
Thank you very much. May I request Dr. Alicia Igbova to give her to share her views. If she is not able to open, can I request Dr. Suraj Das to give his views on today's deliberation?
Well, thank you, sir, and thank you once again for giving me this platform to showcase our result and share our experience. So what I have learned from your talk is that you are really doing very good work on this marine deep sea microbiology. Well, so to remind my background, I am PhD from Anavala University, CS in marine biology. I worked during my PhD in the continental slope area, which I cruised through the Sagar Sampada of this Arabian Sea and Bengal. And so once here in Odisha, we don't have much access. And after learning and listening from your talk, a really fascinating talk, and how you are studying these deep sea bacteria and the effect of all these pressure on the morphology of bacillus is really very interesting. And of course, again, I admire your talk. And nextly, Professor Hata, who is also working on this marine bacteria and described a lot of immense potential of this marine bacteria in harnessing for benefit of mankind, like drugs and various other biomolecules or peptides. So we can make a network out of this talk, and if we can exploit more sustainably these marine bacteria, we can really come up with various kind of novel biomolecules.
Yes, thank you very much, Dr. Das. May I request Dr. Marigowda to give his review?
Can I audible, sir?
Yeah, you are clear and loud.
Yes, sir. All the presentations were very nice, and we really enjoyed all the presentations. And basically, most of the research which are coming out with the novel techniques in terms of microbiology and some biomolecules which are derived from these marine bacteria and other algal forms. It was very interesting, and for these kinds of things needs to be explored further, and this gives an impetus to understand these things in depth and it was a very nice lectures. And I thank all the expert speakers on this, and I also thank the organizers and the chair. Thank you, sir.
Thank you, Dr. Marigowder, and thank you all the participants, all the speakers, and all other participants who have patiently waited until 8:30 and listened to the deliberations. As a customary, I will just take a short time to consolidate the work to the deliberations today we've done with the kind permission of chair. Today, so we have eight technical talks from various scientists from India as well as in abroad. For the first, we had a technical talk on Dr. Dimitri Amini from Bioorganic Division of Pacific Institute of Bioorganic Division. And his work is on systolic heart failure treatment. And it is very specifically working on this. He mentioned that the cardiovascular disease and the importance of the myocardial infarctions and the importance of synthetic compound and the marine natural compounds. And also he has shown some interesting on this erythromycin from streptomyces, which is having a very unusual structure of the of equivalent to synthetic compound, and that is a very potential source of getting this kind of compound from marine. And indeed, it was a very good presentation, so which has opened up a new area of research other than cancer research in myocardial infarction, myocardial disease from marine drugs, which was followed by a talk from Dr. Mohammed Hatta, Professor of Department of Marine Biology and Microbiology and Biochemistry from Cochin University of Technology, and bioprospect of marine bacteria. Of course, he has given a very fundamental idea of what are the difficulties in processing the biobacteria, marine bacteria, challenges in cultivations and non-cultural organisms, and studying the gene pool approaches for ketosynthetic genes as well as PKS type 1 and type 2 genes, and also exploration of the microbial genome to metagenomic approach for functional base as well as for gene-based approaches. And of course, his talk also very in line with the today's presentation. The third presentation was given by Professor Alexander Karselnikova on a very important aspect on aquaculture, that is on preservation of biological resources, and she specifically spoke on cryopreservation of aquatic organisms' reproductive cells. And it is interesting to note that she has worked on the species like sturgeon and other commercially important freshwater species, the cryo protections and the challenges involved in cryo protections, and the specific protocols used for specific species. And I think this would be a very important steps in preservation as well as promotion of aquaculture. The fourth talk was given by Dr. Marigowder from NCCR, Ministry of Affairs of India. He spoke about the Indian water quality parameters and quality standards, and also the designation of seawater for different criteria, and a whole lot of work done in sanitization of heavy metals and other toxic substance standards provided. And it was a very elaborate lecture on the environmental aspect of the seawater. The fifth lecture was followed by the fifth lecture was given by Professor Olia Fedorski from Astrakhan State Technical University, alternate source of protein and raw material for fish feed. It is a very, very essential and very hot topic today because of the growing aquaculture field and the demand for marine finfish protein and the increasing per capita consumption demand, and the requirement is very huge. And all this demand has to be met by only aquaculture. Once we want to do the aquaculture, the feed is one of the very important ingredients. Though there are a lot of difficulties in development of an alternate source, because the marine fish that is particularly carnivorous fish giving an alternate protein is a very different task, and the whole world, including India, Russia, and China, are working on exploitation of deep sea fishes, demersal fishes, as well as krills from Antarctica for this kind of approach. And your approach for alternate fishes with krill and as well as seaweeds, as well as waste, and this sapropole are very much interesting. And I hope in a short time we will have alternate support systems for to support the fish meal fish meal requirement in the aquaculture industry. Of course, the fifth topic is given by me. That is from National Ocean Technology. We have shared experience on the deep sea microbes, the technological challenges involved in isolation, sampling, and culturing of these things, and the importance of studying the fecal tolerance microbes, which is very specific, and as well as studying the non-cultivable genome and through metagenomic approach by functional and gene-based exploration. The seventh presentation was given by Dr. Alicia Igbower, a senior lecturer from Department of Technology, Goods and Commodity from Astrakhan State University. Is signed on implementation of collagen product. Collagen is a very huge industry today, from hide to variety of cosmetic products to the health products. Collagen is very important, and her findings on alternate collagens and cheap collagens from fish and marine resources. Of course, to bring the knowledge, there are collagen-like molecules from lower organisms, even from marine lower crustaceans, even from bacteria, and your work on identification of collagens from cheap sources and the formulation of those things will be a very high demand work for future collagen and for a lot of cosmetic surgeries as well as replacement work in skin and other cosmetic surgeries. The last presentations were released to us presented by Suraj Das, is biofilm, and biofilm formation is one of the very fundamental for biofouling, and which is $1 million industry in biofouling in marine structures, which you use seawater as coolant or use as in seawater as transport medium. All the ships as well as the power plants and other things face a huge problem of biofouling due to biofilm formation, as well as he has also talked about the importance of biofilm in terms of production and the degradation of toxic pollutants, including heavy metals and other chemicals, which is going to be a very important topic for our industrial expansion and to meet our treatment and living standards of the sea and to maintain this. With all these things, really, we had a wonderful presentations, and it is really breathtaking to understand different peoples from different locations of India as well as in Russia as working on more or less relevant area, and there is a lot of synergy between all the institutes and all the works, and there is always an opportunity to interact, and I hope the organizers of the Ministry of Earth Science and Russian Science Federation and the Embassy of Russia and Indian Russia will do a knitting work to integrate these people together. And of course, on behalf of all the speakers and the participants, I take this opportunity to thank the Ministry of Earth Science, the Russian Federation, as well as the High Commission of Embassy of Russia and India for making this wonderful opportunity and allowing us to express our views and express our findings and share our research works. And also take this opportunity to thank particularly Dr. Sisir sir for his meticulous arrangement and continuous follow-up and providing all small input and everyday input. Thank you very much, sir. And thank you very much, friends. And I take this opportunity and also sincerely request all of the participants who have given the presentations to send a small write-up and maybe a couple of lines or 4 or 5 lines or a paragraph about the today's deliberation and your work as well as today's deliberation to consolidate the work and consolidate and prepare a report on today's deliberations. And with these things, I thank you everyone, one and all. And of course, it is late in India. It is nearly 8:30, nearly 9 o'clock. And I thank all the participants for staying late night and sharing their views. And thank you very much. Thank you, sir, for the wonderful opportunity. Thank you very much, sir. Namaskar. I request Dr. Sisir sir to formally close the session.